Oh, that's true. Yeah.
In the lab, where data flows, revolutions underway, bridging gaps, set to show ace power in full display. Flagship. In the lab, where data flows, revolutions underway, bridging gaps, set to show ace power in full display. Flagship sets, cornerstone, a future so bright, where machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Across lands, hand in hand, we craft new reality. Software, standards unify, language shared and bold. Data's truth, we amplify, story yet untold. Fair principles our guide, accessible and free. In this journey, side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein, an artery bridging minds. Future grows in this AI quest. Workforce trained, ready, keen across AI divides. Biomedical scenes I've seen, world forever wide. With each step we close gap between machines and human grace. Between machines and human grace. Bridge your white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. In AI we trust, in AI we must. In unity we find future just.
Good morning. All right. How's the song? Does it give you a dose of energy toward the end, right? Okay. Well, it turned out that that was an AI composed song, and we played it at the NIH Bridge to AI meeting, and we won an award. So in recognition of that, and I'm wearing this music pin. All right. I'm your host. My name is Jake Chen. Welcome everyone to ATTIS 2024. ATTIS is in the in the 8th year, and it stands for Annual Translational and Transformative Informatics Symposium. We have a great program ahead, and I want to kick out this year's meeting with a few comments and introduction. Okay. ATTIS has really grown to become something of impact in the Southeast regions. So ATTIS started, the first meeting started in the library. And I still remember we were in the edge of chaos and it was in the library floor. Dr. Semino and I were getting started with the Informatics Institute. We thought that the proper topic would be about forging new bioinformatics collaborations. And at the time, everyone in the country were already getting ahead with topics such as single cell analysis and sequencing. And we seemed to be a little behind back then. But over the years, and we grew in 2018 and 19, we talk about the collaborations, opportunity to engage researchers in the medical school and cancer center on campus. In 2020, we were the first in the nation to organize ATTIS built around the theme of COVID when COVID was just starting. In 2021, 22, and 23, we explored the topics of cancer data science, genomics, and last year, AI in medicine. So rightfully, this year, we picked a new great topic. That's drug discovery meets AI. I'm very proud to say that there are more than 160 registered participants this year. Some of them may be very digitally abled and they may be hiding somewhere and tuning into the webcast. So we do have a webcast option. They're coming from many parts of the country, from UAB, from Alabama, Arkansas, and even within the UAB, and I'm just appalled to see that there's a rich set of skill sets and people are coming from different departments. And welcome. I hope that you all enjoy this feast of a topic, which can also be new to informaticians. All right, we could not have done this ATTIS organization without the help of all of these people who serve on the program committee. And since the very beginning of this year, the program committee met every other week, and we talk about who to invite, what topic, what sessions, how do we determine the lens, and it was a lot of hard work. If you are one of the program committee member, would you please stand up and then people can recognize you? Thank you. And then we also have staffs and the student volunteers. They are actually running around to making sure that they will have audios and video capabilities. We'll have great food outside. And there's drinks and the food. And where's the restroom? I just want to point out the direction. It's in the other side, right? So during the break time, go out and enjoy any coffees and drinks. If there's any problem, you can just go grab one of them and talk. And we have student volunteers who help with the registration as well. Okay. If you do decide to walk around the campus, and this is right in the middle of the medical school side of the UAB, and it's lots of things going on within 2 or 3 blocks of walking distance. We also have features that you can use on social. If you want to use X, and the channel that we have is a SPARC UAB. You can use the handle ATTIS. We have a YouTube channel, and right now is being webcast. If for some reason that you have to go to your office and leave here, you can continue to watch it on YouTube. You can add comments. We may not be able to interact with you over those comments, but it will be actually recorded. And also, if after the meeting, if you like to stay in touch for future events or webinar opportunity that we may have, feel free to join the Google groups. We also have a Slack channel, and we have already automatically invited all the speakers to the Slack channel. But anyone is welcome to join the Slack to continue the conversations. If you're curious about the most up-to-date program, you just go to attis.ubright.org/program, and then you can get the program on the web page, and also you can download it. Now, one interesting thing that we are experimenting is the audience interaction features. So we have a live polling function. You can scan this QR code, bring out your phone, scan it, and throughout the break, we're gonna ask some questions, and then you can just answer them and then see the results live. Or you can go to slido.com and enter the hashtag or actually the word ATTIS in there. Okay, so this is an overview of the program. In a few minutes, I'm gonna introduce our keynote speaker. After that, there will be a coffee break. There will be research sessions, and the flash talks. The talks are at different lengths that features both late-breaking research results and the trainees who's experimenting with the topics in this area. And we'll have a box lunch outside during the 1-hour break. If you're one of the judges, and you should find Amy, who's sitting in the back, right? And she has a form, and you can just write down your evaluation score, because we're gonna actually have poster award. So everyone, please enjoy the box lunch and look at the posters. Coming back, and we'll have a computational drug discovery workshop. After that, then there's a coffee break, and the panel discussion is gonna be absolutely exciting. We have very diverse group of people coming from different disciplines and industry, and they're gonna talk about AI in drug discovery. Is it hype or hope? And followed by research sessions. A short after the award ceremony, a short closing, then we can have a party and social networking. You can stay as long as the janitor kick you out. All right. So without further ado, let me introduce our keynote speaker. Our keynote speaker cannot be here with us because he's based in Hong Kong, from far away from the other side of the earth. Dr. Alex Zhavoronkov. He's the founder and CEO of In Silico Medicine. And we're really excited about giving a speech, not only because he has founded a very high-profile company, one of the first examples of AI drug discovery companies in Asia, or among the first companies in the world, but also because his huge commercial success. He has raised $600 million of funding to showcase what AI is capable of doing in the real-world drug discovery space. And he had pushed 18 preclinical candidates, and for the short time of 3+ years, and he has 7 human clinical trials. And on top of that, Alex is actually very, very strong competitor in terms of the research. And he's considered to be one of the top AI leaders in drug discovery and advanced healthcare, one of the highly cited scientists. He has written books, and I have actually separately read his blogs on Forbes.com. He is one of the leading visionary person who is among the first, and actually when the ChatGPT came about, I remember he probably has the only author with ChatGPT in one of these articles. But actually, I got connected with Alex, I think in 2017 or 2018 in Basel. And I remember seeing a very young, talented person fresh out of Oxford. And I remember talking to her and realized that there's a new company being formed. And then I was introduced to Alex, and Alex was very energetic. And he has lots and lots of really interesting information that is very different from those people in the conventional drug discovery paradigm. So without further ado, let's welcome Alex.
Thank you very much for such a generous introduction, Dr. Chen. It was a true privilege to meet you in Basel many years ago. Basel, by the way, is the heart of the pharmaceutical industry. That's where Roche and Novartis are headquartered. And in the past, we used to run the Aging Research and Drug Discovery conferences in Basel every year. Now it moved to Copenhagen. So for me, it's a true privilege to be presenting to you today. And I apologize for doing it virtually. I really wish I could be there. I'm actually dialing in from Abu Dhabi today, which is in the United Arab Emirates. And we also have about 70 scientists here in this area. We are truly global. For us, if you are good at what you do, it doesn't matter where you're from. If you are peaceful, and doesn't matter if you have antenna on your head, please come and work for InSilico. I hope you can see my screen, and it's again very true honor to be presenting here today. I really enjoyed the song, so congratulations on winning the award for the song. Kind of shameless promotion of our conference that we co-organize in Denmark at the University of Copenhagen. I usually start with a slide so people don't forget about it. So take a picture if you're interested in aging and the convergence of multiple technologies that are designed to extend human productive longevity. Please come to this conference as the largest one in the world. Many pharmaceutical companies are there. We're trying to explain to the pharmaceutical industry that if the drug is powerful enough to work on aging, it might work in multiple diseases. By the way, the Copenhagen is also the birthplace of GLP-1 inhibitors that many of you are probably using for weight loss management. InSilico is a truly global company. Most of you know us for our work in drug discovery. We have two headquarters in New York, which is now moving to Boston, and in Hong Kong. So we try to span the entire globe and focus on generative technologies, predominantly for target identification, small molecule design, prediction of clinical trials. This year we were selected as the Fast Company's most innovative biotech company in 2024. We were very, very pleased to see that because we're actually not a biotech. We are more of a technology company, but we were very happy to see that now we're also recognized as a biotechnology company. So this is what InSilico is known for. So we have generative AI that covers multiple domains, predominantly drug discovery. So we have small molecule drug discovery tools. We have software for biologics design. We're very heavily involved in biology software. A lot of scientists worldwide are using our PandaOmics platform. We have drugs in clinical trials, and most importantly, the community of the software users, including in silico employees, provide us with proprietary usage and validation data. So we see which models work and which don't. So having a large community of users around your software, it's extremely important because you actually try to protect yourself from their data. We're not using people's data. However, we use this community for validating models. If you are to do it in-house, it will cost you billions of dollars. If the community is validating new models and synthesizing and testing, that becomes much cheaper. It's community effort. If any of you are not familiar with agents yet, I highly recommend getting into this field. So about half a year ago, we transformed the company into an agent-managed company. So we actually have, for every human employee, we have 5 to 10 AI agents. An agent could be viewed as an instance of a large language model that is adapted for a specific task. And many of those agents run model orchestra, the models that are specifically built for specific tasks, like, for example, a deep neural network for target selection, or a deep neural network for the generation of small molecule chemistry, many predictive models, generative models. Nowadays, the LLMs become very good at reasoning. They are not great at solving very specific domain problems, like, for example, quantum chemistry computation. However, they can understand the tools and they can run those tools. So now you can actually replace a lot of your human workforce with agents. Currently, we have about 450 people, but now we have a huge army of agents. And we're also very involved in aging research. I think that you should not be thinking about AI if you are not thinking about aging. And you can actually think about aging as processes that are transpiring in time. So you need to teach your generative AI systems to understand how things change in time. And for that, you need longitudinal data. That's why we work very heavily in aging. And now it's not some kind of weird ESG initiative that we are decided to go after because the investors or the general public want you to. We realized that the same algorithms that we can use for design of small molecule drugs can also be used in agriculture to design very efficient herbicides and pesticides, and also in sustainability initiatives like CO2 capture and novel material design. So now we are also working in that area quite heavily, and you should see a few papers from us in the next 12 months in those areas. So again, we are not only biotech, we do nutrition, skincare and beauty, crop protection, sustainable crop protection, and also sustainability and longevity of materials. If you actually want to select a career in sustainable agriculture, and highly recommend going into Syngenta, one of the leaders in crop protection, and we're collaborating with them on the development of new sustainable agriculture solutions. But now I just wanted to tell you that we're not only drug discovery, we're mostly like a tech company. However, I'm going to switch gears to drug discovery and talk about how generative AI is transforming drug discovery. And my job is to convince you that whatever you are doing right now, whatever career you're planning, just drop everything and just get into this game, because this will change everything. Currently, you can convert time into money, but you cannot convert money into time. AI-powered drug discovery with a focus on human longevity will help people convert money into time. So now our life may get a little bit longer, maybe a lot longer, and I really hope that all of you are going to join this game and make a lot of money and time for the human population. Drug discovery, in my mind, is a molecular casino. It's a casino where one spin of the wheel may take several years. You need to spin several times, and each bet costs you millions, sometimes hundreds of millions of dollars. And if you account for the probability of success at every spin, you lose 98% of the time if you start early. If you start later, of course, the probabilities of success are increasing. So nowadays, there is a lot of discussion about, for example, limiting the ability of new countries and nations to go into AI-powered drug discovery. Most of those people who want that, they don't even know what they're talking about, because you actually need to have more players betting on novel drugs in order to increase the probability of success. So I hope that this area will be off the table for any kind of geopolitics, because it's the most altruistic area, but at the same time, it is also the most difficult area. Takes, again, 11, 12 years to get a drug on the market, and most of the time you fail, and it costs you billions. Very few people understand drug discovery. And by the way, I certainly don't. So I spent 12 years in the field, but I am still learning new tricks. And sometimes you look at the bird's eye view and you realize that you don't know anything. And here are some scary estimates. So in the 80s and, well, 70s, 80s, 90s, the FDA started asking companies about the mechanism of action for their drugs. So you actually have to explain why your drug works. And that led to the development of targeted therapies. So instead of just saying, okay, well, this drug, I give it to this patient and they get better, and here are the clinical results. Now you need to explain why the drug and how the drug works at the molecular level. So that led to the explosion in targeted therapy. So now people started doing rational drug design, trying to understand what is the key driving force behind the certain disease, and what are the key features that are within this driving force, right? So for example, you identify a pathway, narrow it down to a specific target, and then start designing a drug that inhibits or upregulates that specific target. So the targeted drug therapy is actually a reasonably new thing in pharmacology. We're talking about maybe 60 years time. About 20 to 30 drugs are usually approved by the FDA every year. Targeted drugs using small molecules, approximately about 50, 55 drugs are approved by the FDA in general a year. So it's actually a very rare event. About 50 to 60% of those drugs are targeted. So we're talking about 10 to 18 small molecule targeted drugs a year. And my estimate is that approximately 200 to 400, depending on how you count, targeted drugs are approved by the FDA. Again, the assumption, I have another assumption that most of those drugs that are getting approved are actually representations of something old, right? So usually we already have some known inhibitors of a specific target, and the companies try to minimize the risk and try to go after the same target, maybe with a little bit better molecule, going after a specific, different mutation, for example. So expanding the breadth of the application of the drug. So about 10 to 15% of the approvals are novel drugs. So if you actually think about the net totality of all the novel drugs on the market, we're talking about maybe 80 to 100 truly novel targeted small molecule therapeutics, where you have a novel target, novel molecule, all the way to US FDA approval. So the toolkit that the doctors have for targeted therapies is actually very small. Of course, we can argue about this number. It might be bigger, but the numbers are very small. So if in your career you actually manage to get a drug approved, you're a hero. You have less truly novel targeted small molecule drugs approved than there are Nobel laureates in physics, right? Even those alive. Even most countries in the world have never discovered a truly novel drug. Recently I actually asked myself a question: did China ever discover the drug with a truly novel target, truly novel molecule approved by the FDA? Right? Actually, if you go to ChatGPT and ask those same questions, after some argument, you will see that the level of novelty of those that were approved was actually pretty low. And China, of course, participated in the discovery of many drugs. Using contract research organizations, I would say probably they touched 80% or 90% of all the drugs that got approved. However, what about the genuine discovery? And I recently did a poll on my Twitter. Feel free to follow me on Twitter or LinkedIn. I have very nice following there, mostly PhDs interested in longevity or chemistry or biology. And if you ask the community, you ask, okay, well, has China ever discovered a drug? Explain your answer. Most of the people say no, and those that said yes couldn't really provide a good example. So maybe there is one or two. However, it's actually a very, very rare event, even for a huge country with huge infrastructure, to really deliver a novel targeted therapeutic on the market. So the reason I'm telling you this is because if you are going into this field, your ultimate objective function should be to get your drug approved. We all like research papers. We all like really nice headlines. We like software on the market. We like to get paid by the pharmaceutical companies. But if you don't have this as your ultimate light at the end of the tunnel, you're going to fail more often than you succeed. So I think that everybody should have this objective function in their mind. Even the countries, very often leaders of nations ask me, okay, Alex, we want to build a platform locally in the country to get into biotechnology, and we need to build infrastructure, etc. I'm saying, no, don't think about this like that. Just think about one objective. Let's say within 10 years, develop 5 truly novel targeted therapeutics and put them through the FDA, right? If that is your objective function, everything else will fall into place. We shouldn't think about creating platforms, about how to build the infrastructure, the community. We actually need to put ourselves in a situation where we have a very clear objective that is quantifiable. And to get to this point, to get the drug approved, you need to understand the process. This particular slide comes from Stephen Paul's paper, which is very famous, published in 2010 in Nature Reviews Drug Discovery. So pull this paper and make sure that it's your next article that you're going to read. It's still very powerful after 14 years. And since the publication of this paper, the drug discovery process actually got more expensive. The new estimates went from 1.8 billion from target to submission to 6.1 billion for big pharmaceutical companies. There are wonderful academics, Alexander Schumacher and Oliver Gussman. They call themselves the St. Gallen Consortium, that really go deep into the costs and probabilities of success of multiple drugs. They look at the entire market. And they just recently published a paper showing that big pharmaceutical companies are so inefficient that it costs more than $6 billion in R&D spend to put one drug on the market. And usually one big pharma puts one a year. Novartis is the most efficient over the past 20 years. They used to put one a year innovative drug on the market. But what is more important is target discovery and disease hypothesis. For many diseases, we still don't know the target. We still don't have a very good hypothesis. For example, look at Alzheimer's. We still don't have a very good hypothesis. We still argue why it happens and what's driving it and how to treat it. So if you are to bet on one target for Alzheimer's, it would cost you probably more than $6 billion to get a drug on the market. And the probability of failure at the early stages is going to be even higher because you're going to fail in multiple models. So on this slide, you can see the probability of success in the first row, number of years it takes in the second row, and the number of millions of dollars at each stage that you will be required to raise in order to get the drug approved. Actually, those numbers are much smaller if you are looking at an individual program. However, if you are running a large pharmaceutical company, those numbers will be close to reality. And you also need to understand that AI is, of course, a very important tool for many steps of drug discovery and development. And many of the preclinical studies and clinical studies could be even replaced by AI, but you cannot really replace everything. And you need to understand those steps that are required to validate a biological target, to generate a small molecule chemical or a biologic that will hit this target. And then you need to understand the safety requirements, what you need to do to establish basic safety in animals, and then go into humans. Phase 1, it's again mostly safety. Phase 2 is safety and efficacy. And phase 3 is safety and efficacy at a large scale. So many of these steps have hundreds of different components that you need to think about. And even myself, I don't understand most of them. Here are just some that I've highlighted where AI can help, but you still need to perform a lot of experiments. And I would also make an argument today that integrated end-to-end generative AI systems with access to experimental infrastructure and significant funding can accelerate pharma R&D. You actually just recently probably saw the announcement of Syra, or I'm not sure how to pronounce this company's name, XAIARA, that raised $1 billion and brought in a large number of big names from the pharmaceutical industry to actually go after this with $1 billion in seed funding, right? You've never seen startups being funded at that level. In our case, we actually had to go through many rounds of funding. At every step, we had to show validation. But actually, it's a good idea to start new companies with very large amounts of funding. And what InSilico has done over the years, we've developed a system that covers pretty much every step of pharma R&D. We have a system called Biology 42, which integrates multiple tools. The most famous one is called PandaOmics. A lot of academics use it. You can actually apply for a free license and try it for your experiment. It utilizes expression, methylation, proteomics data, multi-omics data to formulate the hypothesis of disease and very rapidly identify novel targets. Now we are also going to introduce a really cool tool in PandaOmics, where after you completed your research, it will help you write your research paper very quickly in the format of the journal that you are targeting. So have a look and maybe write an experimental paper. We can give you some free trial access. But the workhorse of InSilico is a system called Chemistry 42 Generative Chemistry. It's a generative chemistry system that we've developed in 2019 and then expanded dramatically that integrates 42 generative engines and more than 500 predictive engines. Currently, 10 out of the top 20 pharmaceutical companies are using this tool since we launched, so it's very rapid adoption. And even that is not enough. We also have a system called Alchemistry, which helps you fit and dock the generated molecules into your protein of interest using physics-based approach. And we also have in Clinico, that system is usually used by hedge funds and banks to predict the outcomes of clinical trials using artificial intelligence. It's also many models. And usually pharma companies buy these systems separately because they have separate department for biology, separate department for chemistry, separate department for medicine. Internally, we use them all together. And we demonstrated over the past few years, this is actually the most important slide, that we can use this internal tool that we make commercially available to very rapidly convert money into drugs. So you basically use the software, give it a little bit of money, and put it on top of the infrastructure that helps you validate the results that are coming out of AI to develop a very sophisticated pipeline of therapeutic products that are marketable so you can sell them, or you can push them forward yourself as far as possible. And at every time they go through the next stage of validation, they become more valuable and more expensive, or you fail. And every time you fail or you go forward, you actually validate an engine from your software. So this is kind of a virtuous cycle of design, test, validate, get the data, design again. And at Insilico, we have two CEOs. So if I were running the company just by myself, I would probably fail. So just for most of you some background, when we started in 2014, originally in Baltimore at the Emerging Technology Centers at Johns Hopkins, there were probably 500 competitors in the field. So nobody was so dedicated to deep learning, but there were a lot of folks that were starting up. Most of them AI scientists or biotechnology leaders. Around 2018, we had more than 2,000 competitors. Right now, the industry has consolidated dramatically and many of them died. So even some of the buzzier words, buzzier names, I'm not going to name them, they recently went through massive restructurings, firings, and pretty much nobody managed to get into phase 2 with an AI-generated drug. Why? Well, because they were run either by biotechnology company executives that don't understand AI, or by people like me who understand the tech but don't understand the biotechnology at the level that is required to push the drugs forward. So around 2020, I met Dr. Ren, 11 years at GSK, Ph.D. from Harvard, and he ran a large contract research organization that actually did the same thing as a service for the other companies. But he didn't want to do, you can also refer to that as slave labor, because companies just tell you what to do, right? And you discover drugs using your current methods. Usually CROs don't have the cutting edge of the cutting edge biotechnology companies or AI companies. And he really wanted to do something great. So he joined me in 2021. And together we started using AI to advance the pipeline. And since then, 18 preclinical candidates and some of the case studies that I'm going to show you. But he is the real hero. I am mostly inventing AI products, putting them on the market, testing them, publishing them, creating communities around them. However, the real hero is Dr. Ren, who is using those tools and developing new tools to actually put them into the clinical trials. And our objective is to get the drugs approved. However, we have not done that yet. So far in our industry, I don't know of a single company that went through phase 2 successfully with an AI-generated drug. Currently, we are in phase 2. And some case studies now. So first of all, generative AI is not new and it's not magical, but it enables record-setting productivity. And it just recently got commercialized and consumerized by ChatGPT. Many of you who are in the field know this timeline. So around 2014, Ian Goodfellow and Yoshua Bengio published GANs, Generative Adversarial Networks. That's when I got into the generative field. Then Transformers were published around 2017 with a big paper, Attention is All You Need. And then you've got diffusion models, you've got many variations and permutations of those technologies, but the key technology would be GAN, transformer, diffusion, and the variations on the subject. Here is a very quick rundown of my lecture I presented at Hotel Coronado in 2017 when generative was in its infancy. And a lot of people were criticizing me at that time, and Dr. Chen probably also remembers in Basel, a lot of medicinal chemists were very critical.
Good area for
And
AI
to
disrupt.
presenting the
And there are two ways, two strategies for AI-driven innovation in pharma to ensure that you get better molecules and much faster approvals. So one is looking for a needle in a haystack, and another one is creating a new needle. So our company developed a pretty comprehensive pipeline that utilizes both of those strategies. We start with a large numbers of molecules, large numbers of hypotheses, and then take it through a process.
Yeah, so I just wanted to show you this fun story. So I had a different haircut and was much younger back then, but we didn't have any drugs in the clinic at that time. We didn't have a single preclinical candidate, but we were publishing quite a bit. So our first paper on GANs for small molecule chemistry was in 2016, called The Cornucopia of Meaningful Leads. Then adversarial threshold neural computer, reinforced adversarial neural computer, entangled conditional autoencoder. So here in 2018, in August, we published our first validation of an AI-generated drug. So we synthesized and tested. Also got some funding. After one of the CROs saw the experimental results. And then in 2019, we published our most famous paper to date on generative tensorial reinforcement learning. And this is the paper, Nature Biotech. It took a year in review. So we did this work in 2018 in the summer, submitted to Nature, it got rejected. We submitted it to Nature Biotech, and then it took a year, almost a year in review. This paper was done with Alana Spuru-Gudzik, who many of you probably know. If you are in the field, all of you know him because he was a very famous prof at Harvard. We competed. Then he moved to the University of Toronto. We became best buddies. By the way, when you are visiting his lab, usually he will give you a mask like this to wear, Lucha Libre masks. I think he is probably top 1, top 2, top 3 in the world in generative chemistry in academia, and also in quantum enabled chemistry. So in this paper, and it became very, very famous when it got published after many, many rounds of review, after we repeated many experiments. It got very high altmetric. And in this paper, we got an arbitrary target that had very specific criteria. So we didn't want to go after very high novelty. So the partner who pulled the target out of the hat had several characteristics for the target. So it had to be a kinase, had to be not a super novel kinase, but also not something super old like JAK inhibitors or TYK2s or some other very, very well-known kinases. So in 21 days, we used generative tensorial reinforcement learning algorithm to come up with multiple structures. We narrowed them down to 6 that could be synthesized very quickly. So of course, if you are including synthetic accessibility as a parameter, you are dropping the novelty component, right? So you need to go for something that is less novel. Out of those 6, 4 worked. 2 of them we took all the way up to metabolic stability assays and macrosomal stability assays. And 1 of them we took into mice. By the way, every time you do this, you need to synthesize, and synthesis costs money. So this entire experiment costed us, I think, less than 200,000. But still, it's a lot. For an academic, it would be a big thing. And of course, for a startup, at that time, we were not very well funded. It was also a pretty big thing. But in 46 days, we showed that out of generative, we could go very quickly into mice. And at that time, some of the more classical computational medicinal chemists, who I actually deeply admired, they criticized this paper saying, oh, those molecules are not very novel. Human could have done it as well. But they didn't really think about the scalability of this model, right? So you could scale it, you could improve it. And here it's a proof of concept that we actually put out for everybody to see. Internally, we had much more powerful tools at the time. But it was a really great demonstration. And then we continued with those demos. We also showed that we can take a novel target So in 2021, 22, sorry, we decided to go and test our entire pipeline in one go. So decided to go after hepatocellular carcinoma and gave it to Pandaomics, gave a bunch of data sets to Pandaomics, identified a bunch of targets that scored highly in hepatocellular carcinoma in a very specific population cohort, identified some targets that didn't have crystal structure, took CDK20 as one of those dark targets, so to speak, used AlphaFold 2 structure that was already generated. I gave it to chemistry 42. By the way, AlphaFold at that time and even today, it's not exactly your best starting point for a crystal, right? If you are doing a real drug discovery program, you would always go and do an experimental crystal because it costs you $50,000, right, max, or plus minus. So if you are to pour millions of dollars into a real program, you probably want to have a real experimental crystal. But here we just wanted to make the point so that we use 3 generative tools, including AlphaFold. I used the AlphaFold predictive structure for CDK20, gave it to chemistry 42, synthesized several molecules, got to 7.3 micromolar, 7,300 nanomolars, and then took some of the best hits, gave it back to chemistry 42, used template-based design. Before we used structure-based design, and got to 180 nanomolars. And very quickly synthesized, tested, and it worked. And we published this paper with Michael Levitt and Lance Progudzik, very famous paper in Chemical Science. Please have a look if you want to see how all of those 3 systems, Pandaomics, chemistry 42, and clinical work in unison. The study took about 50 days. So just so you know, the real experimental work, 50 days, which is pretty cool. And then it took, again, about a year to publish. And we tried Nature, but Chemical Science understood the paper. And this is the very recent paper, probably the best case study to date that I know of an AI-powered target discovery exercise and small molecule chemistry exercise. Also took us almost a year to publish here. Published March 8th, so it's just over a month now. And in this paper, we used chemistry 42, so many generative models to design novel small molecules for a target called TNIC. It's a very novel target, now less novel than when we discovered it. Only 140 papers on PubMed talking about it, and nothing in the clinic. And it also became a very famous paper, not as famous as our DDR1 kinase paper, because at that time, generative was very unique. And we also put some effort into promotion. Here we didn't. And in this paper, we actually show the entire drug discovery and development process all the way to phase 1 complete, but also with the target discovery exercise in it. So we show how in 2019, we used Pandaomics to discover a target, prioritize it. There we used a very new technique that I invented called hallmarks of aging assessment. So we actually scored the different targets that scored high in Pandaomics in fibrosis by the hallmarks of aging. So the hypothesis is, again, if the target is very powerful and it works in aging, it should work in a bunch of diseases, not only in one, and has very substantial indication expansion, because currently nothing works in human aging, as you know, except for maybe rapamycin, but we still don't have a clinical study. And then we used chemistry 42 after we identified TNIC to generate and synthesize the compound called 055. So we only synthesized 79 molecules in the study. And 055 is the 55th molecule that we synthesized. Usually, and again, this is kind of the rule of thumb, the pharmaceutical companies synthesize around 1,000 to 2,500 molecules per program on average. Now, maybe less. If you look at AI-powered drug discovery companies that claim that they are AI-powered drug discovery, usually they say that they synthesize about 500. So here, for us, we synthesize usually under 70 molecules per program. So here it was 79 molecules. By the way, just some economics for you. Synthesis of one molecule, depending on the complexity of the molecule, costs you about $2,500 to $7,000. So you can easily say it's $5,000 per molecule, maybe $4,000 per molecule with enzymatics. If you want to scale up and do more complex experiments and go into mice, it will cost you much more. Just some kind of tips for all of you who are in this field. In vitro validation, we've performed dozens of experiments leading to 3 preclinical candidates, in vivo, in vitro experiments, dozens of animal experiments. We nominated 3 preclinical candidates for this program, one for lung fibrosis, one for kidney fibrosis, and one for lung fibrosis using inhalation formulation. So for that, you actually need to reformulate your drug. And we also released the data for skin fibrosis here. We also used our robotics facility to show indication expansion. So this drug can work, in my opinion, in multiple diseases, in multiple age-related diseases. And since the target was new and we didn't know the on-target toxicity profile, we actually decided to do a small pilot. So phase 0 in Australia. So 8 healthy volunteers completed during COVID time in April 2022. And then the 2 full-blown phase 1 studies in healthy volunteers in China and in New Zealand. So 73 patients in New Zealand, 48 patients, healthy volunteers, sorry, in China. And then we started 2 phase 2s. One in China, one in the US, 60 patients each. Again, primary outcome is going to be safety, secondary outcome is efficacy. So all of this data, all the way up until phase 1 complete, is in the paper. I have not seen another company do this before. And when we submit it, since it's a very comprehensive paper, includes AI component, biology AI, chemistry AI, lots of preclinical work, lots of biology work, clinical studies. It's actually very difficult to review. That's why it took almost a year, right? And some reviewers were very difficult, so to speak. So we actually, for that, we created a tool that allows reviewers to talk to the paper. So nowadays it's pretty commonplace, but in 2023, mid-year, it was actually pretty new. So if you go to papers.insilico.gpt, you can actually talk to the paper and ask whether a certain experiment was performed, if the target is new, how does this target compare to other targets, et cetera, et cetera, et cetera. So you can actually talk to the paper. And there we also created a tool that allows you to see the primary source where the answer is coming from, and also show you the segment of the paper where the information is present. So pretty cool. That kind of gave us the idea to develop a tool for automated paper writing for PandaOmics. So now we have almost a year of experience in that. And now another example, another case study. So can we take a known target and purpose it to a novel indication with a novel molecule? So there is a bunch of targets that we already know. We know the on-target toxicity. We know the some even cumulative toxicity for the pathway. We know the side effects, and usually drug hunters have very limited resources to try their drug in a specific indication. So many indications remain untapped, and it's very important to recover some of the targets for new indications. So currently, this is a diagram of what we have done with Ph.D. 1/2 inhibitor. Ph.D. 1/2 is HIF1 alpha, a very famous target in cancer, in multiple other diseases. It's a hypoxic target. So we used our biology 42 system to prioritize this target for IBD, inflammatory bowel disease. We realized that this target, if inhibited, it leads to barrier repair function, epithelial barrier, mucosal barrier repair. And if you put it into inflammatory bowel disease, it might help close those lesions that you've got in your gut and repair the barrier. So automatically you get a bonus of anti-inflammatory function and many, many others. However, if you administer an inhibitor of this target systemically, there are lots of side effects, right? So we decided to design a molecule that is gut restricted, that works in the gut but doesn't go into the blood. And I used chemistry 42 to achieve those very specific properties in the small molecule. Did the in vitro studies, so many different studies demonstrating barrier repair, did in vivo efficacy in different animals, and then we did PK and tox study in multiple animals, including monkeys and dogs. And actually here, the idea is to show that the molecule doesn't get into the blood, right? So now you actually have questions like, how do you calibrate the dose if you don't have blood PK? But we found alternative approaches, and currently it is in phase 1 human clinical trial. So hopefully this paper gets to see the light and doesn't spend another year in review. But this is a case study that we've already completed. And with this Ph.D. 1,2 inhibitor, we achieved these properties that you would not be able to achieve just by screening molecular libraries or doing fragment-based design. We tried. And again, the most important feature is that it has epithelial barrier repair and inflammatory properties, and it has very exceptional safety profile. Now, can we use AI to repurpose drugs? I know I don't want to go over time. I think I've got another 15 minutes, right? But this is a really fun study that we did over the past 2 years. 2022 in March, I came to the Answer ALS Consortium meeting at Johns Hopkins. It's a very tiny meeting. We're organized by Merit Chutkowitz, the chair of neurology at Harvard MGH, and Jeffrey Rothstein, her counterpart at Johns Hopkins, both phenomenal researchers. They created Answer ALS Consortium, where you've got repositories of data from ALS patients. The most abundant data there are data sets from iPSC-derived neurons. So you basically take some cells from an ALS patient, reprogram them into neurons, sequence, and put the data out, plus metadata. But those are baby cells, right? Reprogrammed cells, not real biopsies, because as you know, it's very difficult to biopsy the brain in live patients. So we combined it with post-mortem ALS patient-derived brain samples that are in PandaOmics, put it in our AI engine that, again, a lot of, maybe some of you are using as well, and identified novel targets and drugs to repurpose. And then we used the fly model from Zhavoronkov lab at Mayo Clinic. He is now at a different university to validate. And at that same time, I met Professor Bailu, very famous professor at Tsinghua University and the founder of RB Technologies. He's an American-Chinese scientist, probably one of the most famous in APAC in neuroscience. And together we ran this study where we took 26 predicted targets and used RNAi screens on fly eye neurons, visual neurons, to see if we can rescue the ALS phenotype. And out of the 26 targets, 18 showed strong rescue or moderate rescue. Only a tiny number showed actually the worsened ALS phenotype. So what Bailu's company RB Technologies did during this study, they looked for drugs to repurpose, selected one, and took it into human clinical trials in a principal investigator-initiated study, because we know that the drug is safe. They put it in patients and did the study in China. So the beauty of collaborating with China is that it has more than 1.4 billion people. And now the level of healthcare and data analytics, data informatics, quality of the data is supreme. They managed to make a giant leap over the past 10 years. So it's actually very fast you can very quickly enroll patients for a clinical study. And here, within a year, they enrolled 64 patients for an ALS clinical trial, right? So in the US, the same number would be impossible to recruit in the same time. So now we're waiting, we will see the human efficacy study, hopefully by the end of this year. But the fact that it was possible to go from a paper to patients in a year, it's incredible. I have not seen that done anywhere else, and AI helped a lot. So, but you really don't know how good your AI is until you either get a regulatory approval, which again takes a very long time, and the phase 3 study may cost you $300 million. And then another way to validate is to sell several drugs to expert developers. So it's kind of like, if you don't know whether you're a good cookie maker until you sell your own cookies to other cookie makers who are super expert, right? Or another analogy would be selling fridges to Eskimos, right? So you need to ensure that the fridge is very, very good. So usually the quality of the asset that you are selling to the others as an AI company has to be higher by default than if you want to progress this asset yourself. Because usually biotechnology companies have lower requirements, lower quality requirements, and lower data package requirements than those companies that are buying those assets. So also, I thought that it would be worthwhile to tell you about the evolution of pharma partnering AI companies, because it will help you in your career if you are deciding which company to go and work for, or if you're starting your own. So originally, in the early days, investors and pretty much everybody, the media, valued strategic collaborations with pharma. So you come and say, okay, I've got an AI platform, and now dear big pharma, pay me some upfront to use my platform, and we will collaborate on a program. Those are very difficult to start because you need to work at the very high level of the pharma company, takes some time, but actually any company can do that, right? So if your board members know each other, it's a kind of big boy club. And if some of them know each other, I've seen it many times, they will partner. They will give you this upfront, and you'll try to help the cookie maker make their cookies. So those strategic collaborations, I have never seen a single one progress into phase 1 complete, for example, in my 12 years in the business. Those are useful. Also, I sometimes partner like this just because you get to learn some new things. Plus, it helps you, keep your company afloat, because the pharma company is subsidizing part of the research. The more interesting way to partner with is to produce AI software. So once you actually developed your software to the level where it's production grade and you can put it on the market, so it needs to be very, very sophisticated. It's not just Python library that you put out and say, oh, I've got open source, now everybody go use it. Industrial grade software is much more difficult to produce. But once you do it, the partnering becomes very easy. So pharma companies or academics just look at the software, test it, do a free trial, and they buy it. But that's very, very low impact as well, just like strategic collaboration, because usually internally they will accelerate their R&D, but for many reasons they also have not produced a single phase 1 complete program. And then another way to partner is assets. So you get to a certain point in your discovery effort, for example, preclinical candidate. So preclinical candidate and IND enabling studies, that's, in my opinion, is the best time to partner with pharma, because they can start the clinical trial from the clean slate themselves without taking it over from you. And to get to that point, you really need to be very sophisticated. So your asset needs to be pristine. But it's moderately difficult for partnering, because if your data is good and you're outperforming everything else out there and the target is trendy, you are going to sell. And if you are going to sell to a company which is very committed to your program, the probability of the drug to go into the human clinical trials and then pass is increased, because you already have provided a working molecule to the pharma company. So I think that assets and software are the most important ways to collaborate. And we started doing that pretty successfully. So after we showed some of those molecules to to other big pharmaceutical companies, many of them liked them. So Sanofi, it's a standard collaboration, as I've explained. So helping the cookie maker design their cookies. But Exelixis and Menarini, those deals happened just end of last year, so it's pretty recent. And we managed to sell the USP1 molecule for $80 million upfront with very substantial milestones and royalties. And Exelixis is one of the best biotechnology companies I actually know in the world. They're publicly traded, they generate several $billion in revenue, but at the same time, they were founded by expert medicinal chemists who understand the value of the molecule and understand the entire development process. That's why they don't try to be cheap. If they see the best-in-class chemistry, molecule with the desired properties optimized and tested and all the way to IND approval, they took our molecule for 80 million. And then Menarini took our molecule for $500 million in total value, but only 12 million upfront. Why? Because the molecule was at a preclinical stage, preclinical candidate stage, so it's much earlier. And I actually think that it's much better to collaborate with companies of the size of Exelixis or Menarini, because management changes within big pharmaceutical companies very often lead to destructive pipeline reshufflings, and very often you lose your programs just because some CEO changes, CSO changes, head of the therapeutic area changes. In medium-sized pharma, the probability of those changes is lower, and they don't have hundreds of phase 1, phase 2 trials like big pharma do, and for them it's much more valuable. So I actually like to give kind of our best kids to the best parents, so to speak. Yeah, it was a pretty famous deal. Now I'll try to wrap up, but I want to show you something that I'm pretty proud of. We use a lot of contract research organizations to perform validation experiments. So usually once we get a molecule, we develop a set of validation experiments and run them in parallel at several CROs, contract research organizations that do it for money. But we decided to build our own lab that takes a small part of those experiments. Again, it's just small, tiny number of experiments that need to be performed, and decided to make them in a fully robotics fashion. So we have a lab, I call it the 6th generation laboratory, that can contribute at every step of target discovery, target validation, lead generation, preclinical candidate, indication expansion. And this is how it looks like right now. So I did it during COVID, so spent 14 weeks in quarantines, and we were allowed to bubble in that place. We built it in Suzhou because it's the area where a lot of robotics companies are. Highly recommend visiting the place, even though nowadays geopolitics are making this a little bit more difficult. And I really don't like that, but our job is to make patients healthy, so we go wherever we can collaborate. I started the development of this lab in April 2021. And December 2022, you see this footage. What you're gonna see right now, it's not animation, it's like real footage, just a quick camera workflow. And I wanted people to feel like they work in the future. So people who work in this lab, they really enjoy this kind of spaceship environment. So you walk into this lab, it opens up, it recognizes your face. You walk through it, you've got dimmed windows, you can undim them if they're not confidential experiments, and just do a walkthrough with a laser pointer. This is the real footage, real workflow. So you throw in tissue, cell, organoids, the robot picks it up, grinds it, microplates it, does quality control, no mycoplasma, no fungus, passes it to another room. There I have an army of autonomous guided vehicles, AGVs, micrometer precision. Part of the sample goes into the incubator, part into the imaging station. Depending on how much money, how much sample you have, you can perform 4 or 5 types of imaging. Then part of the sample goes into the NGS room, library prep room, also very clean, separated. There you have fully robotics library prep, methylation and transcriptomics on whole genome. Then after sequencing, all this data goes into the AI layer, so mostly pandaomics. And here is where magic happens. Depending on your novelty and risk profile, you select the targets automatically. Then pick the compounds for those targets automatically. And then incubate the original sample with the compounds that AI picked. And then complete, again, the same process. So phenotypic screen, methylation, transcriptomics. And based on the response, AI either gets punished or rewarded, depending on how well it did. But for the experiments where you had a lot of hits with the known molecules, and those hits were real, but many targets lacked molecules, you actually pursue those targets, those novel targets with RNAi or on CRISPR screens, try to understand the nature of those targets. So we have a lot of novel targets, but at the same time, we train AI using this lab. It's fully automated, so humans can come in only to change the reagents, and maybe for one step where you have to load the chip into the Illumina machine. It wasn't designed for full automation. But except for that, everything is running fully automated in a fully automated manner, and you can leave it running for a week, for example. So now a little bit about the future. So if you are not an AI agents, get into this right now, right? Super easy. I actually started coding again and making my own agents. I think multi-agent Gen AI research systems are real and it's the future. You should start thinking about quantum computing, even though it's not there yet, but it's definitely going to be there in the next 2-3 years. No question, just the infrastructure needs to scale up. And fully integrated AI drug discovery and personalized medicine solutions are going to be a big thing. Some papers where we talk about quantum in terms of near-term applications, if you need to read one, just read this one, Drug Discovery Today. We explain where in generative chemistry quantum can be used and how. Another one is this, exploring the advantage of quantum generative adversarial networks in generative chemistry. Here we have some virtual validation with benchmarks, but again, it's nothing compared to real experimental one. And on arXiv, we have a really fun paper with Alana Spurogudzik again. So we share the priority here. Here we used a real IBM quantum computer as a generator hooked up to LSTM generator and the chemistry 42 discriminator. So you can actually train the system to generate valid molecules. So we are not claiming that it's better than GPU, but we are just saying that just like with the DDR kinases paper, we're saying that it's possible. So look at this paper, and what you're looking in the figure, in figure 1A, it's the first real quantum generated molecule on a real quantum computer synthesized and tested for KRAS. So look at this paper. And here I'd like to stop, but one more thing very quickly. So multi-modal transformers are a big thing. So if you didn't see this paper by DeepMind called Gatto, but by Nandan Freitas, very wonderful scientist, you should. You know, when it came out, I was at a scientific conference, and that's me. I took a selfie. And in 2023, we published a fun paper called Precious One GPT. It's also a multi-modal transformer for picking the targets from methylation and expression data, where we train on age and then retrain on diseases. But that's not a true kind of multi-modal. It's two different models training each other. But now we are releasing Precious 2 and Precious 3 GPT. Precious 3 GPT will be available open source. So that's a truly multimodal system where we tokenize different data types separately and have one global model doing the work. And it has many different omics data sets integrated and used for training. It has AI-assisted quality control, and also we are planning to have community-based QA via web interface releasing soon. And it will allow you to do drug discovery, omics interpretation, screening for natural compounds that might reduce your biological age. And also you can talk to it about aging in general. So here, thank you very much. Happy to collaborate. And if you're interested in Pandomics, request your trial access online. It's usually enough to write a paper. So yeah, thank you. Happy to answer some questions.
Well, I know that we're past time, but the good news is we have a cancellation in the next session. So if with your consent, then we can actually just take maybe two or three maximum quick questions. I'll start actually. This is really amazing. You have shown not only what you have done, but this seems to be the tip of the iceberg, what's going to happen in the future. The future is great for you as a company to disrupt the drug discovery industry. But is there a role for academic research that you cannot possibly do yourself? And what is the advice to us?
That's a tough one, right? So I think that much of the real practical applied research in AI-powered drug discovery will be in the companies, because academics need to publish. And once you publish your molecule in the open, it's demonetized, even if you patented it, right? Because if it's if you're going after a hot target, somebody somebody around the world in China or India or somewhere will recognize what you have done and make a few molecules that go around the patents, to patent busting, and go quickly, and your molecule will be demonetized. So I think that a good idea would be to start startups around specific targets and molecules, and ensure that you can protect that IP, because again, the most value is in the molecule, right? So you need to ensure that it's safe, it's effective, and easy to make, and achieves your goals, your therapeutic goals. So my recommendation is to start startups around specific molecules. Second, of course, is I think that nowadays just alone you wouldn't be able to achieve a lot. You need to collaborate. So massive consortium collaborations where people share data, share models, share benchmarks. That's the future. And of course, if you want to become more efficient, going with many AI agents, developing many AI agents within the university framework that will allow you to pass some, I would say, simple and stupid tasks to AI, and then orchestrate this AI to automate part of your work. So you start working on something much more intelligent. That would be another recommendation. But I think in terms of the real practical application of AI to drug discovery, you need to have a company, because the amount of money you need to raise to put the drug on the market is just incredible. So the reason why you see us collaborating, licensing out, accumulating funding, is because we are preparing to take a bigger leap to go after more complicated problems and take the drugs on the market, right? Put the drugs on the market. Hopefully ourselves one day, but most likely before that with big pharma partners. I think that until preclinical candidate stage, you should own the molecule, and you should try to make a startup around that. Nowadays, the timeline, for example, for us to get to preclinical candidate, average is about 13 months, so just over a year. So after that, you complete IND enabling studies, and you can start your human clinical trials, right? So basically from 0 to human, I can go in just about a year. Oh, sorry, about 2 years. So I think for academics, it might be a little bit longer, and you need to get, SBI or STR grants. But still, it's not 5 years, right? And after you get to preclinical candidate, in maybe 1.5 years, you can sell it to a pharma company or biotech for a lot of money. And that's I think that's the best way to go.
All right, thank you. There's a question in the audience.
Hi, Alex. It was a great talk. I'm Rahul, and I was a postdoc, and I'm basically like you. I'm from AI core machine learning. I'm still learning biotechnology. I'm fascinated with your lab. The lab that you show robotics. Is it autonomous?
Yes.
Okay, so
it requires a bunch of humans to operate, but once you start the run, it's fully autonomous. And nowadays you can program it using objective-based programming, and it will even set itself up automatically. But it's usually better to do it with a human.
So the question is related to that only. It's like, if it is autonomous and machines are not creative, are they doing mechanical tasks, not creative tasks? Because making a molecule, observing pattern, still humans are much better than machines when it comes to temporal pattern recognition. So do you have
Okay. We have to actually.
Yeah, human is in the loop later. So the objective of this lab, three major objectives. One objective is target discovery. So to identify novel targets, to understand which protein is more important in a specific disease than another. Another objective is to do indication expansion. So you basically use your already existing drug to see if it can work on additional diseases. And the third objective is to train your AI, right? And to make your AI better. So many of the tasks, as you've said, are rather primitive, right? So prepare the sample, clean the sample, incubate the cells, image the cells, sequence the cells, get methylation, transcriptomics, and then there is a creative task, right? So we use more than 30 AI models to understand the data that is created from the sample and pick the molecules automatically. So that step is actually sometimes better performed by a machine than by a human being, because humans are super biased, right? So if you talk to any head of a therapeutic area within a big pharma company, they will already know all the targets, right? They will say, look, I don't need target discovery. I know which ones to go after. Just help me build a little bit more kind of like tell me which one to go to prioritize, right? Out of this list of 100 that they usually have. And each one of them will be super biased, right? They will be super biased to the old stuff, right? So something that has worked before. When your target hypothesis comes from original data, you are debiasing the process, actually. So usually even myself, I know that a few hundred targets now, I would always be cautious when AI spits the list of targets where I don't know a single one. Right?
Can I ask a question about that process?
Yeah. About what?
About the process, the flowcharts. You put a lot of flowcharts throughout your talk, and I was curious about the timeline for the flowcharts. And exactly, you mentioned a 2-year timeline for clinical trials, the phase 1. What is the bottleneck in that process?
Sure. So I will. So this is the flowchart, right? I hope you can see the slide. If you're going to use a
flowchart, maybe you could go to the one with the DDR inhibitor with the you showed from AI all the way to, in a mouse.
Yeah, sure. This one, but this one you already know the target, right? So in this one we didn't know the target, right? So actually this is a better flowchart because it shows you years, and you can see that target discovery and small molecule chemistry is basically a year. So we need to perform many different experiments. So once you zoom into a target, for example, XYZ123 for a specific disease, let's say it's like arthritis, or let's say cancer. Cancer is easier. You take a bunch of cancer cell lines and primary explant primary cells and do an in vitro experiment, right? So you basically try to knock out this target, this gene, in those experiments and see if cancer cells die, right? Or you put cancer in a specific state that is more susceptible to other forms of therapy. This step, you cannot avoid this, right? So you really need to do the experiments because first of all, the regulators will ask you for this kind of data. Second, you actually have a lot of choices for targets, so you need to see which one to prioritize because later experiments. So this step, in vitro validation, right out of AI. So this step may take you like 2 weeks. This step will take you about half a year, right? Because first of all, you need to procure the material, right, to test your chemistry on. No, not the chemistry here, the target on, right? If you are doing, let's say, CRISPR or siRNA. And this step will probably cost you about 6 months at least, and maybe $500,000 to $1 million, right? If you are using contract research organizations, you want to have 2 replicates of the experiment. And you want to ensure that the quality is good. Sometimes if some of these experiments fail, you actually extend the timeline, right? So that becomes longer. And then you select the target, right? You kind of, you understand that the target works and you do the chemistry step, right? So you synthesize the first batch of, let's say, 20 molecules that are going after this target and test them in enzymatics. That step would take you maybe about a month and cost you, if it's 20 molecules, maybe it's going to cost you about $100,000, including compute. And then you kind of pretty much repeat this, but in parallel with other things. Try to see if your tool compounds work in some of those experimental assays. And at that time, you might be already at the second round of synthesis. You understood where the molecular properties need to be optimized. You do the second one, maybe synthesize another 30 molecules. And with that, sometimes you're already, those will be the molecules that will go into preclinical candidate. You perform a large number of animal experiments. You need to demonstrate at least 2 animal models of disease that respond to your therapy. Find the dose, do PK studies. So these 2 steps could be a year. So you nominate preclinical candidate usually within 13 months on average. It also depends on the novelty of the target. If the target is very new, many of these experimental assays, and especially animal models, will not be available, right? So you actually need to see which one is more relevant to your indication, to your disease. And sometimes you will need to take whatever is available. And the model might not be representative of the disease, but you still need to do the experiment. And it might take longer.
Yeah.
So here,
Alex,
it's a novel target. Yeah.
So I think the time is really short, and I wonder if we can take the discussions and they may be able to find you on the online blogs and continue the conversation today. But it's
for sure. So this paper, you can actually ask the questions to this paper and ask how long does it take and go through the entire process. So this paper is actually pretty powerful.
Yeah. So it's been a really inspirational and fascinating topic. And really congratulations for the many achievements that you have done. I wish you the best of luck in your company's commercial success, and see you more often in future conferences, maybe in Basel in later this year. All right, thank you very much.
And please welcome to our conference in Copenhagen. So ARDD, thank you.
Yeah, and the aging conference in August. Thank you.
Thank you.
All right, so we'll take a break for 10 minutes and come back, and then at exactly 10 o'clock. Thank you.
In the lab where data In the lab where data flows, revolutions underway, bridging gaps, set to show ace power in full display. Flagship sets, cornerstone, a future so bright, where machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Across lands, hand in hand, we craft new reality. Software standards unify, language shared and bold. data's truth, we amplify story yet untold. Fair principles our guide, accessible and free. In this journey side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein, an artery bridging minds. Future grows in this AI quest. Workforce trained, ready, keen across AI divides. Biomedical scenes I've seen, world forever wide. With each step we close gap between machines and human grace. Between machines and human grace. Bridge your white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. In AI we trust, in AI we must. In unity we find future just.
Okay, let's start our research session 1. In our research session 1, we have 3 speakers today. Our first speaker is Dr. Stephen Barnes. Dr. Stephen Barnes is a distinguished professor at the Department of Pharmacology and Toxicology at the University of Alabama at Birmingham. He received his Ph.D. of biochemistry from Imperial College, University of London in 1972. He joined UAB in 1977, which I think is earlier than many people here, and pursued research on bile acid and later the polyphenols. He received the 2012 UAB Medical Center Distinguished Fellow Award, Faculty Award, and is a fellow of the American Society of Nutrition. And in 2019, he became a UAB Distinguished Professor of Pharmacology and Toxicology. Dr. Barnes' topic is the plasma pharmacome, EHR, and the promise of the AI beyond the drug discovery. Without further ado, let's welcome Dr. Barnes.
Okay. Thank you. Well, we heard from Alex about the emphasis was on discovering a drug that's feasible. The reality of the clinical world is more about toxicity. And so toxicity is one of the driving forces in clinical medicine to better understand. And so my presentation here will look about how we can look at the pharmacome, that is the compounds that are present in the blood that circulate around a patient, what is known in the clinical record as opposed to what is actually in the pharmacome, and then looking to see how AI could maybe help to develop a better way of handling patients. And I should say, just to give you an estimate or a feel for the size of the issue, in the US each year, in hospitalized patients, 100,000 patients and more die from drug-drug interactions. Okay, so let me get started with this one. Okay, so if you're going to do data science on patients and you want to integrate any of the omes that we heard discussed before in a meaningful and actionable way, it depends entirely on the quality of the data. I think that's true of any AI system. If you don't have good data, you will get really strange answers. So the plasma metabolome that's in all of us reflects the all the small molecules that have been circulated around and then distributed to the tissues. And these consist of endogenous metabolites, the ones that the cells in our body make, food metabolites, because we actually have a very weak genome, drugs and environmental exposures that happen to us, as well as, of course, the bacterially derived metabolites that are coming into us from the amazing bacteria in our guts. So the pharmacome I'm going to talk about is the collection of, and in quotes, administered drugs, because you might think that it's only the prescribed drugs, but of course it's everything. And it also includes their metabolites. So just a few definitions from the point of view of a pharmacologist. The first thing is, what is a drug? Well, it's a drug that has an effect. It may already occur in nature. Nature is a wonderful thing. It's been doing drug discovery on this planet for about 3 billion years. And so we can modify that compound, or we can encapsulate it to deliver it better. It could be an entirely new compound, some of the ones that were discussed earlier, made synthetically to target either an enzyme or a receptor or a channel. The other thing we have to worry about as pharmacologists is that just because you put the drug into the patient, it doesn't necessarily get into the blood. And if it does get into the blood, the body is trying to get rid of it all the time. In fact, drugs that don't get rid of all the time accumulate and give toxicity. So you want a drug that is going to be there for long enough to have effect, but not too long that it would cause toxicity. So we determine the area under the plasma time curve, so the plasma concentration time curve, and that can determine how many times or what distribution of dosing we should give. Can we give this drug and take 1 per day, or do we have to take it every 2 or 4 or 6 hours? The other one we nearly I'll come back to this several times over, is that all drugs have a therapeutic index. And therapeutic index is the division of the maximum tolerated dose for, and that's for everybody, but not necessarily you, and the minimum dose that will have actually an effect. And you want that window to be as wide as possible. So over-the-counter drugs usually have a very wide therapeutic index. Sometimes you can take drugs that you're using in the right concentrations, but instead you now are using multiple drugs to attempt to deal with the issue you have. And this, my good example here is of Heath Ledger, who was taking drugs to try and overcome his inability to go to sleep. Each one of these drugs was being used in their proper way. But the trouble was that the combination was toxic, and in his case, fatal. So these are the drugs that he was in, the diazepam, oxycodone, and doxylamine. So we decided, with the help of Jia Li and Karen Yara, to take a look at a group of patients to find out what drugs they were taking and how many of these drugs were in the patient record. The study was actually designed, these were people who were recovering from spinal cord injury. You might expect that they would have quite a few drugs. And they were put onto 2 different diets. I'm not going to talk about the 2 different diets. I'm going to talk about what we saw in the drugs in these people. So the 36 patients were entered the study. They provided a blood sample at the time before they were assigned to their particular diet group. And then they went on a period of being exposed to this diet to see if it improved their recovery. And then 18 of them, but not 36, provided a plasma sample after that so that we could look to see what had changed. When we do this, we do something called LCMS. We take the patient's plasma, we precipitate the proteins to get a fraction that we can now inject into a liquid chromatography system that separates things based on their hydrophobicity. The material coming through the column then goes into a measuring device, a weight measuring device, a mass spectrometer, and we can measure the weight of the compounds with an accuracy of usually better than 5 parts per million. So we've got a very good idea of what it might be, but we can also take those ions and subject them to collisional dissociation, the sort of thing that happens to a space shuttle coming back through the Earth's atmosphere. If it gets heated up and if it's not maintained its integrity, it falls apart. Well, these molecules fall apart in the same way and produce a mass fingerprint. And this mass fingerprint then can be matched to databases, and we can tell what that particular ion is and how much of it is there. So what do we find in these patients? Well, let me start by saying that the clinical record said there were 28 therapeutics. Actually, only 14 of these were actually drugs. The rest were infusions. The mass spectrometry detected 80 drugs. The first ones were very simple, sweeteners that people put in their coffee. That probably is not in the clinical record. There were over-the-counter drugs, Tylenol, ibuprofen, phenacetin, and so on. But then there were the more serious drugs, painkillers, anti-anxiety drugs, anticonvulsants, blood pressure medications. I even take one of these, the losartan. Acid excess was controlled by ranitidine in one of the patients. Several of the patients were on chemotherapy for the cancer that they had in addition to the spinal cord injury. and the use of antibiotics. When I looked at the data, I also found that there was cocaine. Cocaine wasn't prescribed by any of the physicians, and nor was the morphine or the methadone. The patients, of course, can go out and get on the web and get all sorts of additional materials for their needs, or perhaps go down to the street corner and get them there. So this is a problem. Very interestingly, you remember there were 36 patients that came to start the study, but only 18 came back. When I looked at the 18 that came back, 12 of them were obviously taking cocaine, either on the day they came back or the day or two before, because I could see all the cocaine metabolites. So there's this huge gap between what's in the medical record and what the patient actually takes. And this is enormously important because drugs interact with each other. So we now consider what happens when you take a particular drug. As I said earlier, we want the drug to go into the system so it can have the activity we want, but we don't want it to stay there for too long. So many drugs have been optimized so that they can be taken up from the GI tract. So they're oral drugs. And that's important because the wall of the intestine actually has metabolizing enzymes. And this is a product of human history. You know, long before we had any of the science we see today, people would eat whatever they could. There was no shops to go to. And the people who chose the wrong things, because they couldn't metabolize them and therefore the toxicity took over, were the ones who didn't have kids to procreate. So bit by bit, you have high selectivity for people who can metabolize foreign compounds. So this is one of the real challenges of drug development, is to overcome the issue of pre-metabolism before the drug ever gets into the body. Now, drugs are metabolized in two ways. The first step is called phase 1, and it's principally denoted by the cytochrome P450s. And these are enzymes, they're monooxygenases, and they either remove alkyl groups to expose an OH group, or they will add an oxygen. And by doing so, they create a tag. And that then becomes part of what the second group of enzymes can do, the so-called phase 2. And these can glucuronidate, sulfate, put an amino acid on, and some other events. And what this does is to make those forms of the drug now excretable. So this is really, really important. And so the drug can be absorbed through any part of the GI system. From the mouth, you may have had drugs that you put under your tongue, and so they can be absorbed from there. Drugs can be absorbed from the stomach. Tylenol, drugs like that can easily be absorbed, although surface area is hugely important. So the part of the intestine with the greatest surface area is the small bowel. And so typically there are drugs that have a 30-minute effect. If you take a Tylenol or something like that for a headache, you want something to act in 30 minutes. But there are others that may be take an hour or 2 hours. And occasionally those drugs that have been specifically made so they're targeted to be only active in the colon. So they put a diazo group into the molecule, and the bacteria have a unique enzyme that can cleave that diazo bond. So now the drug is active there. And then there are also, as I sort of wave my hands, there are all sorts of chemistries, enzymatic chemistries, which are used to either modify the drug or send it out of the system. There are occasional drugs that actually get activated by the cytochrome P4 system, by the dealkylation event. So one of the issues, when Alex was talking, he was talking about the development of one drug at a time, and that's not real world. As we saw from these patients, the patients can have a very large number of drugs. In fact, I've seen a figure for elderly patients, somebody more my age is taking 8 to 12 different medications at one time. So we're always looking at drug-drug interactions. The number of drugs greatly exceeds the number of cytochrome P50 genes. And because of that, drugs can compete with each other, and therefore they can affect each other's clearance from the blood. And this is particularly important. Remember, we talked about the therapeutic index. So if the therapeutic index is not very wide, and another drug is using the same system to be metabolized as the one of interest, then that level of the drug for a given dose will go much higher and may move from the therapeutic range up into the toxic range and lead to death. So some of the foods we eat, I was looking to see if there was any grapefruit juice up there. Grapefruit juice is a bad interaction with a number of drugs because it uses the same cytochrome P450 that the compounds in grapefruit juice do. And then there are things like tyramine, which is a monoamine oxidase inhibitor. And so it can stop compounds that would normally be metabolized by that particular enzyme. It can stop this occurring. So I came across some papers which will interest the informatics people listening that are now trying to tackle this problem of drug-drug interactions and try and put it into a computer so that you can now evaluate what it's going to be. So the problem at the moment is there are 2,700 plus drugs that the FDA approved that are small molecules. The people doing this informatic development are looking through the literature to find out what particular CYPs, the cytochrome P50s, are metabolizing that drug. And then for each drug and CYP combination, what is the KM of their interaction on their own? How does that change as other drugs become involved? And can you do a simulation of all of this? And there is a paper that explains this and puts lots of equations up, which I won't bore you with at this point. The goal of this was to assess the extent of drug cross inhibition. Now remember, when I show this, I'm only going to show it for two drugs interacting. Remember that what is really happening is a multi-drug interaction. And the extended assessment in their system was to now layer this on top of knowing about the variation that occurs in you versus the population. Drugs are generally developed for a population and not for you. So if you're in the hospital and you happen to have a defective copy of one of these cytochrome P450s, the capacity of that system to deactivate the drug may be reduced. And therefore, for a given dose, you will have a higher level and possibly a toxic one. Now, the question is, did we get to this just because of science we've done in the last few years? And the answer is no. Actually, our predecessors and those who are living a different lifestyle to us are, in fact, already aware of this. So Richard Schultes went down to Amazonia in the 1930s with the World War II on the horizon. The US wanted to find alternative places for tire, for rubber for tires. And so he observed what the shaman were doing. They had a plant that they ate to turn into a trance. This has a compound in it that's a hallucinogen. But they also found that if they put another plant in, they were able to extend the lifetime of the hallucinogen to the point that they could now have an effect over several hours. So they were using an inhibitor of the metabolism, and so they found it out by trial and error. And then this is the results from this computational expression. So the gray things you see are the ones that were already in the literature. So what we're looking at is the enhancement of the area under the curve, the plasma area under the curve. And the higher it goes, the worse it is. So up in the top left-hand corner are really serious drug-drug interactions, the ones that are brown. And so they took 59 substrates, 35 inhibitors. They noted that ranitidine, which is the big, then I can show you, but it's the one in the front. It's all white, so that there was no interaction between ranitidine and any of these drugs. But on the other hand, the ritonavir, indinavir, metronidazole, ketoconazole, clarithromycin cause very, very strong interactions with other drugs and would have to be avoided. And they had their effects on the solodipine, lovastatin, bispirone, milazolam, triazolam, and sildenafil, which is better known as Viagra. Be careful what you put with your Viagra if you happen to use it. So the patient's use of drugs far exceeds what's captured in the electronic health record. LCMS can overcome this. Drug-drug interactions can cause serious toxicity, even death, due to delayed metabolism and excretion, which elevates their plasma levels. But data from PK studies and in vitro enzyme studies with purified CYPs can be combined into working models and generate predictive drug-drug interactions. So there are opportunities for modeling and application of artificial intelligence and machine learning that will be incredibly beneficial. We need to expand it into the multiverse, because I said it's not one drug on one drug, it's one drug on maybe 10 drugs, depending who you are. And if we had that, we might be still watching Heath Ledger as an actor. So finally, I'd like to thank Xia Li and Karen Yara, seen here. They're sadly at a higher state now. Also Landon Wilson and Taylor Berryhill from my lab, who did the mass spec work. And I got support from NIH for the instrument, and Xia Li got the Nielsen funding. And we just got a new instrument. All of this didn't overlap quite as badly on my Mac, but we did pretty well. Thank you.
Thank you, Dr. Bynes. Due to time limit, we may take one question from audience.
Very, very interesting talk. So in terms of the AI predicted metabolic half-life you showed that some drugs they predict to inhibit those P450 enzyme, but then that is one way to slow down the metabolism. However, for other drugs,
well, so another thing I didn't mention about drugs is that they induce their own metabolism. So are you thinking about a negative effect on metabolism? It would depend on a drug-by-drug basis. So it's important to understand which of the different enzymes, the cytochrome P450s, if we go back 30 or 40 years, were just considered to be one enzyme. But with the advent of molecular biology, we realized that there were multiple ones. And so the makeup of the enzymes that are in you is important, because what's in you may not be what's in me. And therefore, we don't know. At the moment, we largely don't know that. We only know it in the more extreme cases. I belong to the Precision Medicine Institute, and we take people who have overt phenotypes, usually children, but not always. And in those people, we know the whole genome. I think where people are now moving to do whole genome studies on patients. And so in England, I think they're advocating that all cancer patients actually have a whole genome study done, so that you can much more selectively choose what drug to treat them with, because there may be a unique target in that genome that can be used. Thank you.
Our next speaker is Dr. Andrew Tan. He's endowed professor in oncological sciences, University of Utah. He's a senior director of data science enhancement Cancer Institute. He received his Bachelor of Engineering from IF Technology, Malaysia, Ph.D. University of Glasgow. His research topic including translational bioinformatics, cancer system biology. His study topic is computational strategies for drug repurposing, drug combination prediction. Let's welcome Dr. Tan.
Thank you, Dr. Chen, for the kind introductions, and thank you, Jack, for the invitations. So I'm a new kind of senior director of data science enhancements, and then this is kind of like our cancer data science visions. So it's basically trying to bring data and computational tools to empower researchers so that they can do innovative and discoveries to bring to a cancer-free frontiers. I'm just going to start by showing these plots. So as previous speaker was kindly saying that the way that we think about precision oncology is to start to go back and sequence all this whole exome or whole genome sequencing, try to see whether there's any defects that we can identify some disease mechanisms. And then hopefully that particular mutations can lead to a new drug discovery. And then the idea is basically how do we deliver them to the clinic so that patients with that particular mutations, the red lines, have better and durable survivals compared to the one that doesn't have that mutations. So this is basically kind of like known as the precision oncology. I call it 1.0. So this is one example that has been published about like a decade ago. So on the left is melanoma patients. As you can see, this is all the melanoma tumors, cancer cells. It's only 38 years old. When they do these biopsies and then when they do a whole exome sequencing, they found that these patients carry this BRAF V600E mutations. That is why the disease progressed. And at that time, there's a drug known as BLX4032 that specifically inhibit these variant versions of BRAF. As you can see, the patients after 15 weeks of taking the drugs, all the cancer basically disappear, right? So this is kind of like the ideal case of a precision oncology. You identify a target in your cancer genome, and then you deliver a drug. So as a computational tools, my lab over the years developed all kinds of methods. The idea is trying to see how we can bring this data, process it, and then help the clinicians. And we do that in kind of like a collaborative fashion. So the idea is the tools that we develop could potentially apply to melanoma, could also apply to lung cancer so that we can keep on reusing the model. So one thing that I put in the slide is the patients, same patients, right? When the patient continues to develop, taking the drug for another 8 weeks, you can see that all these tumors started to return, right? And when they rebiopsy the patients over here, they found that the patients still carry these BRAF mutations. However, there's a new acquired mutation downstream of BRAF. This is a MEK pathway. And now basically the cancer cell is basically acquired a new mutation that cause this resistance to the drugs, right? And the idea that in my talk is basically trying to say that one drug might not be able to cure cancer, so you might need drug combinations. So at that time, in the field of melanoma, they do kind of like develop a new drug that is basically MEK inhibitor, and then there's a lot of like a different combination of MEK and BRAF, and you can see that the red color lines basically indicating the combinations, and then the blue line is a single agents. They do kind of like increase, but it's not kind of like very durable. It's only like about 0.5 months, for example, these combinations, and for example over here it's probably about like 5 months and so on and so forth, right? So this is a little bit old data, but what I'm trying to show you is how we use this kind of computational strategy in coupling with the experiment to find drug combinations. So at that time, this is probably about a decade worth of works. So we basically use these functional screens. The idea is basically assuming that you have cell lines, you can transduce some of these shRNA libraries. The same concept could apply to CRISPR screens. And then you basically split the cell lines into two groups. One is taking the vehicles, the other one is assuming that you treated with drug X. Every one of these color-coded indicating that there's a gene. For example, a red gene is being inhibit, knocked down, or knocked out in a CRISPR screen sense. And then you basically let it recover for a few days, and then you basically do some sequencing. So what I'm trying to show you here is, in this particular experiments, right, the green and the black is basically disappear from the treatment's arm. So this suggests that when you want to find a drug combinations or synthetic lethality, you might want to find a drug that can inhibit the green or the black genes combining with your drug X, right? So I just want to show you one example. So this is, like I said, we develop the tools and then we apply to different diseases, and some of them eventually can go into clinical trials. So at that time, we are interested in understanding colorectal cancer with KRAS mutant. So for this particular colorectal cancer subtypes, they are basically didn't benefit from EGFR inhibitor cetuximab. So what we are trying to do is trying to see whether we can find some target downstreams, which is a MEK inhibitor that we are interested at the time. So the idea is, if we use this MEK inhibitor, can we find other synthetic lethality partners? Because we know that one drug is not going to cure cancer. So when we do the screens, we identify the top hit is Wnt pathways. So all these different color coders suggesting that they are either from a screen hits from two different cell lines, or looking at the gene expression, they are overly expressed. And then at that time, we are trying to figure out what is the best drug combinations, because there's no FDA approved small molecules that can inhibit Wnt pathway. We found that there's one compound is called calcineurin inhibitor, which is this particular complex. There's a cyclosporine A. This is an immunosuppressant drug, which is not a cancer drug. So our hypothesis is, if we combine cyclosporine A with a MEK, we want to see some combination effect or synergistic effect in colorectal cancer KRAS mutant patient. So the first thing that we do is we tested this in all these in vitro screens, and then we bring into a patient-derived xenograft. So this is basically a model of a KRAS mutant patient-derived xenograft, PDX. The red line is vehicles, no drug treatments. The blue line is a MEK single agent. We know that it's not working, right? And then the brownish line is the cyclosporine A single agent. However, when you combine these two together, you see a very nice kind of like inhibitions, right? So this is one model, this is another model showing the same kind of trend. So at that time, the postdoc, Anna, she basically said that, okay, usually we are going to sack the mice at this end of studies, but she was saying that, hey, now the tumor is kind of like big. If we put in the other treatments agents, so for example, if it's MEK starting as a single agent, if you add in this cyclosporine A, or when you start with cyclosporine A, you add in this MEK, right? What is going to happen, right? So we basically see that the tumor is big, and then it started to regress, right? And then this is basically, you can see that the regression of the big tumor when you add in the other compound. And then obviously the arms that it started with the combinations continue to inhibit it, right? So this is one model, this is another model. So this is kind of like very interesting. And working as a team science, we basically have clinicians, Chris Liu, he basically tried to bring this into a clinic. So this is basically the clinical trials where we're trying to bring in the MEK inhibitors, selumetinib and cyclosporine A in treating metastatic colorectal cancer patient. I put in here that this is a timeline about 5 years that we work together as a team to start from all these experiments and then bring into a clinic. It's obviously less superior compared to the AI-driven 1 year approach, but this is at that time a very kind of like, for me as a computational researcher, it's very fulfilling. So you asked me what is the result. So here's the result. So they recruited about 39 patients into this phase 1 trial. And you can see that the first, obviously, phase 1 usually is starting with safety. And then the phase 1B, basically, they kind of like enrich the population so that they can do some early indications. So among these 30 patients that we can get measurable tumors. 2 of them have a partial response, the green line, that means the tumor shrink after taking the combinations. There's 18 of the patients, the blue lines basically saying that the patient's tumor shrink a little bit between -20% and then go up to like 20% of growth. So this is considered as a stable disease. And then there's about 10 patients, the tumor after taking the drug continue to grow, right? It depends on how you want to see the graph, right? So the combined clinical benefit is basically complete response, partial response, and stable disease is about 67%. So there's a lot of things that you can say, oh, maybe cyclosporine is not the right drug because there's a lot of toxicity, not very targetable to the canonicals, right? Or maybe there's some biomarker that we can further refine between the green and the red kind of like patients, right? What is the unique thing? So this is something that we are continuing to work on, but I just want to give you some hints about how we kind of like use computational approaches to find drug combination. But one of the challenges is basically when you get all these hits, the Wnt pathways, how do we find the right drugs, right? So at that time, we basically asked around a lot of clinicians, read a lot of papers. So in a computational world, it will be ideal that if you have lookup tables, right? So I'm going to show you a quick 2 kind of like computational approaches that we developed that can do this kind of linking with gene lists to some drugs. So the first one is if you have something like a Google, right? If you input your gene list, you do a query, you get a ranked list back, right? So this is basically the idea that I kind of like inspire my research a lot. Is from this connectivity map. In the afternoon, you probably can hear more from Dr. Bin Chen. He's one of the pioneers of using these approaches. So the idea is assuming that you have a way to comprehensively characterize the gene expression changes. So assuming that you have cell lines, no drugs, you treated with this particular compounds, you can measure the gene expression changes, right? And then blue lines indicated that genes that is upregulated after treated with this compound. The orange line is basically showing downregulations, right? So you can do this for 1 cell line, you can do it for multiple cell lines, right? And then assuming that you have a query coming in, this is a cancer patient, this is normal patients, you get the gene expressions, you say that there's some genes that is up, some genes that is down from cancer versus normal. And then the idea is basically a matching, right? So you take this as your query, you try to find among all these genes that have been induced by different small molecules, how similar are one of these compounds induce the same gene expressions, right? For example, you can think about like a simple correlation. So this will be, this compound show all the genes that is upregulated in cancer, all the genes downregulated in cancer after treating with this compound. So it has a strong positive correlation. And then you can do this for the other way around. So this is basically after the cell line treated with this compound, the upregulated gene is downregulated, downregulated gene is upregulated, right? So it has a negative. So each one of these correlations, you can basically give a score, right? For example, +1 to -1, right? And then you can basically rank the results. So this is basically saying that if you want to find a compound that induces the same gene expression as the query, this will be the best compound, right? This will be basically the negative compound because it reverses, right? So obviously, if you want to treat a cancer patient, you don't want to give this drug because this is inducing the same cancer treatment. You want to find a compound that can reverse, assuming that reversing gene expression can cure cancer, right? But this is kind of like the concept. So we basically took that idea, and then at the time we are interested in kinases because this is one of the mostly mutated protein class in cancer, and then there's a lot of small molecules being developed. So over here, what I'm trying to show you that some of these kinase inhibitors, they are very selective. They only inhibit 1 kinase, right? But some of them could be like multi-kinase inhibitor. They call it as a dirty drug. So we see this as an opportunity to repurpose some of these multi-kinase inhibitor that they kind of like have some off-target to use it as a cancer treatments. So we basically build a very simple connectivity map. We call it a kinase connectivity map. Instead of using gene expression, we basically look at the selectivity of these kinases, right? So the idea is from your screen, you might have identified kinase XYZ as your target. And then the idea is instead of finding one drug that inhibits kinase X, we want to find, is there a a compound that can inhibit XYZ together, right? So in a very simple term, similar to the connectivity map idea, this particular compound have a high kinase inhibition selectivity against XYZ. This one have a low, right? And then you can basically rank them, things like that. So when we develop this, we have to test it out with some validation. So we use a lung cancer cell lines where have carry a EGFR mutations. We know that when EGFR mutation lung cancer, you prescribe EGFR inhibitor. But for this particular cell lines, H1650, it doesn't kind of like super sensitive to EGFR inhibitor single agent. So our hypothesis is maybe there's some other kinases driving the survival of this cell line. So when we perform the functional screens, we identify about 14 kinases. EGFR is one of them, but it's not the only one. And then we also look at the gene expression between the cell lines and a normal lung line. And then we found that when you overlap these two together, these are basically the 7 or 8 genes that is upregulated in cancer and then also be essential. So like I say, EGFR is one of them. We took this as our query and then we query our kinase connectivity map. This Gefitinib and Alotinib is the classical EGFR inhibitor. You can see that they have a intermediate score. This particular compound, bosutinib, is kind of like rank higher. The rank number 1 and 2 is basically pan kind of like kinase inhibitor, right? And then to show that we want to take this bosutinib as a validation, we pick another multi-kinase inhibitor that is ranked lower. So when we do the experiments, we want to see that bosutinib have lower IC50 compared to this one, the EGFR and another multi-kinase, right? So that's the prediction. So when we do it, it basically kind of like trend what we predicted. So this is the 1650 cell line. Bosutinib have a lower IC50 compared to EGFR, gefitinib, and the pan sorafenib. It also induces caspase. And then we took another cell line that is acquired this EGFR gatekeeper mutations, which is not going to sensitive to EGFR again. We kind of like see that they also kind of like have a better lower IC50. So like I said, one drug is not going to cure cancer. So we basically took the 2 drugs, bosutinib and EGFR inhibitor, and then do a combination assay, and then we see that there's some synergistic effect, right? So that is kind of like one kind of tools. The other one is basically we try to use it as a way to find drug combinations. So we developed this a molecular type signatures. Instead of looking at pathway where you use in gene set enrichment analysis, we basically use it for a drug. So that when you put in your gene expressions, you can basically get what is the top drug that you want to look for, right? So this is basically collected from different kind of databases for drug activities. And when we publish it, it seems like somebody is finding it useful, so they do a lot of citations. I just want to quickly show you one thing that my lab is currently doing is presently, before that, I only focus on kinase, right? This is very signaling pathway within the cancer cells, but we know that cancer cells is not just isolated in a human body. They interact with other immune cells. So we're basically trying to use computational tools to decode the ecosystem so that we can find ways to do the combinations. And I have to skip through a lot of this because the idea is we are going to use the same kind of concept where we find genes that is present in kind of like the cold tumors versus the hot tumors. We want to find a signature that can convert those signatures from cold to hot using computational approaches. So again, we're relying on the connectivity map. We identify some compounds, test it in some of these publicly available data sets, and then do some experiment validation. So this is something that my lab is currently working on. And as advertisements, Huntsman Cancer Center, as well as the Scientific Computing and Imaging Institute at the University of Utah, we are looking for a co-leader position in computational oncology and AI, which is somebody that will partner with me in trying to foster these kind of visions at University of Utah. We also have tenure tracks, junior faculty positions. So if you are interested, please come and look for me. I'll be somewhere around here for the whole day. For that, I'll thank you very much and happy to take any questions.
Thank you, Dr. for the exciting talk. We have time for one question.
if you look at molecular biology data, MEK inhibitors, they enrich cancer stem cells, and they also turn on autophagy. So when you predict all this combination and you don't know this ahead of time, how AI can help actually to figure this out before we spend years on with the bench work?
Yeah, hello. Yeah, a very good question. So the way that I show you here is we are using these gene signatures as a readout. So obviously there's other ways of measuring readouts, but here the concept is using the gene expression. So the idea is all these signalings at the end is going to make some changes in gene expressions. And for these gene expression changes, we have high throughput, systematic way to measuring them. So that is kind of like the way that we read out to use as a whatever mechanism that could be happen if the gene expression is a way that we want. For example, certain gene set is going up or certain gene set is going down. That is kind of like in the example that I show you, that will be kind of like the readout that we use to predict, right? So you can use in-house data, you can use a lot of publicly available data sets, like all these large scales, cancer cell line encyclopedia, the genomics of drug sensitivity. They screen a lot of multiple cell lines with different drugs. They have, some of them have pre and post treatments data. Those are the ones that we use it as a way to find similarity. At the end of the day, AI is trying to find similarity between whatever that you want to predict, right? Obviously, I didn't include dosage, and then there's no toxicity involved, right? This is purely just based on certain genes going up, certain genes going down. There's room for improvement, obviously.
Yeah, let's thank Dr. Tan again.
Thank you.
Our last speaker today is Dr. Christopher Whaley. He's a highly Stephen Royal Endowed Professor, Associate Vice Chair for Translational Research in Radiation Oncology at UAB. He's also the Canon Core Director at Department of Radiation Oncology. Dr. Whaley received his Ph.D. from Medical University of Southern Carolina 2023. His research spans the central nervous system, head and neck. Today, his topic is pharmacokinomic testing in glioblastoma. Let's welcome Dr. Whaley.
All right, thank you. Thanks, Jake, for putting on this nice symposium. All right, so what I say now is I'm usually the chief spear catcher. I took that from Yancey Gillespie, used to be in neurosurgery here. The people who actually do the work is actually not me, so I always want to thank these people. So I'm including a postdoc in my lab. Manoj Kumar is doing a lot of kind of the drug testing of our models. Josh Anderson, who runs our Kinome core as a technical director and as a scientist in my lab. And then Ihsan Sagapur from Jake's group has helped us on some of the bioinformatic approaches as well. So quick disclosure. All right, so I'm a little bit more disease focused. I am a clinician, I'm a radiation oncologist. I was watching you guys online at the beginning because I was doing some radiosurgeries on a couple of brain tumor patients. But one disease that I do treat is a tough one, is glioblastoma or GBM. It's the most common brain tumor that originates in the brain. Most tumors that are in the brain that are cancers actually secondary. They come from like lung or breast or somewhere else. We struggle to treat them. We've had some improvement in the last decade or so, but we're still looking at 15 months of survival after diagnosis. Our initial approach is to try to cut out what we can safely without making the patient look like a stroke victim. And so sometimes that may be just a biopsy. And then we follow it up with my modality, which is high energy radiation, X-rays, with a chemotherapy called temozolomide. There's a device that actually did some research on as well that does alternating electrical fields afterwards to try to disrupt mitosis and so forth. But the tumor always comes back. And so part of the problem we've faced over the years has been, because people have been studying this for a long time and really don't have a whole lot to show for it as compared to other cancers. And there are a few different reasons for that. And so some that I'm going to touch on today is related to how we model them. And so we usually use highly artificial systems. So I'll talk about that. sometimes can't manage the tumor microenvironment very well. And then the blood-brain barrier, which is what keeps some drugs out of the brain. And so there is certainly a lot of work that machine learning, artificial intelligence, so forth, they're really helping us on better dealing with these issues. The in vitro selection issue comes down to how you model it. And so we've taken an approach much like the Huntsman group. Actually, I'm on a consortium with Brian Whelm from Huntsman on breast cancer, but we're doing it with GBM. And so we've developed a bunch of models that we can grow in different ways. You know, how you grow them can alter their behavior, but I'm going to talk to you about how we're looking kind of a little bit farther down the central dogma at functional proteins, which are really the target of most drugs. So if you look at how most people have tried to generate a cell line model, this is the approach. You take it from the patient and you extract. So this is what we call a sagittal MRI, T1 plus contrast. And this is the tumor. It's heterogeneous. That's because there are parts of this that are kind of dying or dead, and then you've got parts that have leaky blood vessels around it. But anyways, you take the cells and you grow them in a dish. And then once you have them, it's a cell line. You can propagate them. You can put it into the flank of a mouse that's immunocompromised, and it will form a tumor. You can put it into their brain, and then it's now an orthotopic intracranial xenograft. The problem with this is that the way this tumor develops is in a very harsh environment. You have acidic conditions, sometimes very low oxygen, low nutrients, but that's the exact opposite thing that you do when you grow them in culture. You tend to give it buffered media, you put a bunch of oxygen on it, you give it fetal bovine serum, a ton of sugar, and very quickly, the fastest growing cells will basically take over. And so it becomes a very nice model because the cells are homogeneous, but it's very different from the patient. So we've focused more on patient-derived xenografts, or PDX. You heard about them in the last talk. But this is basically showing an example. So a classic GBM model is the U87, which is one of these immortalized cell lines. So if you put it into the brain of a mouse, this is an H&E purple being tumor, it looks like a marble. It's basically a very sharp border. This can be cut out. You can do radiosurgery like I did this morning for a patient. It's not very invasive. This is what a metastasis looks like. When you take a tumor that's a PDX, put it in the brain, it infiltrates in the brain just like a patient's tumor, which is, you know, difficult to target, difficult to cut out, and so forth. When we've looked at them, like groups of GBM cell lines versus patient tumors, just from microarray data, even back in the day, equivalent genes are shown in red, and there are a ton between cell lines and patient tumors. The problem is these ones that are upregulated only in the cell lines tend to be very druggable, like PDGFR or, you know, certain cyclin-dependent kinases. So when you would try to target this in the cell line, it works great, but then when you give it to the patient, it doesn't do anything because it's not altered. When you start doing a PDX, they get better and better. So with that context, this is kind of the program that I got connected with when I first came to UAB, mainly through Yancey Gillespie, who was running the brain tumor animal model core. And so I basically talk about these as PDX versus derivative models. The term they use is probably patient-derived models of cancer. We used to call them xenolines. But basically, we've taken these and tried to profile them. The challenge with making one of these and applying it for the patient, so some people use the term avatar. I'm going to grow out a tumor from you, and then I'm going to, you know, blast it with a bunch of things and then give you information. By the time I developed this, the patient may have already died, or the tumor's progressed or changed, and so forth. So what we've tried is really to build a panel of models that are representative of the disease. And I think we've got a pretty good group now. It keeps expanding. So what I'm going to touch on here is the functional proteomic approach that we're utilizing for this in the context of all these other multi-omics that you're hearing about today. So the platform we use is called the PAM station. It's from the Netherlands. And the way this thing works is you basically have an array of phosphorylatable peptides that are embedded within an aluminum oxide matrix, about 60 microns thick with lots of surface area. And basically you have these amino acids that have a phosphorylatable residue like a tyrosine or a serine, threonine. And what we're basically trying to do is measure the transfer of phosphates to those, which is what a kinase does, and do this from the lysate in, the patient tumor, your xenograft, whatever you're looking at. And so we get kinetic information because this is a reaction, essentially an in vitro kinase assay, and we can measure the pattern of basically this phosphorylation event. So sometimes clinicians I work with or scientists say, hey, I've got patients at different outcomes. What can we do with this information? Well, sometimes we have like fingerprints that may associate with an outcome. So we do like supervised analyses. We say, okay, this patient, did very bad. This patient did very well. What's different about them? We've done that with our PDX where we've come up with kinomic subtypes, and we've tried to match that up with, let's say, a transcriptomic thing. And they don't exactly match, which is not that surprising. One of the cool things that we can do, which I'm going to touch on now, is because this is an enzymatic assay, I can spike drugs into the system and see what changes. So to kind of look at that, the idea is, we're basically taking these pictures every time we pump this material through, and you see this pattern come up over time. So here's early versus late in the reaction. Well, what I can do is I can spike in a small molecule and see this is with, vehicle, but as soon as I add in the drug, I now suppress that single spot on this chip. So we can use that information to try to predict the kinases that are targeted, because we're really reading out the substrate, basically what these kinases can phosphorylate. And it's very promiscuous. That's the challenge with kinases. So back when we first started to test these models, Tika Benveniste with Braden McFarlane, who's in cell biology here, basically had a drug that targeted STAT3 phosphorylation downstream of JAK in GBM. And so they took 2 of our PDX, actually took a third one that basically looked more like this one, and essentially had this treatment, this a Kaplan-Meier curve. And so they got the drug for this amount of time. This tumor did really well. They actually had to sack half the mice at a half year because they had no sign of tumor. They would have lived, till old age. This tumor had the same kind of protein-based match as this one, but it barely did anything. It added a few days. And so what we did is we looked at their baseline kinomics among these, between a low sensitivity like this one or the high sensitivity one in the intermediate, and we saw how they kind of grouped out, identified some potential substrates that would predict perhaps, and then matched it to a larger group that we'd already profiled. And we seem to see groups that come together. We also tried the spiking, where we took this drug and spiked it in. We saw that probably even cleaner system where the low sensitivity one, really did look quite different from the high sensitivity. So we started doing ex vivo profiling. So the thought was, could we do an ex vivo test and predict sensitivity? And so we got an R21 to kind of go after this. And so we have a lot of models that we've developed, and this is just showing, a group of them and just some example drugs that we selected that hit some targets that we thought were important. And then, some of these are actually being used in clinical trials for GBM patients. So we know they get into the brain. So the approach that we've taken is a few different ones. And so this will real quickly just show how we can group these drugs based on the change on the PAM station kinomic profiling. And so we were able to use a partial least squares discriminant analysis to really separate out across a number of tumors how each of these drugs might be, and try to create, perhaps like an index to say, okay, this drug seems to be effective in one tumor versus another. And this is simply based on the kinome, not based on the drug testing yet. So then what we want to do is, can we visualize this? So this is some work with ASON where we took one of these tumors and we try to use essentially a kinome terrain, much like Jake's gene terrain, to display these. So there's basically a lot of different probes, and you can change the sigma to look at this. And so ASON's been working on creating a nice visualization tool where we can basically examine this data and see, really look at these curves and try to find regions that seem to associate with a particular sensitivity to one drug versus the other. Then what we do is we start testing the drugs and finding, okay, are there certain drugs that are particularly resistant or particularly sensitive? We're going to find out, okay, is there something about their kinome picture that's going to be relevant? that perhaps could be a way to test. And why is this important? Well, if you're a breast cancer patient and you have a breast tumor, they can put a needle in it before you start treatment, maybe put a needle in it after. For a GBM patient, most patients don't say, yeah, why don't you cut my skull open and just, poke around in there. So it's often difficult to get a second or third sample from a patient who has a brain tumor. And so we think that maybe there's some information that we can interrogate from that original sample using a technology like this. So some of the other things that we're working on is, in addition to this tool, I think we'll see if this video even works. No. Just trying to come up with computational tools, validate these and, in these PDX, maybe in animals, because some of these could get into clinical trials with the right preclinical data. And then how does this data fit in with all the other things we can do, like, quantitative mass spec, proteomics, RNA-seq, epigenetic evaluation. There's so many things that go into the biology of these tumors. How do these all fit together? And we're doing longitudinal studies. You heard about that as being important as well, because these things are not going to be static either. So that was the whirlwind tour. And so in terms of looks like a couple of these things didn't exactly transfer perfect, but anyways, a lot of people involved in this, and I can take any questions.
Now in cancer, we have a lot of novel targeted therapies, and a problem what you compare to naive tumor, yes? But meanwhile, this patient got a lot of therapies. So we are comparing two cells what are completely different. Any thoughts how we can improve this?
Yeah, so I totally agree with that. So the Cancer Genome Atlas had that same problem. Most of the time they were taking the original tumor, not the one that actually killed the patient. So there is a dearth of those type of models. So we actually set out to do that. So part of the work that I've done with Jake and like Anita Gemlin and others is we actually started developing resistant models to standard of care. And so we published a few papers over the last few years. I didn't have time to show it, but we've taken our tumors that were initially sensitive, let's say radiation or temozolomide, and drive them to resistance through in vivo selection of these PDX. We've then gone back and looked at patient databases, both and then even our own tumors, like tumor tissue microarrays we've generated here, and we're seeing a lot of the same alterations. There is a consortium called the GLASS Consortium for GBM, where they did look at patients longitudinally with omics, and in general, at the genome level, most alterations are retained. They don't become like hypermutated per se. It's usually something more downstream, which could include kinomics. But we did, we've seen a few things. We published that we found this chromosome 12q14 amplified in 25% of the radiation resistant ones. We're seeing that same thing show up in our patients here. We're seeing the same thing in some other published studies and databases. So there may be some things that we can model decently and test, but we're really struggling for this disease. And so I think you have to make some of these more robust resistant models. The other thing that the PDX also captures is that there's this thing called a brain tumor initiating cell or cancer stem cell. They tend to be enriched upon treatment, and they're what kind of survive and repopulate and grow. And these PDX tend to preserve them nicely. And so you'll probably hear a little bit more about this maybe with some of the flash talks when I saw the list. So
let's thank Dr. Wade again. So this will conclude the first research session in the morning. What's the next?
No, flash talks. Flash talks.
Flash talks, yeah.
Great.
Yeah, my name is Chindo Hicks. I'm from the University of Louisiana State University, New Orleans, and I'll be chairing this session. So we'll be transitioning to the Fresh Talks session. First, I want to thank Jake Chen for including me in this. I did very little work except driving here, but other than that, all the work was done by Jake and the other members of the team. So without spending much of the time, so the speakers have about 8 to 10 minutes of making the presentation, and then about 2 minutes of giving the answering the questions. So without spending too much time, our first speaker will be Dr. Sharma, the informatics architect from the Department of Biomedical Informatics and Data Sciences at UAB. Right, that's you? All right, welcome. All yours.
Hi, everyone. I know lunchtime is coming up, so I won't take much of the time. I'm Rahul. I was a postdoc when I was doing this work, and this is something, it is twofold work. One is building a library. So it's a computational library where we are focusing on extracting meaningful features from drug smiles. Second thing is looking for collaborations. If you are a programmer, we would like to connect with you, and we want you to be with us and build the Python library that we are developing. This is my walkthrough. So I would like to introduce why drug smiles, and then give you an overview of the Python library. What are the key features and capabilities of this library? So I'll be speaking non-technically. I'll be talking in a way that biologists, mostly biologists, will understand that why do we need these kind of work, which is in silico. And explanations will be fairly simple so that you can correlate with the real world things. And then I would like to showcase a use case. Actually, the use case is outside in the poster. I'll just show it. It's a digital twin work. And then how do we use this library, and some closing remarks with a question and answer session. So why smiles? It's easy to store. So you can see compact representation. A smile is textual, it's continuous, and big molecules can be represented in a small text and can be stored in normal databases. Scalability in handling the data. So when the storage is easy, we can very easily store or scale it, like wherever we want to use it, in a web app or something else. Integration with databases like and the cheminformatics tool. So we use lots of cheminformatics tools like RDKit library. So what's the purpose? To understand the molecule structure and perform different kind of analysis on those structure, develop new drug or repurpose the drugs. So it can be very easy with this notation. The most important two ones which I'm interested in is virtual screening, or I also call it clinical trial or virtual clinical trial, or a digital twin, and AI and machine learning application. So that's my main focus. I'm not a biologist. I'm a data scientist by profession, and by training, I am an engineer. So I'm more interested in building AI applications so that we can help build a better healthcare. So this is how drug-smile looks like. On the left side, it's a pictorial view of a compound, camptothecin. Did I spell it correct? So this on the right side, we have several characters written. So in the red box. So this is the smile representation. This representation is also not used by the computer. So this is a smile notation which is user-friendly for us to understand. We can discriminate. We can interoperate between this notation as well as this operations if we are doing if we are using the cheminformatics tools. But for machine learning, we need something else. So does machine understands this when it comes to AI? No, it does not. Well, with the advent of LLMs, or we can also say GPT, ChatGPT, we can really do those things. And many people are working on it. But originally, it only understands feature vectors like Excel sheets. So we need to extract meaningful features from these drug smiles. This is what we have done in this library. We're actually integrating many methods. One method that we have created in the lab, in AI Med Lab, that is from where it started. Now we are integrating large number of methods where we can extract meaningful features. You bring your smiles, we are going to give the features. It's user-friendly, and you can directly integrate into your pipelines because it can be used as a Python interface. We are also developing an API, so whichever language you are using, you can use it. So this is the library, the purpose of the library and the design, as I said, and how it is bridging the chemistry, the gap between the AI and the chemistry. So as I said, molecular structures are represented very subtly in using smiles, and we can extract meaningful features. This library is going to, the main vision of this library is this, how it is going to bridge the gap between the chemistry and the AI. So if you understand chemical structures and you know how to represent them in smiles, you can easily use this library to integrate it with your AI applications. So AI applications feeds on data. And if you want drug data, you can use this library. So you can extract features, and then you can use those features in your AI models. Key features of this library: extracting features, obviously, drug-smile-fet, feature extraction tool, integrating with machine learning pipelines. So right now, we are developing this, but you can integrate it as a method in your Python code. But in future, we'll have functions where you can simply plug and play. So it will be very user-friendly, ease of use. So that's the main purpose. We want this to be used by the biologists. So we are building visual tools so that you can use it in your own system for visualizations purposes also. This is the recent paper that we have published over this, which is based upon this methodology that we have developed from where this library got started. So it's a very simple way of using natural language processing and extracting meaningful features. So these features are extracted the way the animation is displaying. So I won't get into the depth. If you really are interested, we can have a discussion on the poster. And Morgan fingerprints. So right now it has two kind of features. One is based upon Morgan fingerprints, and through my experiments with the NLP-based features, I have competing results. So we are working on build more algorithm on using NLP, natural language processing, and bring those methods over here besides methods developed by other researchers. How to use this library? Three simple steps if you are a Python programmer. Prerequisite, installer RDKit library. Simply in the command line, run the pip install drug-smile-fet. It will install the library in your system. And then you just import there is only one module right now, which is FE_1mol. So one molecule infrastructure. And it imports both the methods, RDKit, that is Morgan fingerprints, as well as the NLP-based method that we have developed. So very simple if you are using Python programming. Other than that, the first one I have already described. The down one is the interface that we have developed. We are about to put it online through UBright. As a tool, it will be available internally to UAB because the UBright platform is under development. And only people who are using UBright can use it, but we can also make it available through different portals, or they can download it through a GitHub library and run it locally. So it's an open source tool, they can use it. Importance of drug discovery, as I said, there are many why we are interested in AI-based drug discovery. digital twin. I'm personally interested in this, drug targets interaction, toxicity, accelerated drug discovery. So these are a few that I have listed. And why we are interested in this, faster drug delivery. So not drug delivery to the patient, but to the market, because testing, identification, all these processes involve a lot of time. If you do the screening in silico and identify the right drugs for the right disease, it's really amazing. And I think AI is it's playing its role. A lot of people are adopting it. This is the poster that we are presenting with Dr. Deepal is there presenting the poster along with me. So we can tell you how and what is a digital twin and how you can use it in clinical trials. So it's a kind of virtual pre-clinical trials. So I'm using the terminology because many of your wet lab professionals, they must be doing preclinical trials like mouse models or cell lines. How can we do those things in silico? So in the poster, we can have discussions on that. And I have some great results, so we can actually do some real world examples on this. I would like to conclude, library, the main purpose of the library, extract features, engage users, collaborate. You can collaborate in a very subtle way, and integration with AI. This is our team, and right now we have 4 contributors. We are looking for contributors, as I say. So if you're a programmer, want to be part of our team, please volunteer. Thank you so much. Any questions?
Yeah, we are. We have time for at least one question.
Have you compared your fingerprints or list of factors with the traditional structure-based fingerprints or the advanced like graph neural network-based fingerprints?
The results in the industry? I didn't get your questions correctly.
Yeah, so I saw your output from your NLP. You get a list of features, which can be used to build machine learning models, right? And those features can be extracted using traditional approach like structure-based chemical fingerprints or the more advanced one, a graph neural network. Have you compared yours with others?
Yes, I did. And we have the competing results. And right now we are using only one molecule sequences. We are interested in multiple molecule or sub-molecule sequences. So if you have CH3 coming up 3 times, we consider it as one sequence. And we just want to identify 3 molecules, 3 molecule sequences there, and how many are coming in. So this is how NLP works, natural language processing. It identifies the sequences. So in a temporal manner. So how can we use those sequences to make to build an understanding of the conversation going on? Same thing is over here. So how those small molecules are related, how often they are occurring. It's a feature, and we can use those. And right now we are using only one molecule, like carbon, hydrogen, nitrogen, all those things. How often they are occurring in sequences.
All right, thank you. So our next speaker is Dr. Brittany Lasney, Assistant Professor of Cell Developmental Biology, Developmental and Integrative Biology from the University of Alabama. Did I remember your name?
It's fine. It's a tough one. It's Lassane. It's a tough one. So thank you so much for the opportunity to speak today. I'm excited to tell you about our work prioritizing repurposing candidates to avoid adverse events associated with sex and tissue-based drug targets. So our lab thinks a lot about context. So what is the context in which a particular variant or a suite of genes is acting? So for example, if you have a Mendelian disease where that variant is present in all the cells of the body, how come you only have a clinical manifestation in certain tissues? Or thinking about different patients who take a drug and it works well for one, and the other, maybe it works well, but they also have severe adverse events. So those are the kinds of things we think about. So this is a figure from a student who's now graduated in the lab, kind of outlining some of these different mechanisms of cell and tissue specificity that we think about. But there's also additional factors like species, sex, and age, some of which we've touched on here today, differences between cell lines and animals, and other extrinsic factors like diet, exercise, or environment that impact the molecular and clinical manifestation of diseases. So we work on many things in this area. So if you're interested, please come find me at lunch or check out our papers. But today I'm going to focus on some of the sex bias work that we've been doing. So drug adverse events are any unintended effect of medication. And we know from prior pharmacovigilance studies and clinical trial retrospectives that women are about twice as likely to experience adverse drug reactions compared to men, but men are more likely to experience severe adverse events, so those that result in hospitalization or death. So sex bias adverse events are those that happen disproportionately in one sex compared to the other. And there are lots of different factors that can lead to a sex bias adverse event. So I've shown you some here on the screen. And this is from a review that we wrote recently in Biology of Sex Differences that you can check out if you're interested. And so we really were starting to think about, well, what is it that's in common between these drugs, and can we learn something there to help us prioritize for preclinical and clinical studies moving forward? So we did that, and that work has now been published as well. Given time, I'm going to just tell you one tiny little piece of it and then kind of show you where we're going with that moving forward. So our hypothesis for this study was that we really thought that there were going to be common drug targets and differences in gene expression and gene regulatory features at these drug targets between the sexes. And so we did a lot of work showing that in this paper. And like I said, I'm just really going to focus on one small aspect and how we're moving forward with that. So just to be clear, we use data from the FDA Adverse Event Reporting System. So this is a database that contains adverse event reports, medication error reports, and product quality complaints that result in adverse events that were submitted to the FDA. And it's designed to support the FDA's post-marketing safety and surveillance program for drug and therapeutic biologic products. So I'm going to tell you just very briefly about how we identified those drugs and about the drug targets. And so we did extensive filtering from this database. So for example, we only included reports that were submitted by medical professionals. We only included adverse events if they had been identified multiple times. And the adverse events had to be something that was possible in both biological sexes. So we had a lot of different work that went into filtering these down that you can check out in our paper. But at the end of the day, in line with previous studies that used older iterations of this data, we identified a more female-biased drug adverse event pairs, so a drug with an adverse event that was sex-biased, than male-biased drug adverse event pairs. And here's a volcano plot plotting on the x-axis the log of the reporting odds ratio, and on the y-axis the negative log of the adjusted p-value. And so you can see in purple are female events, and in teal the male events. So in all, we identified 164 drugs that had female sex bias adverse events, 44 drugs that had male sex bias adverse events, and 208 drugs where they had different sex bias adverse events. So for example, perhaps a neurological adverse event in males and a gastrointestinal adverse event in females or something like that. So what we really wanted to do next was try to identify if any of the known drug targets or enzymes, drug metabolism enzymes that these drugs work with, if any of those were enriched in this set of drugs compared to random sets of drugs. And through that analysis, we identified 84 drug targets and 32 drug metabolism enzyme genes that were enriched for the sex bias adverse event drugs compared to randomly selected drugs. So there's a bunch more work in the paper that I don't have time to go into today, but we do a lot of work with gene regulatory networks to look at tissue-specific bias as well as gene expression bias. And the punchline is we found that most of these drug targets that were in these sex bias adverse event drugs did have either sex bias gene expression or gene regulatory properties. So this suggests that sex bias adverse events may be, at least in part, due to gene expression and regulation differences between the sexes. So this has been shown in limited cases before. What we did here was kind of look at it in kind of this broad view. So the thing that I want to focus on here for the last 2 or 3 minutes of my talk is, so how can we use this information? How can we leverage that to improve preclinical and clinical trial success rates? So can we use this information to help us better prioritize drug targets and candidates based on their known associations with sex-biased adverse events? So similar to what you've already heard this morning about how to identify candidates. So one of the things we do in our lab is think about drug repurposing candidates. And you guys have already heard this explained by at least 2 of the speakers this morning. But this idea of developing a disease molecular signature and then using these large libraries of thousands of cell lines treated with tens of thousands of drugs or other perturbations to try and identify those drugs that will most reverse that disease profile. And so we've done this successfully, including in vitro and in some cases in vivo validation of the drugs in 4 of the lowest survival human cancers, as well as in polycystic kidney disease. But what we can also do is kind of flip the question and say, instead of just looking for drugs that can treat a disease, can we look for drugs that can mimic something good? So an example of that would be exercise, an exercise mimetic, exercise in a pill. So we all know that physical activity is really good for us and has a lot of protective effects, particularly neuroprotective effects. So we have some funding with a collaborator at USC to identify these exercise mimetics and then test them in rodent models, both as an exercise mimetic and as a protectant against Alzheimer's disease. And so what's cool about this is that in addition, you've heard about developing these disease signatures today. We're working on different ways to further develop these initial disease signatures by using different machine learning and transfer learning approaches to better identify that starting disease signature that is going to be effective across both preclinical and clinical models. And so we have funding from a couple of sources to do that now. And we have a few papers out that you can check out that kind of get at pieces of this as well. But some preliminary, whoops, looks like I got messed up. Some preliminary data that we have produced in this idea of exercise mimetics. We're waiting on some additional data to incorporate in our model from our collaborators. But if we just think about some of the 3 of the top candidates that we've identified using this methodology and think back to the sex-biased adverse events. So this first drug here, listed drug 1, is a known exercise mimetic. It's a building block of testosterone. And when we look at our information about drug targets and enzymes that are associated with sex-biased adverse events, we don't have any of the known drug targets or enzymes that process this drug. So this might be a point in favor of this drug, given limited time and money to follow up drugs. Oftentimes we're getting dozens or hundreds of these drugs. So how do you pick 3 to move forward with in a preclinical study, for example? The second drug here, we don't actually know anything about its drug targets or its drug metabolism enzymes. We just know some different pathways it's associated with perturbing. So that would be no added information. But this third drug here, we actually know based on our prior research that several of its known drug targets and drug metabolism enzymes are enriched in our sex bias analyses. So this might be something that, given limited resources, we would deprioritize for follow-up. So just in summary, I've shown you at least the very first part of our work demonstrating that sex-biased adverse event drug targets and drug metabolism genes were differentially expressed and regulated between the sexes. And we think that these findings, especially combined with cell tissue and species specificity, might really be useful for prioritizing drug candidates for future studies. So our ongoing work, we are continuing to validate our predictions in preclinical and eventually clinical trials. We've set up some wonderful collaborations here with both clinicians and folks who work with model systems. We're continuing to develop our drug identification approaches and prioritization algorithms. And we just found out Friday, it's embargoed, but we got some funding from a foundation that's going to support us leveraging drug combinations to improve efficacy and avoid adverse events. So we're really excited about that. So this work, we have fantastic collaborators and our funders to thank. And then, of course, the team. I think everybody here on the screen contributed to at least one of the studies I talked about or alluded to. And we've been fortunate to have multiple NOAs in April. So we're hiring if you're looking. Thank you. Excellent. Yeah, 10 minutes on the dot.
I assume if there is sex-biased effect, so we're talking about hormones, okay? Hormones, they have very cyclical levels, and their level is 100 times higher during morning times than evening times. Do you have any thoughts about when it's better to give drugs due to these phenomena?
Yeah, I mean, I think that's a great point. So if you didn't hear, the question was about essentially the role of hormones and when to give drugs and how to think about sex bias adverse events in that. So, so there are a lot of studies that show the time of day that patients take drugs does matter. And so I think that that's something that's underappreciated generally, specifically with effect to hormones. While we know that some of the sex bias adverse events are certainly tied to hormones, there's a lot of research too that shows that it's at least not all the effects are not directly due to hormones. And some of these studies involving different mouse studies where we're looking at surgical and genetic interventions. And so we know that it's not 100% driven by hormones, but yes, the time of day is going to be really important for these avoiding these events too. Yes.
For example, those type of method normally would discover like drug can be repurposed, but not backed up by any existing evidence of research or even any pre-existed in vitro assay result. And how is your view on the new starting point? Do you think they have a great potential to discover something new, or it could be just the noise in the model? Like how do you perceive?
Yeah, so I think the question is around the fact of how transfer learning or even other computational methods that identify a drug for a new use where we don't have any literature support or anything else. Yes. So we certainly have found those examples and we've gone forward. Dr. Willie spoke earlier about some of the GBM work. We've been working with the Gemilon lab, and we've actually shown some of those drugs where there was no prior support even for use in cancer, that we've had some demonstration of efficacy in PDX models. So we have another study that we're working on right now in polycystic kidney disease, where we think there's drugs that you would not expect to target. They're known for their action in the brain. We think that they might really be efficacious in the kidney. So I think that there's probably a lot of noise to sift through. And so I think that's where having multiple different data sets, multiple different approaches where you're identifying a drug, and then there's just no substitute for the downstream preclinical testing. And so that's why we're so focused on like, how do we find our best drugs for that? Because I'm sure, as you know, it's similar for you guys as you find dozens or hundreds of drugs that rank well. And so then the question is, which ones do you pick to move forward? Because these mice and rat studies are tens of thousands of dollars. So that's where I think some of these things like sex bias adverse events and things like that can help you also prioritize better. All right.
All right.
Thank you.
Very much. So our next speaker will be Ethan Sagapo, a postdoc fellow in the Department of Biomedical Sciences, Biomedical Informatics, and the Data Science Department at UAB. All right?
Yes, good. Thank you.
Right?
Yes, Sagapo. Hello, everyone. This is Ehsan Sagopur. I'm working as a postdoctoral fellow at UAB in Dr. James' lab. Today, I'm going to talk about my current research from statistic to dynamic temporal visualization of gene expression in cancer cell line. Traditional gene expression visualization techniques such as heat maps, clustering, bar charts, and box plot are foundational, but limited in showing dynamic gene interactions. We developed a new way to see how genes in cancer cell lines change over time after treatment, which we called a temporal gene terrain. This is the method. We create a framework for constructing temporal gene terrains involving two steps. The first step is generating the gene terrain from visualization using network biology of genes and the gene signal. After correlation between genes Then we create gene-gene interaction network. Then we map Gaussian distribution for each node or genes into 2D Gaussian distribution. Then we call the gene terrain. If there is a data based on the time series, like time course, we can create a GIF file or video clip to show what happened genes during time. So we call the temporal gene terrain. This is the one case study about this method. And we can use the, instead of the another visualization like the heat map. This is the, I applied for the one cancer cell line in prostate cancer, treatment with DMSO as a control test, and the 3 drugs and their combination for a different time interval. For this approach, first of all, I selected 1,000 genes that have a high variance. Then I calculate the correlation between them, and using the threshold, the 0.7, then I selected 590 genes to generate network biology. Then we create the network, create the gene terrains. As you can see, when we use just individual treatment, there is no changing in the pattern into the gene terrain based on the 0 hours and the 24 hours. But when we combine together the drugs, it seems that there is a you can see some of the pattern in the 0 hours that play as a downregulated. In the after 24 hours, it will change and play as a upregulated. This is the show about we can create as a GIF file, and the first row is show about vehicle control and the 3 drugs, and the second row is related to the when we combine together. And it's very easy to follow what happened for the genes during the time. This is the after that we can use the we can analyze this pathway enrichment analysis, and this is just one example for when we combine drug T and drug M. And there are 2 heat maps. The right heat map is about which one of the figures, which one of the genes that play as a upregulated, and we extract the pathway, and the left, the left side is about the upregulated, and the right side is about the downregulated genes. And we can see that some of the pathway appear all of the time points, but some of the pathway just appear in the last times or the first times. And for validation our work, I used another cancer cell line, and there are 3 cancer cell line, and is related to the GBM cancer, and were treated with 1 molecule per liter DBCAMP for 3 days. And this area, for the first tumor cell, we can show some of the genes that this area has very upregulated. But when we compare between the U87 and the LN18, the change, the variation of the change is not very, when we combine, when we compare the first one, it's not very obvious. And also we create the application for our work, and now there are I found 4 data set, and this is the just about the validation our work, and the user, after finding which one of the genes are sensitive, after using the lasso selection, find which one of the genes, then extract the pathway. Then we can create, find the pathway based on the bar chart and bar chart. Then we can create a pack-to-pack network. And we believe that the temporal gene terrain generated profile revealed several key insights. Firstly, this knowledge could advance targeted therapy development by considering the time depends nature of gene expression in drug response. Secondly, the identification of genes that are either sensitive or resistant to treatment at different time interval is a significant advancement. And also, I would like to thank my supervisor, Dr. Jake Chen, to support my research, and also the UAB. Thank you.
Right, so we have 1 minute for a quick question. Are you ready to answer the question? Anybody want to?
Just have a very basic question. So in your visualization of the gene terrain, do you have the ability to actually distinguish or label which genes or pathways are up or down? So it would be really cool to see exactly like the red spots, what kind of pathways are up or down in like a real time. Do you have ability to do that?
For this one, no, but we can add, for example, which one of the part is about the pathway. This is your question?
Yeah, so my question is like when we are looking at this picture, at this image, right? And it's changing.
Image and we convert to the GIF file or video.
Would you be able to annotate those spots? Like what pathways are these or genes?
Yes, we can do this one, yes.
Okay, thank you.
All right, moving along. So our next speaker is Dr. Ilana Chavez. Okay. Oh, that's right. So. Still here? No. Thank you, Ms. IT. Thanks. Yeah, so our next speaker is Dr. Manika, she's associate professor at the Mesa University School of Medicine. And she's going to talk about
Doesn't move. Oh, you just go with this. Okay, thank you.
All right, over to you.
Sorry, thank you so much for your introduction. So in cancer, unfortunately, perfect chemistry is not sufficient. Majority of novel cancer therapy, what they do, they just prolong disease-free survival. They do not prolong total survival. So for a short time, patient feels better, but then even novel therapy or no therapy, they die same time. So I'm going to present very basic biology explaining where we have to pay attention and why it happens. Ovarian cancer is one of the deadliest disorders. Unfortunately, due to being diagnosed very late, the use of cisplatin paclitaxel, which are developed in late 70s, disease comes back very soon. So initially patient respond, and within a few months, it comes back. Why? We think due to existence of ovarian cancer stem cells, which are very chemoresistant. So chemoresistant ovarian cancer stem cells They have different functionality. One is to initiate tumors in vivo, survive in 3D form, and they are very chemoresistant. We've used a variety of markers. One of them is aldehyde dehydrogenase, several stem cell markers such as SOX, nano, KLF4, and CD133. So as you can see here, cancer stem cell can differentiate and they can propagate tumors, but they also can keep their stem cell properties. Okay, so this is not sufficient to kill highly proliferative cancer cells because we always have these progenitors which can differentiate asymmetrically and keep their stemness, but can also differentiate asymmetrically, become regular cancer cells, highly proliferative, and propagate tumors. Okay, so in our lab, we also looked at MEK because MEK is very relevant to cancer. It's involved in differentiation, propagation, proliferation, and there are drugs which are FDA approved for MEK mutated BRAF, KRAS mutated cancers in different cancer, melanoma, colon cancer, and so on. As you can see, despite lack of mutation in upstream targets such as KRAS, BRAF, many of MEK pathways are amplified in ovarian high-grade serous ovarian cancer. So due to this amplification and relevancy and activation of this pathway, we decided to check one of the MEK inhibitors, trametinib, as I mentioned, which is FDA-approved drug for melanoma. As you can see in ovarian cancer cells, these inhibitors can block proliferation of MEK and ERK, and you can see also this based on downstream targets. But unfortunately, we see enrichment of cancer stem cells, okay? So from one side, initially we block tumor growth, but when we develop resistant cells, these resistant cells are not sensitive anymore to MEK inhibitors. And what is important, they overexpress cancer stem cell markers. So one of the cancer stem cell marker which is overexpressed following MEK inhibition is aldehyde dehydrogenase. Aldehyde dehydrogenase was my topic of research during my postdoctoral studies, and we actually developed a pan-LDH inhibitor which induces necroptosis in ovarian cancer stem cells. Why it's important, which cell death pathway? I'm being asked about this very often because as you can see in this picture, this is very dynamic. The same molecules, when they combine in structures, they can cause apoptosis, they can cause necroptosis, but they also can go to pro-survival pathway. And the only difference, there are post-translational modification. So when we look at LDH inhibitors, we can see a phosphorylation in RIP kinase 1 and RIP kinase 3, which are key factors in necroptosis. And you can see key differences in morphology. Following necroptotic cell death, cell becomes bigger, there is rupture of cellular organelles, and there is release of cellular content. So you can see as we use the LDH inhibitors, they actually decrease the cancer growth both in vitro and in vivo. So when we thought about combining MEK and LDH inhibitor, we thought what both of these hits will be very successful. And indeed, you can see this is the synergy map. The darker color is the more synergistic is that. And you can see now when these compounds are combined, we actually not only block cellular proliferation, we kill cancer stem cells. And this is really important. And what is the mechanism which is responsible for this mechanism? You can see here DNA damage as we see these comet tails. So following treatment, this combinational treatment, cell deaths undergo a lot of DNA damage, both in single and double strand level. As you can see, there is a lot of DNA damage, as you can see by phosphorylation of H2X, and there is also decrease of DNA repair. So taken together, we have this dual effect. We block DNA repair, but from the other side, we increase DNA damage. And this is why this synergy is so efficient. And you can see what in vivo, this combination is efficient as well. You can see some stainings from in vivo. We decrease ERK because we use, of course, trametinib. And you can see there is degradation of BRCA2. And this is why this combination is effective. So when we think about AI, unfortunately, just to design a perfect drug is not sufficient for cancer. Cancer cells know how to modify themselves constantly and develop resistance. As a basic cancer biologist, I think we need help in all of these factors. First, how we predict synergy between two drugs, how we manipulate cells to die because they know how to overcome apoptosis. Can we switch between different cell death pathway? How we predict this regimen, which is based on an AI and not just wet lab design. How we decrease concentration of these drugs because, once again, high concentration, cancer cells will develop chemoresistant. How we take into account all the previous regimen because cancer patients undergo a lot of treatment. How we predict side effects, especially right now, with many drugs, many novel target therapies are being used as maintenance for many years. So there is combination of subclinical effects which can be turned on after 2 or 3 years of use. And how we pay attention, better attention to cancer survival, balancing, we don't want to miss secondary cancer or metastasis, but from the other side, always checks up a lot of radiation. So we don't want to expose them to too much radiation. Thank you very much. Any questions?
Any questions from the audience?
Okay.
All right. Okay, so we are getting close. So our last speaker, but not the least, is Kevin Song, a PhD student at UAB in the School of Biomedical Engineering. Okay, it's all yours. Yeah, don't stand between us.
Hey everyone, good morning. I'll keep this short and sweet, but I wanted to delve into my work that I've been doing now that I'm into my second year in Dr. Jake Chen's lab. This is PharmAlchemy, a comprehensive knowledge base for mining drug-gene disease relationships. So here's the table of contents for my presentation today. So I want to present about the background and what is the problem that I want to address with my work. Well, there's a need for a comprehensive database that integrates the relationships between drugs, genes, diseases, and clinical trials. So previous databases like PubChem, DrugBank, they have not been well integrated for these different types of data. The reason why we care about this is obviously for drug repurposing and accelerated drug discovery analysis, patient selection, design of clinical trials for experimental therapeutics. We want to potentially accelerate precision medicine because not just getting drugs on a population level, but also matching treatments to individual genomic profiles, improve patient safety through drug side effects, looking at adverse effects, drug-to-drug interactions. And hopefully this application will help scientists and researchers and clinicians address scientific queries from the relationship between drugs, genes, and diseases. So this is a review of some of the current data sets. I talked about DrugBank, just GeneNet is another one. There's also CTD, PharmacoDB. And then you'll see that the data that my team and I have integrated into PharmAlchemy is more comprehensive, and you'll see the coverage the comprehensiveness and the coverage of PharmAlchemy in comparison to the non-integrated data sets. So the overall goal of the PharmAlchemy data model was to unify representations of entities and relationships between drugs, genes, and diseases. And the way that our lab thought about this in our team was by using this triangular relationship between gene, drug, and disease. And then gene to drug would be considered drug targets, drug to disease would be considered indications or side effects, and then gene to disease would be considered disease genes. And then you have permutation combinations where you have things connecting to themselves and connecting to sets of entities. We actually implemented this using Oracle SQL, and we were able to create an actual triangular relationship between drugs, genes, and diseases with various SQL tables, attributes, primary keys, foreign keys. So this was our implementation of the schema for creating that unified representation, which is one of the main innovations of the model. And you'll see that here are some of the results that we have. It kind of got cut off, but we have more than 350,000 clinical trials. The full data set from PubChem is about 54 million approved and experimental compounds, 46,000 genes, and 11,000 diseases. And we have about more than 1 million ligand target interactions in our database, as well as gene to gene interactions from string. And you'll see that this is a lot of it, like the approved and experimental compounds for the drugs. There are a lot of experimental compounds that no one's ever heard of before. So I'd like to do a quick demo. All right, so when we pull up this application, I can do a brief case study off the top of my head. I might want to search for fentanyl. A search for fentanyl. It's going to probably take 1.5 minutes to run this search. It's running against 9 million compounds right now. So let's say in my case study, I know there's a opioid, fentanyl crisis. I might be interested in looking at trying to get drugs that have an inhibitor effect on gene targets of fentanyl. So I might do a quick exploratory analysis and then look into, first off, why are people taking fentanyl in the first case? Let's look at some of the indications for why people would take fentanyl. Like, why do people have their hands on this in the first place? So let's wait for the search to finish. Okay, so here we have our query. We have pulled up 240 search results and we want to filter this. So we're going to go to filter and we're going to have drug name. Let's see. Let's select all 240 and let's try to get some indications. It looks like most of these are variations of fentanyl. So if we do a scan of this, it looks like these are mostly all variations, 240 of them. So if we click on indications, okay, I think I have to limit the scope to just fentanyl. Hang on. gonna have to research again. So the first question I want to ask is, why are people taking fentanyl? And then the second question I want to ask is, what are chemicals that can inhibit the gene targets of fentanyl, which are the opioid receptors, mu, gamma, and delta? So answering the first question, let's get those indications. Yeah, the gene and disease searches are a lot faster than the drug search. So I hope I don't take too much time with this. Okay, so let's just do a filter. Drug name, column, equals fentanyl. Save. Okay, that I'm just gonna run to the second. Oh, there it is. Okay, so we have their indications. So these are all the indications why people would be taking fentanyl. There's 39 of them. And we have the UMLS identifier, Unified Medical Language identifier, as well as the, let's see, fentanyl. Oh, sorry. So fentanyl has indications such as sleep apnea. It's used for sleep apnea. It's used for mental disorders. So these are opioid abuse. It is also used for opioid abuse. And so these are some of the indications why people would get their hands on fentanyl, like toothache, depression. So the second question I want to, just really quickly, I could finish in a minute. So if I search for OPR, which is the opioid receptor, I get my genes, and then get all my genes, filter this based off of gene name contains opioid, in case not insensitive, get my OPRDKM, and then get my G2R, get drug targets. These are sorted by the inhibition KI constant, KI inhibition constant, and then looks like we have about 16,000 compounds that are inhibitors or somehow interacting with the opioid receptors. So that answers my second question. And then I think I have 2 slides left. Maybe it's here. And then, yeah, so these are some of the questions that I hope to answer. What drug candidates with similar molecular fingerprints and gene targets, disease coverage, disease subtyping, and implementing features such as, in the near future, what are some drugs that could reverse gene expression? And we hope to use AI, we hope to use text mining, we hope to use APIs in looking at the FAIRS data set and other natural products. Thank you very much.
Running away. Are there any questions from them? All right, so there's one.
I have one quick question. Because sometimes when we build AI models, one thing affects the accuracy of the model is that the data we use may be not accurate or inconsistent. So when you use your database to search things, do you have any algorithm to find the inconsistency between 2 studies? Let's say if in one paper it said drug A active against target B, but in another paper it said it's not active against target B. So do you have any algorithm to handle the inconsistency?
I think this one. Okay, I think that one way of doing that is to cross-reference different data sets. And some data sets have more data and they have higher quality, and some data sets have less data, but they have more quality. And I think one way is to, well, you obviously can't trust all the literature for various reasons, but I think one reason is to use cross-referencing between data sets. Another thing is you can try text mining, but you have to, I think it's hard because the quality is very important. The quality of the data set is important, but the coverage might be diluted if you have a lot of data that doesn't have high quality. So that is a challenge, and AI is gonna analyze your data, but it's not gonna make you better data. So that's another problem. So I think trying to collect all the data and like trying to cross-reference and filter is one way to approach that.
All right, thank you, Kevin. Thank you. Yeah, so that concludes all the fresh talks. Maybe Dr. Chen has all the yeah, I was just saying that concludes the fresh talks. Maybe Dr. Chen is going to make a short announcement.
In the lab where data flows, revolutions underway, bridging
now we
gaps.
should dance, right? That's amazing morning sessions. The lunch box, I heard it's ready, right? So it's outside. If you have any dietary restrictions, and you can talk to us and we'll try to find alternative. Amy, you have some specific instruction? Yeah.
Hi, everyone. It's great sessions. Thanks for attending. Wow, it's really, crowd is multiplying, kind of like all of our innovations. So we have our poster sessions right now outside to the left and right of the auditorium. And I hope that you can stop by and take a look at the posters. People put a lot of hard work into it. A lot of you and your trainees and the people that you mentor have made these posters. If you signed up to be a judge, then thank you very much for that. I know some of you have said that you might have a conflict at noon, so that, but the posters should be up until at least 3:30. And I would ask the poster presenters to also hang around a little bit longer for accommodating people who can't be here during lunchtime. If you have not yet signed up to be a judge and you're interested in being a judge, then I encourage you to come, and that way you can have that extra motivation to go talk to the people and ask those insightful questions and learn more about their important research. So feel free to go around there, browse. Also, if you are a judge, I have the judging forms that you can fill out that has the scale on it, and make sure you get those to me maybe like in the next hour after lunch, so we have time to collate the results. Any questions about the posters or the poster judging? All right, if not, then, come up to me to get if you're interested, or if you signed up, come to me, get a form, and go out, enjoy some lunch, and the posters. Thanks, and we'll see you back here for the afternoon after lunch. Thanks. You said you weren't going to be here. I saw some email that you were like kind of.
In the lab where data flows, revolutions underway, bridging gaps, set to show ACE power in full display. Flagship sets, cornerstone, a future so bright, where machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Across lands, hand in hand, we craft new reality. Software standards unify, language shared and bold. Data's truth we amplify, story yet untold. Fair principles our guide, accessible and free. In this journey, side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein, an artery bridging minds. Future grows in this AI quest. With each step, we close gap between machines and human grace. Between machines and human grace. Bridge your white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. AI we trust, in AI we must. In unity we find future just. In the lab where data flows revolution. In the lab where data flows, revolutions underway, bridging gaps, set to show ace power in full display. Flagship sets, cornerstone, future so bright that machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Cross lands hand in hand, we craft new reality. Software standards unify, language shared and bold. Data's truth we amplify, story yet untold. Fair principles our guide. In this journey side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein and artery, bridging minds. Future grows in this AI quest. Workforce trained, ready, keen across AI divide. Biomedical scenes I've seen, world forever wide. With each step we close gap between machines and human grace. Between machines and human grace. Bridge your white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. In AI we trust, in AI we must. In unity we find future just. In the lab, where data flows, revolutions underway, bridging gaps, set to show ACE power in full display. Flagship sets, cornerstone, a future so bright, where machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Across lands, hand in hand, we craft new reality. Software standards unify, language shared and bold. Data's truth we amplify, story yet untold. Fair principles our guide, accessible and free. In this journey side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein, an artery bridging minds. Future grows in this AI quest. Workforce trained, ready, keen across AI divides. Biomedical scenes I've seen, world forever wide. With each step we close gap between machines and human grace. Between machines and human grace. Bridge white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. In AI we trust, in AI we must. In unity we find future just.
Really? They can hear outside? Really? Hello, everybody. The workshop is about start. You guys can bring your food in the audience, I guess. Let me go out. Yeah. No, probably. Yeah, let me go out.
Why not?
Yeah, we're gonna start the. Yeah. Hey guys. We should take a picture. Oh yeah. Yeah. Yeah, it's fine. I'll make it. Okay. Oh, you look great. Yeah. Yeah, I talked a lot. Okay, great. I think there's. Hey, the workshop started. So is that everybody? We can go back and we can come back later and yeah. Okay. Yeah, the workshop started. You guys can bring food if you're still eating. Yeah. Okay, thank you everybody for coming to the workshop session. So this session, we're very honored to invite Dr. Teresa Welbon and myself, Dr. Luron Pan, and Dr. Edward Acosta, and Dr. Jack Chen. In this section, it will be more interactive and more practical. So we will introduce some background about drug discovery and introduce some useful tools and demos so that students and researchers are welcome to ask questions. Let's introduce our first speaker, Dr. Teresa Welbon. She's a professor and interim chair of UAB Department of Pharmacology and Toxicity. And toxicology, and she has many years of experience teaching pharmacology and witnessed a lot of drug discovery projects. Thank you.
Okay. Good afternoon, everyone. It's really nice to be here with you. So I just have a very brief part of this. When I first talked to Jake and Laurent, they were saying a lot of the people are going to be informatics people. And so will you just give a very brief overview of kind of traditional drug discovery and development and kind of introduce some of the things that they're going to be talking about. So that's what I'll be doing for the next few minutes. So, you know, as some of the people have mentioned, ever since mankind's recorded being on Earth, they've been trying to find substances to help with their ills. And so most of the drugs that have been developed through the ages were found serendipitously, all the way from things like realizing that the resin from the poppy seed had morphine in it and was used to relieve pain. And then this picture of Alexander Fleming, who found penicillin by noticing that the staph on his culture cells were dying when the fungus invaded the culture. And so then in the mid-1900s, things got much more progressive with being able to do medicinal chemistry and organic chemistry. So they had been able to isolate a lot of the natural product compounds and start modifying them to come up with better treatments. And then as we moved into the omics era and had the genome sequence that was available, we started getting much more data on individual patients as well as diseases. And this has led us to this place where we have tremendous amounts of data that we're trying to link together to come up with the best drug for the best person. And that's where I think AI is so important for the place that we're finding ourselves now. So this is you've seen a few slides that are similar to this to go through the drug discovery and development process. But generally, when we think about drug discovery, it's the early stage of the process where the target is identified and then some sort of screening goes on to see, does it bind? Do these chemicals bind to the target? Do they have an effect on the target? And then once we get those hits from that initial screen, the hits can be modified through medicinal chemistry, as Alex was mentioning. It's very costly, but you want to get down to just a few leads to test. And then they do a lot of in silico modeling and trying to predict whether that molecule is going to be a successful drug. And after those early stages, then it goes into the preclinical stages, into animal models. But during the preclinical stages, we're still doing tests on things like solubility and pKa and this polar surface area of the drug. And then finally, when you get the data that you need to submit it for a new drug application or the IND, then it's shown to be ready to go into the human trials. And the phase 1 trials are generally with a small group of healthy people. And the main point at that is to just show that it's safe and work out the initial dosing in human beings. And then you get to phase 2, where this is with the group of people that have the disease, that you want to check not only the safety, but the efficacy of the drug. And this is where a lot of drugs fail, is we think everything predicts they're going to be efficacious, but they get to phase 2 and they don't show the efficacy that you're hoping for. in phase 2. But once it does show that, you can move on to the very large trials with the patients that have the disease and show further efficacy in the large number of people, monitor for a lot of the adverse effects and things. And then finally, when the NDA is able to be applied for, it's ready to go to the FDA to check out all of the data that's been gathered during the approval process. So with the typical drug discovery, the traditional way, it's generally taken 10 to 15 years. And the amount that I have, 1 to 2 billion, may be, from what Alex said, much higher than that right now, but it's very expensive. And so anything that we can do with computer aid or data mining will really help to improve this overall. Just a few things. Laurent is going to talk a little bit about target identification. So just a bit of history on this. The targets generally from the 1960s on, when we had these chemical libraries, was done by this method of screening large numbers of chemical compounds to see if it has the effect that we want. And then once we saw the effect, it went into animals and then into humans. But since 2000, with the sequencing of the human genome, and then the ability to do sequencing at a much cheaper cost and look for genome-wide association studies where we were able to detect targets that we could then use to try to develop drugs against, we came to this place where we were hoping that it would be one gene, one target, and then for the disease. But they found that this one target hasn't really worked as well as they wanted to. And so now we're looking more back at trying to find drugs that can target several things in a disease process, because we know that for many diseases, it's not just one thing that's an aberrant, but it's a multiple group of things that go wrong. And so now we're looking for polypharmacology drugs that can have several actions to be able to treat these complicated diseases. And so now, of course, we're hoping to get to the place where we can pick the gene that's maybe a problem with one patient or the disease and tailor the treatment for that person. And this was just an interesting figure that I found where it showed where a lot of AI-assisted drug discovery was taking place, particularly in the early stages, the target identification, validation, hit and lead identification, and some of the things. So I won't go through it because some of the people have talked about these, but we're really seeing a lot of benefit of AI helping, particularly at these early stages of drug discovery. So Dr. Zhavoronkov is going to talk about AI and machine learning and drug discovery. She's going to talk about target validation and modeling, virtual screening, structure determination, binding, molecular interactions, lead optimization tools that she uses. She's going to talk about ADME and toxicity screening, and then integrated systems for precision drug design. So after the target is selected that, or we're trying to base the drug on, we still find that at least 90% of the drugs that enter the clinical studies fail. And it's largely due, as I mentioned, to failure of having the clinical efficacy that we need. That's the largest percent of failures. But then a lot of the drugs show unpredictable toxicities. And then poor drug-like properties. Some drugs don't have those nice binding pockets where you can try to synthesize a drug to fit in. And then a smaller percent is due to poor strategic planning. So I'll just mention a few things about pharmacokinetics and dynamics to introduce Dr. Acosta's portion, because I know if you don't work in PK, you don't tend to think about the fact that we're always thinking about how does the drug affect the body? But we also have to think about how the body affects the drug. And so with pharmacokinetics, we know that most of our drugs are given orally. They have to be absorbed in the intestine. And to get from the intestine into the bloodstream, they have to pass through epithelial cells. And in those epithelial cells, there's metabolizing enzymes. These are present to help fend off target or things that may be toxic that we've ingested. So we have things like CYP enzymes. We also have transporters that are there to help protect us, but the drug gets into that epithelial cell and the transporter can transport it back out into the gut lumen and it can be eliminated. So these type of things can affect how much drug gets into the blood. And then once it gets into the blood, it's taken straight to the liver. And the liver also has many more enzymes that are present that can metabolize the drug, often inactivating it, but sometimes activating it, or sometimes converting it to a toxic species, as well as many more different pumps that can pump it out of the liver or into the bile, and can affect how much of the drug finally gets to the systemic circulation. So we do have drugs that target the GI tract locally. We do have some that affect the liver, but most of our drug targets are going to be reached by the systemic circulation. And so the bioavailability tells us how much of that drug got out to the systemic circulation and then can go to the site of action or maybe the site where it's toxic. And then the drug is eliminated. Kidney is the most common route. There's enzymes present there, there's transporters there, and then there's the other route that's primarily used for excretion is through the GI tract. And so we have to think about the ADME absorption, distribution, metabolism, and excretion in pharmacokinetics. And then just some of the things that are also looked at as far as the drug itself. During this process, you have to figure the molecular weight, the polar surface area. There's modeling that goes on. Is the drug soluble? It has to be able to be formulated into something that can be taken by the patient. Is it metabolically stable? Is it a substrate of transporters? Does it inhibit CYP enzymes? So if you have a drug that inhibits CYP enzymes, it can cause a lot of interactions with other drugs the patient's taking that are substrates of those enzymes. So a lot of times, the drug company doesn't want something that's going to inhibit CYP enzymes due to the risk of drug interactions. And then we look at the clearance of the drug, how quickly the body eliminates it. We look at dosing in rodents and also non-rodents, so usually 2 animal species. We identify the metabolites of the drug. As several have mentioned, there's many metabolizing enzymes. And so we want to know, what are those metabolites? Are any of them toxic? Are they active? And we look at things like PXR, whether they're substrates for a PXR, which is a xenosensor, it's a nuclear receptor. And when it gets activated, it turns on the expression of multiple different enzymes and transporters. And so that would be an indication there could be a lot of interactions with it. Does it produce metabolites that are conjugated to glutathione, which indicates they may be reactive and cause serious drug side effects? And then the PK/PD is done at several stages in the animals, and then in early first-in-human stages, we look for signs of toxicity. Then after it goes into phase 1 and phase 2, it then goes into the phase 3 with the larger number of people. We look at long-term pharmacokinetics. Are any side effects that we didn't see early on now being expressed? Then there's the new drug application process. And then as time goes on, are there new indications for this drug? Can it be used for other diseases? I think a lot of times now, whenever I teach about drugs, I have to really limit how many mechanisms I say a drug has, because all of them have more than one. And sometimes the mechanisms are equally active in a given disease process. Sometimes there are things that we don't want. But there might be an indication that's found that's completely separate from what the drug was initially discovered to accomplish. And then new formulations, as he mentioned, they were coming out with a formulation for their antifibrotic drug that was delivered by the pulmonary route. And so that's going to require that there be possibly a modification of the molecule so that it is able to be given through that mechanism. So there's a lot of things that are studied about the drug chemically, in addition to its effect on the target and the patient's effect on the drug. And so lastly, Dr. Chen is going to talk about AI's transformative role in drug discovery and how you can use AI for not just thinking about how the drug causes the response, but the patient may have other diseases that affects the response. Environmental things can affect the response. Other therapies or things they're taking for other diseases can affect their response, as well as their genetic profile. So in order to accomplish looking at all of these things to see what the final outcome is going to be, I think AI is really crucial to putting all this together. So that is all I wanted to give that little brief background, and I'll turn it over to Laurent.
Thank you, Teresa. Right, thank you, everybody. So in my session, I'm going to work through some of the commonly used AI technique and also publicly available and free for all researchers so that we will have a more direct understanding of how those tools work. There will be a few sessions. First, I'm going to talk about general drug discovery process and challenges and how AI tackle those challenges and how we use different tools for target modeling, lead generation, and lead optimization. As our keynote speaker said, nobody knows every step of drug discovery process. Actually, we have very good information source right here. It's a 4D map. It's a website developed by NIH. It's actually talked about exactly every step of the drug discovery. This is probably too small, but generally speaking, we have the basic research, translational research that's mostly done in academic translational medical center. And after that, we would normally start with develop the target or a very putative assay to generate leads. And in the traditional way, we will use HT assistant to generate those chemical leads. And then there are going to be a group of chemistry biologists work together to optimize the drug candidates through lead optimization process. And then they will bring to the trials. As you can see, I'm just not going to introduce the detail redundantly, but this is looking almost like engineering blueprint. And every step there are data generated, and data is sometimes publicly available. Sometimes we'll have a delayed sharing into the public, but both I think in modern era right now, most of the data in each step are available somehow, and we're trying to integrate the data and then automate the process in order to really review the entire industry process and be able to leverage the AI's power. And this is actually a multi-sector and multi-organization collaborative effort that involving both public, private sectors. So, but those resources are actually knowledge-wise open for everybody. So students and researchers can look into the details. And let's talk about the cost. So this is just very brief, like a selective process, like how many compounds will be synthesized and eventually approved. The odds are really low. That's why the billion-dollar process was actually based on most failure trials. And also like we said, like every compound being synthesized is about $5,000. And as you can see, that's how much money for that process. But if we can leverage AI, be able to actually reduce just the number of novel compound being synthesized by like 1,000 times, that would be basically save dramatic money and time and enhance the outcome. So AI is already a hot topic, and a lot of people probably access to AI like ChatGPT, but it's actually the general AI discipline has been well established in 1955 as the theoretic base. And this is probably one of the best textbook for AI. The first version is published 2010. And it's like a first year computer science AI major graduate students should have that. But basically what AI can do, they can do reasoning, knowledge representation, planning, learning, natural language processing, perception, and also manipulate object. Those are all textbook definition. But what AI can really help is it can do whatever human can do, with unlimited energy resource 24/7. And also it can think and with a cognitive function that human can think. So that might even be helping humans solving problems that beyond humans' capability. So we think this is just lay some background of how we integrate these two disciplines together and help solving our problem. And then we're trying to build here in UAB is basically the foundation for AI drug discovery infrastructure. There are three major components that has to be in place, and we luckily have all of them. First, the algorithm. Algorithm is basically a bunch of software and open source tools, and also, of course, people with the skill sets that be able to analyze complex data set with advanced mathematical modeling process. And those are all equipped in the Chiha cluster. And then the second component is big data. Big data is just a buzzword, but for us, like earlier, like Calvin has introduced as the pharmacome, which basically shows the disease target and drug relations. And those lay the foundations of any on top of the AI exploratory, no matter from the data cleaning, correlation, and data set foundation level, or to advanced modeling and predictive of any biological or clinical question you're trying to ask. And the third is the computing power. So in the past, like I remember when I was in graduate school about 15 years ago in chemistry department, we have to assemble a cluster to simulate just a peptide for a few nanoseconds within like a few months. But now we have equipped the GPU, most advanced GPU, and we can simulate a virus or even a cell membrane proteins within just like a few days for about 1 microsecond of movement, which is it's like probably 1,000 times more efficient and more powerful. And all those facilities are available in one institute, which is quite amazing. So that we think we're laying a good foundation. And I would have a little, sorry for the typo, just a very simple introduction about what's the algorithm out there. It seems very complex for people without any mathematical background, and most biologists and physicians might not have the training, but it's actually quite simple that in the discipline of machine learning, we classify all the problem into three. One is supervised learning, which is you have some answer like a ground truth that you know in the past, and then you train the data, and then you be able to answer the same question. For example, you can replicate the result of the same assay, and so you with the historical data of that same assay, and then you can predict the property of the chemical. And unsupervised learning is like you don't give them a label, you just tell them, I have this bunch of patients, I don't know what the diagnosis is, but they probably belongs to 3 diagnostic like groups of the same symptom, and then you just assign, and they were just based on the features they're trying to automatically grab, and then just unsupervised, we call it unsupervised, and they just find the pattern by themselves. The same as the chat GPT, the underlying LLM is also unsupervised learning model. We don't tell them any directed objective, just let them figure themselves what's the internal logic between among a big chunk of data. And the third one is probably the most useful and practical is in reinforcement learning, is we based on the base model, and we try to give some feedback to the model to either make it more accurate towards certain problem, or make it answer just the very subset of particular question we're trying to ask. So based on that, and all those infrastructure and algorithms are actually in the AI world is completely open to public and for free. So if you wanted to learn anything, or we're trying to find some existing architectures, is all freely available in the AI community that students have access to. And then we normally just represent our data in based on its native form. Like for example, if it's a picture, a cellular image, or or it's like a sequence data, like electronic healthcare records, or the small strand of a chemical structure, or the DNA sequence or protein sequence. It can be represented by some of the algorithms such as RNA or transformer. Those are all very commonly used architect. And then we have the generative adversarial network is normally to generate new entities that are of interest and with some kind of constraints you put for certain purpose. So hope it's not too dry. And this is all the publicly available computer resource that offered by NSF, and also this is the UAB Chiha resource that students can also apply for count. So then I'm going to briefly go through some slides of the case study use and softwares, and I will give some of the demos. So the first step is normally we're trying to identify targets and validate them. So as we talk about therapeutic targets, we normally talk about a particular gene or its downstream expressed protein. And then when we analyze data from clinical genomics, and there's normally multiple recommended association or correlation with target, and there's also biomarkers that shows correlated to certain disease. But how can we select a putative therapeutic target? So the first tool we're going to use is some kind of ranking algorithms. So, and for example, we have like this is called the winner, and also grape is some representative softwares that Dr. Chen's lab has developed. It's very easy to use, that it gives direct ranking, and as you can see using those name maps, that you can see how important the hierarchy or connectivity of certain gene to the other. And then this is just a way to help us to prioritize targets for the next step of research after a raw data analyzed from patients. And then the second step normally is we have to validate the target either in real, normally it's through like mouse model or other, but another thing is we try to model it and then understand its function. And understand how it interacts with molecules. So in that way, it's more of a domestic, like precise level of understanding rather than just a micro scale of observation from an in vivo outcome. So there's several very important breakthrough. First is AlphaFold has been released in 2021. So what it does, it takes a protein sequence and with a we call it a 3D transformer type of algorithm, and they be able to predict a 3D structure of any given target. Even though right now it's not like a perfectly work for all the targets, but it's a good starting point. Since in the past, a crystallography lab or structural biology lab would take like $1 million to solve certain difficult proteins. So this is a good resource, and it's already implanted in the public domain that people can just input their sequence and get the result. And the second one is once you have the structure, you will observe where the therapeutic strategy could be. Where is this, where the drug could bind to either inhibit or activate the cell, the function of this target. And these are some of the very complicated structures, for example, a membrane protein or a transcriptome factor, that you can see that we can put drugs in different places precisely and to interfere. And then, so normally we have two types of binding. One is orthosteric and also allosteric. So that's how we can use molecules to interfere with the function of the native target. And this is like one simulation I've done 10 years ago, actually, with a substrate and without the substrate, you can see how the allosteric of this membrane proteins, certain like domains has been shifted by allosteric effect. So those things nowadays can do like 100 times faster and more automatic. And recently, there is also a big breakthrough on the protein or other molecule interaction prediction problem. In the past, we have to use a bunch of computers to simulate the docking process between different biomolecules. But now we just directly enter just the string format of your DNA, protein, and also small molecule. And then it will instantly predict how they are come together as a complex form. And this work is published by David Baker's lab in University of Washington. And this also a publicly open source software everybody has access to. And so the second step, after we figure out the target, we figure out how drug is being selectively bonds to certain target with a potent properties. Normally, we will have to optimize that we call lead, that with multiple dimensions of drug-like property. But in that case, we cannot really we have to validate through more phenotype or other type of assay result. So normally we use ligand-based deep learning model, so that we only takes the information of the drug and then most of the time use the supervised learning model to approximate those data to whatever assay, biochemical, cell biology, even in vivo result, and to be able to correlate certain domain of a molecule, how it correlates to those function, and be able to directly predict the result. For example, this is the first generation of ligand-based deep model that they have explicitly describe different type of 1D or 2D or even 3D matrix for any given molecules. But now they natively integrated in the ChatGPT domain, that you can directly ask for example, what's the lipophilicity of this small strain? And it directly tell you a number with a regression model. So this is how this technology evolved. And you can see like every 2-3 months, there's a new code or paper published, and to get a more and more accurate result in this type of problem. And this is one like open source, also a web-based tool that which has this comprehensive multi-parameter optimization interface that developed by Swiss Bioinformatics Institute. It's already being evolved for so many generations, and they basically covers almost all the ADME toxicity prediction domain that we are interested in the drug discovery project. But I'm not sure that how accurate they are. They're constantly improving their model, but basically that's a good starting point. If you have a novel compound, you can have a quick check. And so I will basically do a simple demo on how some of those things are used. So for example, if we try to build a like a 3D model for any protein you discovered that has no structure, either you can go to, for example, the unipro.org. They already have this 3D like information that AlphaFold has been generated that has innately in it. But sometimes as you can see that they have very low confidence. Those red spot could be completely modeled wrong, and those very high confidence region could be correct. So those are the that's how you can examine. And then if some of the low confidence region that we can't trust AlphaFold, we can use other tools. For example, that the Swiss model, they can search a more closed template that can model some subregions more accurately. Okay, and then another one is this one, the Swiss admin. So we try to actually build a more advanced version, but basically you can put a list of small strain in it, and then they will have different pharmacology profile. That layout has this in silico prediction from pharmacochemical property, lipophilicity, and other water solubility, pharmacokinetics, and drug likeness. And you can see, for example, we talk about the metabolism a lot in the drug discovery process. That, for example, this model, it predicts it has no CYP1A2 inhibition, and then they also document how many molecules they trained for this AI model and what's the accuracy for that AI model. So you have some idea of how confident this result is. So this might take a while, but it's just a quick demo. And in the last, this is another commercial company. I just think this is a very cool video so that everybody could see. This is one of the first AI drug discovery company, Accenture in UK. This is the demonstration of how we optimize multiple properties at the same time and with on-target efficacy and also other pharmacology properties within the AI system. Sorry. Okay, just a simple demo. Okay, thank you. Yeah, so that's gonna be all my presentation. Welcome to take any questions. Yeah, sure.
Do you have any experience using the ATTIS simulation? I mean, it is very fast, but I don't know like how realistically useful it is. Do you have any opinion?
Yeah, okay. So ATTIS is interesting. It's a super cluster just dedicated for MD simulation for biomolecules. And I started, I think they have ATTIS 1, 2, 3. The major invention for that cluster was they trying to make it more energy efficient. So that basically they have 1,000 times more efficient than general GPU cluster, so that it can, you know, like since how we measure the computational efficiency is on how many the power consumption for the certain hardware, and that's how their achievement is. They optimize just for MD simulation. I actually tried once. They donated that one to NIH. It was actually invented by a hedge fund. Like super rich person, D. Shang Research, and he invented this and then he donated to NSF, so everybody have access. I think it is for large molecule is actually quite efficient. Yeah. Thank you. Okay.
So in many others, like phenotypic-based approach, we often identify some interesting molecule, but we don't know the mechanism. Do you have any tool to able to infer the putative targets for those compounds?
There's actually plenty of tools that you can use for do target mapping. Because this is an interesting, it's like for any molecule, you can search on, for example, Kimball or some commercial database like Rexis on their historical test on direct enzyme binding or enzymatic active IC50 value. Those all can be searchable. So normally, if anything that has at least 1 micromolar of efficacy for those molecules, and then you would consider it to be on that target, right? So normally if a molecule exists, and then we know for sure that's the case. But if it does not exist, there are 2 ways to do it. One is you can do a similarity check on all the known molecule that has a high chemical similarity for the ones you're looking for and see what their target is. And that's one way. Another way is all those target, they actually can build its individual binding model so that you can actually mapping it. And also the docking tool I'm talking about is also useful because right now we already can do whole genome docking using the AI system. So any novel target, you can directly do like a whole genome docking over we call the back docking back to the proteins. So you can just lay out all the proteins you're trying to screen off target or want to fish in it. We can just do it in silico as well.
Yeah, I'm assuming you're mentioning about the AlphaFold, right? They can predict the 3D structure so you can run the docking. Are you aware of any tool can do this?
I'm sorry.
Any other tool to screen the whole proteins, right? Using maybe using the space and structure from AlphaFold.
Yeah, they don't have a directly tool, but normally our computational chemists we will set up a streamline to just do it in-house. But everybody has been doing that. So yeah, if you need help that we can collaborate. Yeah, we just basically integrate different parts of different algorithm and a module piece and to achieve a particular goal. But that has to be customized scripts in-house. Yeah, but I don't think there's just available tool right now because those normally very computationally intensive. So that public open source tool, they probably won't be able to run. Yeah, it's very expensive. Yeah. Any question? All right. Then we welcome the next speaker, Dr. Edward Acosta. He is the professor of Department of Pharmacology and Toxicology and also the director of the PK/PD core of UAB.
Thank you very much. Thanks. I appreciate the invitation to be here this afternoon and appreciate everyone coming out to hear these presentations on a relatively new, but yet important topics. I was asked to talk today about clinical pharmacology, in particular pharmacokinetics and pharmacodynamics in phase 1 and phase 2 studies. The phase 1 and phase 2 clinical trials along the process that Teresa had touched on is pharmacokinetics and it's why they do it, especially phase 1. And so later on at the end, I'll touch on a study that was done in the field that I work in that was, I think, really perfectly done phase 2 type PK/PD study. So just very quickly for an outline, I'll briefly review the development process again and touch on what Teresa had mentioned. We'll go through some of the pharmacokinetics, what it is, ADME, absorption, distribution, metabolism, and excretion, and then touch on the phase 1 and phase 2 pharmacokinetic and pharmacodynamic studies, including SAD and MAD, which is single ascending dose and multiple ascending dose pharmacokinetic studies. I'll talk about the example that I mentioned, and then at the end, just a very brief overview of some of the modeling and simulation type work that we normally would do and how that might integrate, although I don't know how to do it yet, integrate with AI technology. I think down the road, that's going to be at least an area initially where it's just absolutely, it's in its infancy right now, but merging artificial intelligence with the ability to model and simulate concentration time data from drug studies, whether it's in animals or what I do is all clinical in humans, that's going to be a very key aspect of the future. In terms of the drug development process, we haven't spent much time on the early stages here, the in vitro studies, animal testing, et cetera. I'm not going to touch on that, but as Laurent had mentioned, there's a very large diagram, complicated of various tests that need to go into the early, not just the development of the drug, but the early testing, both in vitro and animal. Once the investigational new drug is filed and it's safe to proceed, this is when phase 1 begins. Now, these are all kind of in stepwise fashion, but you have to keep in mind that during this whole process, all of this is overlapping. So animal testing will still be going on, probably more for toxicity purposes, even during phase 1 and phase 2, and maybe even phase 3 clinical studies. But once the drug gets the approval to go ahead for use in humans for phase 1, that's really what pharmacokinetics is. It is to assess the kinetics of the drug in the human population. Now, these are going to be almost always healthy volunteers in these phase 1 studies. During phase 1, start to narrow down the dosing a little bit, and then move on to phase 2, which is now where you look at the drug in the patient population that it's going to be intended for. I do a lot of pediatrics, but also antivirals, antiretroviral drugs, HIV infection, as well as other viral diseases. And so in phase 2, we will now start to narrow down what these doses are that might be useful prior to going into phase 3, because phase 3 is now the giant clinical trials, hundreds, if not thousands, of patients to be enrolled. And the last thing they want to do is get the dose wrong. Just a very brief aside on that, there was an example of a HIV drug. They did not do a lot of the proper early, this was many years ago, a lot of the proper early work that needed to be done, in particular protein binding. They didn't check how protein bound is this drug to albumin, alpha-1 acetylglycoprotein. They moved on, they got through phase 2, moved right into phase 3. Couldn't figure out, the drug did nothing, absolutely nothing for any of the patients. It turns out it was 100% protein bound. So it never worked, never had a chance, but they never looked at it appropriately. So that type of information obviously is critical moving forward. But phase 3, typically phase 3 is going to be, these days, it's usually multiple phase 3 studies. It's not just one. But once the data is collected, long-term safety, additional pharmacokinetic work will be done during phase 2. Typically, that's going to be what we call the population pharmacokinetic analyses. So fewer samples collected on a larger number of individuals for modeling purposes. Then an NDA, the new drug application, is submitted to the FDA, and hopefully they get their approval. And then it's not over at that stage. And phase 4, post-marketing surveillance, starts. So they have to the companies have to keep collecting data on their drug for years and years and years and years to get further information on safety and efficacy. So PK and PD, in its simplest terms, pharmacokinetics, it's the time course of the drug in humans. It's what the body does to the drug. Pharmacodynamics is the relationship between the dose, or I prefer the concentration of the drug in the body, and the measured effect. Or in other words, what does the drug do to the body? And this could be in terms of efficacy. It could be toxicity. So pharmacodynamics could be either of those, depending on what's being important to look at. So why are the pharmacokinetics important? Every single drug that is out there has a therapeutic window. We might not know what it is, but they all have a therapeutic window. So at some point, the concentrations get too high, and there's some toxicity that will be observed. At some point, the dose or the concentrations are too low, and there will be no effect, little to no effect. Somewhere in between is what we call a therapeutic window. And this is the target that we're trying to find in early phase 1 and phase 2 drug development. What is that therapeutic window? What is that index? Some drugs, warfarin, for example, have a very, very tight, tiny therapeutic index. And as a result, the concentrations of that drug need to be monitored, therapeutic drug monitoring, to make sure for every patient they stay in the right window or the right range of concentrations. Most drugs these days that we deal with, if there is a tight therapeutic window, it's dead. They won't even move forward with it. They'll find another drug. They'll grab a backup and off the shelf. They don't want to have to deal with having to do monitoring drug concentrations. And so they'll go back and go back and go back until they find the drug that's effective, has minimal toxicities, and has a wide therapeutic index. Some of the drugs included, it would be like third generation cephalosporins. I mean, you can give grams of these things and there's no toxicity, but they're effective. So this is why the pharmacokinetics is important. In phase 1 and phase 2 are to figure out what this therapeutic window or the index is, as well as the early efficacy. So ADME, or the absorption, distribution, metabolism, and excretion, is traditionally how we've always thought of looking at the pharmacokinetics or of the drug and moving a drug through the pipeline. Both, again, absorption, we typically will think of that as oral absorption. It could be other forms of absorption, topical, IM, etc. Distribution, where does the drug go once it gets into the body? And then the elimination is comprised of both drug metabolism as well as excretion, both of which Teresa had briefly mentioned. So this is a representative concentration time curve, or oftentimes we'll call it a PK profile, where simply on the x-axis is the time post-dose, the y-axis is the drug concentration. In this case, it's in the plasma. It could be tissues, it could be other matrices, could be cells. But this is actual human data for this particular drug. So here at time 0 is when the dose is administered. What we have to keep in mind is, so this is actually a real patient data set. So this was a real person, came into a clinic somewhere in the world, I don't know where, and sat down for 24 hours and had their blood drawn. So each of these triangles here is a sample that was collected. Not to mention this was also a pediatric patient. So not easy for the family. So when we're talking about the data that are generated, especially from clinical trials, we always have to keep in mind these are real people, real families behind the data that's being generated and coming to our lab to look at. This curve, as the drug is absorbed, the concentrations go up. This is obviously where we'll get the absorption phase. At some point, you reach a peak concentration, the Cmax, at a certain time, which would be the time to maximum concentration. And then the drug starts being eliminated from the body. And this terminal phase down here is where most of you are probably more familiar with the half-life of the drug. This is where the half-life is calculated from the slope of this terminal elimination line. And then for some of the drugs, especially the ones I work with, these concentrations at the very end of the dosing interval, we call them a trough concentration because it's at the trough right before the next dose is given, are also very critical. If those concentrations go too low in certain disease states, resistance to drugs can develop, as well as other lack of efficacy. And then underneath this entire curve is the AUC. In this case, these samples were collected for 24 hours. This is an area under the curve from 0 to 24 hours. And so from these parameters, we can derive other parameters, clearances, volumes, et cetera, that we then can use to not only identify what these curves need to look like or should look like to best fit the data. But in my perspective, it's also then to be able to go back and simulate. Once you have the model in hand, and you know what these parameters are, and you know what the variability around these parameters are, you can go back into the model, fix the parameters and the variability, and simulate any dose you want at any dosing interval you want. You can give this every 4 hours if you want and see what it looks like. And that's one of the powers of the modeling and the simulation. So absorption, very quickly, is simply the movement of the drug across the biological barriers from the site of administration to the blood. There are many routes of administration for absorption, again, oral being the most common. Obviously, there's parenteral, IV, sub-Q, and intramuscular. Other routes, inhalation, rectotopical, transdermal. We typically will think of absorption as how quickly the drug might get into the system, but we think of it more in terms of bioavailability, which is simply the fraction of the dose that you ingest, for example, that reaches then the systemic circulation or the circulating blood. And so bioavailability does not dictate the rate of drug absorption, but rather how much gets in from a given dose. The distribution volume, it's a theoretical volume of fluid into which the total drug administered would have to be diluted to produce the resulting plasma concentration. So volume relates to the amount of drug in the body to the measured concentration in the plasma. So when we measure a concentration, we draw a blood sample from a person, we have to measure how much drug in there. It's typically going to be in units such as nanograms per mil or milligrams per liter. There's always a volume component. And so in order to calculate all these PK parameters, we have to know what the volume is. And that's what the distribution volume reflects. Some drugs have gigantic distribution volumes, 300 plus liters. Obviously, that's a huge volume. All that means is that the drug is distributing everywhere in the body. It doesn't mean that that person has 300 liters worth of material in their body. And so low distribution volumes typically are going to be a 3 to 5 liter range. And mostly that type of a drug would stay in the plasma. And then there's medium, which is slightly higher. And then the high distribution volumes will distribute more into tissues, brain, bone, other matrices in the body. And then briefly, Teresa had mentioned this as well on elimination of the drug, both metabolism and excretion. It's the irreversible loss of drug from the site of measurement. And so metabolism, the goal of metabolism in the body is to produce a more polar compound. Once it's more polar, it can be more readily excreted, typically via the kidneys. But the metabolism can occur in a lot of different areas. Typically, we think of the liver as the primary source in your body that would metabolize drugs, but the gut is also a very important component to drug metabolism, as there's quite a few metabolizing enzymes in the gut. And then excretion, again, just typically is going to be eliminated via the kidneys. But there's other pathways too, kidneys, lungs, saliva, sweat. There's various pathways that drugs can be eliminated or excreted. So application to drug development. So the phase 1 clinical studies, again, the goal is to conduct a single and multiple ascending doses using an adequate dosing range. This is where many companies, I'd have to say, fail. They do not look at a wide enough range of doses to get a good understanding of what the pharmacokinetics are. Let me give you a good example. So you're looking at a drug, 100 milligrams, once a day, oral drug. What happens when you double the dose and you go to 200 milligrams once a day? Well, if you go back to that curve I had, the area under the curve, that should double, right? You're doubling the dose, so the area under the curve should also double. If you do not look at a wide enough range of concentrations, you might miss the fact that you could be doubling the dose, but quadrupling the AUC. And that's the nonlinearities associated with some of the pharmacokinetics, and why these early phase studies are important is to understand what those concentration time curves actually look like. So the samples need to be collected over an appropriate time frame, and then also to look at, especially in phase 2, concentration or dose response relationships. This is an example of a real drug, phase 1 single ascending dose trial in healthy volunteers. So in this case, they did an extremely good job looking at doses. This is single ascending doses, so 10 milligrams all the way up to 1600 milligrams, and very linear. It's kind of hard to see on this graph, but it's a very linear increase in the drug exposure as the doses increased. So in other words, the AUC from the 800 milligram dose was double the AUC of the 400 milligram dose. And this insert is just the same plot on a log scale. But that would be an example of a single ascending dose study. But it's not just the single ascending dose part. There's other things that need to be looked at. In this case, they weren't even sure what the formulation of the drug was going to be. And so they had 4, actually, I think they had more, but they looked at 4 different formulations of the drug. So these curves are all the exact same dose of the same drug with different formulations. Obviously, very different looking concentration time curves. So that needs to be looked at as well. I had a slide in here. I took it out just for time, but also food. What happens with food? Very, very common. Take a drug with food. There was an HIV drug from many years back. When the drug was given with a high fat meal, and I'm talking a Big Mac and a half type high fat meal, the AUC will increase 5 times. but yet they got it approved. This drug can be taken with or without food, doesn't matter. Yeah, right. So we figured that out pretty quick. But so the food can have a dramatic, it can also decrease the absorption if you take a drug with food. It can also blunt how much drug actually gets in the system. So then they move on. This is still, it would still be phase 1, maybe early phase 2 to multiple ascending doses. So now they've narrowed down the doses. Instead of this 10 to 1600, they're now down to 100 to 600, kind of finalizing, the range of what they think the dosing is going to be, in this case, based on susceptibility information. And so now they're looking at multiple ascending doses. What do we look like at steady state? So in this case, I think it was dosed for at least 7 days or 10 days. So a huge commitment by the volunteers, patients that do these types of studies. Ultimately, they ended up with this 400 milligram dose. But both of these need to be looked at. And all this information collected from the phase 1 into this, the single and multiple ascending doses and into phase 2, over time, all that information gets start putting into models to really identify the best model to capture what these concentration time profiles look like. All right, so if concentration response data from phase 2 have been generated, now they'd be moving into phase 3. At this point, moving into phase 3 is really oftentimes, unfortunately, just a guess. And sometimes they'll enter phase 3 trials with more than one, sometimes 2 or 3 different doses, large groups of patients over 6 months to a year to figure out which dose works and which ones might not work as well. All of that can be negated by simply doing the appropriate concentration response or pharmacodynamic assessments early on in phase 2. And this is probably where a lot of, I think, down the road, some of the AI-assisted modeling work will be very, very useful to save a lot of time and cut back on the number of studies, the number of people enrolled. We're already doing that in pediatrics. So you imagine you've got a drug that's now approved in adults, but that drug needs to be used in the pediatric population. We can't enroll thousands of babies into clinical trials. So we have to rely on modeling and simulation based on adult data and translating that down to what a fetus, or not fetus, a neonate or a 1 or 2 or 3-year-old might look like, how they might handle that drug. And this is where I think a lot of the integration could really take place and help and save a lot of time, effort, and money. Anyway, so phase 3 will be started, and it'll determine the long-term safety, greater than usually typically 2 years, efficacy and safety. And this is also now at the end of phase 2, beginning of phase 3, when they also start to think about implementing a pediatric development program if that drug is to be used in pediatrics. So this is the example of a phase 2 proof of concept study design. This is a drug called dolutegravir. It's an antiretroviral used for treatment of HIV. It's probably, I don't want to overstate it, but I'd have to say at this point, the best antiretroviral drug that we have out there, that we've had out in a very long time. This was a very simple, small, very well conducted study. I'm not going to go through everything here because I'd rather just get to the results, but there are simply three groups of 10 patients. They received placebo, 2 milligrams, 10 milligrams, or 50 milligrams. That was it. And they received it for 10 days. And they were in the clinic for 10 days. So huge commitment. And everybody was blinded. I don't need to worry about the inclusion criteria. But the primary endpoint over this 10 days, just 10 days treatment period, was the change in the viral load. So in antiretroviral therapy, as well as others, we can look at how much virus is in the blood. And when you start drug therapy, does the amount of virus drop? Hopefully it will if you're dealing with a good drug. And that's what this is. So here's the top line is the placebo patients whose viral load did not drop. The green part here is the 10-day treatment period. And then here's the 3 different doses. The blue and the gold and the green are the 2, 10, and 50 milligrams. So nice differential response. across these different doses. So why does this matter? Matters because of this. So on the left here is just the pharmacokinetics, dolutegravir, concentration time profile. Here's, didn't show up too well here. Oh, well, but this is from 0 to 24 hours. And here's what the curves look like for each of those 3 doses. Over here, go back up one. No. The trough concentration, the concentration at 24 hours after the dose for this drug, a lot of these drugs, is critical for efficacy. So simply plotting that trough concentration versus the change in the viral load over those 10 days, we're able to get a very nice model, what we call a maximum effect model, which then identifies, in this case, it's 300 nanograms per mil. What is that trough target? What does it need to be in this patient population? So now we can move forward saying whatever dosing regimen, dosing interval we're going to use, we have to maintain that 24-hour concentration above this value. In this case, it's 300 nanograms per mil. That's why this information is critical, especially moving. So what if now they had to change the formulation for some reason? And now you get a completely different looking pharmacokinetic profile. Well, it's still the same drug. So 300 is still the target for the drug. But now they know that. They can simply look at the kinetics in another phase 1 type trial and understand what dose is needed to achieve that concentration. So lastly, I just wanted to touch on some of the slightly more, well, probably not to you guys, but to us, slightly more complicated aspects of pharmacokinetics and pharmacodynamics. This is Tamiflu. Most of you are probably familiar with Tamiflu, treatment of influenza. Many of you have probably taken it. Tamiflu is actually well, its name is oseltamivir, but it's actually a prodrug. So what you eat, what you ingest, isn't the active component that inhibits the virus, the influenza virus. It's oseltamivir carboxylate, its metabolite. So I'm not going to get into all the background behind this study, but this was a large critical study that we had done here at UAB with the Collaborative Antiviral Study Group, and looking at the pharmacokinetics and dynamics of Tamiflu in pediatric patients, trying to figure out what the dose should be, because there was essentially no information in children less than 2 years of age. And this was back in the 2009 H1N1 outbreak. So people were quite concerned about what the dose would be for that drug. So we ended up having a 2-compartment model for the parent. So drug is administered here in the gut, goes to the plasma, essentially the central compartment, and then distributes to other places in the body. We don't really know what those are, but there's a distribution of the drug. The drug is then also converted in the gut and hepatically to the active metabolite, the carboxylate. This we then described with a 1-compartment model. Now, I'm not going to get into all the information here today, but we also linked this then to a viral dynamic model where we linked the exposure of the carboxylate to the change in the amount of virus in the patients. And so this is, I was talking to Laurent earlier, but so this was a giant set of differential equations to describe the movement of the drug and the virus through all these different compartments. And at some point, I could imagine these differential equations just won't even be needed anymore. And this type of modeling can be done a lot more smartly and quickly, because it's a tremendous amount of work at this point. And so this then is what a population pharmacokinetic model result would typically look like. So this is the parent drug, oseltamivir phosphate, over here on the left, and here's the active drug, the carboxylate, on the right. You can see the variability. This is a very well done, well-controlled study. And there's considerable amount of variability that we have to deal with, but we got really good curve fits through both of these. And so this information, along with the viral data, viral dynamic data that we had modeled, was then used for simulations to predict what is the exposure of this drug that is needed in this population for influenza. And And so that's one of the reasons why the PK and PD and the modeling aspect of all this is so critical, is not just to develop the model and describe the kinetics, but go back and linking it to response, to efficacy, and conducting simulations to figure out what is the best dose that we should be using. Not just the dose, but the dosing interval as well. So then just to wrap up, so applying basic PK principles, it's critical during the drug development process, especially, I mean, that's really what phase 1 and 2, especially phase 1, are all about, is understanding pharmacokinetics in humans. But proper collection of the data in the animal studies prior to phase 1 provides very useful insights into dosing in the humans. And establishing the concentration response relationships is absolutely critical for proper dose selection and bringing a drug into the pediatric population. So modeling and simulation tools can be used for better initial dose and estimation and increased efficiency. We use this is used in adults, but we use this modeling and simulation approach every single study I'm involved in to bring a drug that's already approved in adults into the pediatric population. We've seen a drug get approved in pediatrics with an N of 6 babies, and that was it. But there was a ton of modeling support and simulations that went in to support that. And so this is an area that's absolutely, it's in its infancy. It's ripe for integration with AI technology and the modeling simulation approaches. And so hopefully with systems pharmacology and AI group here, we can start to move forward and make some good progress on this. So I'll stop there, and thank you very much for your attention. Are we taking questions? Too much. Okay. All right, thank you.
All right, the last, Dr. Chen will give us some perspective of the future.
All right. I have 12 minutes to talk about the future, right? We live in the current, so the future doesn't matter that much. No, I'm kidding. So I think you've got a really good understanding based on the previous 3 talks, and I think you should conclude that yes, indeed, $3 billion, 1 approved drug, 10% success rate, it's probably even optimistic. Right? And we are living in an area, just like Alex says, if you don't have a lot of funding, don't think about the drug discovery. Or is it really true? Right? So that's what I'm essentially trying to tell you. When you look at the AI-enabled drug discovery today, you get a hint that if you isolate the problem for every one of them, like finding target, you probably can find the use network and rank the target, not a really big problem. If you want to find the hit compound that doesn't have all the property, you probably can do it too. Optimize compound based on the chemical property, you can isolate that problem. Preclinical trials, animal model, we can cure all the cancer in mice. It doesn't matter. In clinical trial, that's where the trouble begins. In fact, I would argue that in the pre-AI era, we can do pretty much everything quite well. It's just that we cannot deliver on the end result. Why is that? You have to ask the question, then can AI really make that difference? And I think there are lots of papers published, but I want to show you quickly some statistics. Alex already alluded to this in the morning. He said, well, how come the drug development cost is not going down, even with all the genomic technology and AI? And there's a review paper that published that says that there's 3 times increase of the development cost in the past 20 years, not decrease. In fact, earlier this week, I was at the Stanford Drug Discovery Symposium. I was asking all the big pharma's chief research officer this question about improving success rate accelerating drug development cost and maybe the lower the time required for drug discovery. Is that an impossible triangle? So one of the person thought for a while and then said, no, it's possible to do it all. But the data certainly doesn't show that yet. So most of the time we're actually spent on the clinical development. And in the keynote this morning, there's very little mention about the clinical. I think this is where the AI can really play some roles. Earlier, you saw my student, Kevin, talk about the gene-drug disease relationship in the context of human clinical trials. And we do believe this is a very important paradigm. You have to actually think about all of them together. Another paradigm triangle is what I think is cell lines, animal models, and human models triangle. Right now, we're doing it in isolation. There are different pros and cons of each model. If you can do it in cell lines, you can do it really, really fast. But nothing is as fast as doing it in computers. I can do 1 billion calculation or billion models per second. And so of course, it's faster than doing in animal models, but NIH is already saying that they're going to reduce the animal model use over maybe 10 years and they will phase out. And then you have to actually choose between cell lines or human. And I'd rather choose computer models. And but Alex was very correctly in pointing out that in order for our academia to compete and make an impact, we have to actually put the puzzles of different pieces together: biology, chemistry, and medicine. In fact, here I'm seeing medicine, chemistry, and biology. And if we actually connect all of us, we can hold quite significant piece. So I think the disruption seems to be coming not only from AI, but how we organize, how we conceptualize these things together. So the opportunity is there, right? For example, there's a paper about immune checkpoint biomarkers. If you start with the checkpoint target genes, to use it as a biomarker to do prediction of sensitivity of a treatment outcome, you get some result, like PD-1 here, PD-L1 here. But if you're starting to incorporate information from the network, and these are protein interaction networks, these are proteins that are affected but may not be mutated or perturbed, and actually the performance become better. So here is that the use of network at the molecular level certainly would help. What else could help, right? So we're involved in a project called the Bridge to AI. One of the projects that we're doing is called Cell Models for AI. So essentially, the team of scientists from UC San Diego, UCSF, Stanford, and all of us are working together is to deep molecular phenotyping of these cells, iPSC and cancer cell line. So we're asking the question, can we not only collect the protein component that have changed, but also how often they were observed together? And maybe in the protein interaction, you observe protein interaction network. In immunostaining, you see that they show up in different co-occurrent regions of the cell. And then you put that together into protein-protein interactions, and then you also map it into a knowledge representation. This is called a music tool. That's actually available today. If you can visualize it correctly, then when you have genetic perturbations like chemotherapy or not, and then you can observe this, right? This level of deep phenotyping at the subcellular structures are really collecting data at a level never been seen before. And we think that this is probably one of the solutions, right? So earlier, my former postdoc, Rahul Sharma, talked about using drug chemical information combined with disease gene expression to build models that can predict digital twin IC50. He has a poster, so I won't talk about it. Alex also talked about using generative AI. And these generative AI can actually start with some chemical that work, but may not have perfect binding affinity. And then he would actually just use that to train GAN models, and nowadays probably diffusion models, and train these GAN models so that it can generate features that can be reconstructed into chemicals that will have much better binding affinity. So these are actually the opportunities of future drug discovery. But what about the biology side? I think many people have ignored the biology side. That's why I think we heard a comment earlier today that the cancer is not about the chemical. It's actually all about the relapse and the outcome where sometimes we cannot make much of an outcome. When you think about this, the salutogenesis, essentially the disease recovery, is not solely determined by the chemical. Let's not forget that our body is having this immune system, and we're constantly just having internal tumor microenvironment that is also helping to fight the tumor. And the tumor is constantly evolving depending on your environment. It's essentially a tug of war. And if there's a tug of war, and then we have to think about how to conceptualize the whole battlefield as a game. Right? In a game, you have to observe where we are in this game and how to come up with a strategy. So I would argue that the modern view of drug discovery is not just about AI, but it's about this three-pronged approach that very few people have thought very carefully. One of them is that you have to really think about the target by understanding deeply about the biology. Right? Earlier, one of my postdoc, Asan, talked about using gene terrain, and Dr. Woody talked about the kinase terrain. It's really about understanding globally what's happening before you zoom into a target. Right? So these targets are easier because you can pick any proteins to design chemicals, but understanding biology turned out to be difficult. And the second is about the modality. So modality, currently we only think about the small molecule as a modality. But I would argue that with 30 years of the development of chemistry, and we are already saturating the low-hanging fruit of a chemical molecular space. Think about a protein interaction that have very shallow surface. No matter what small molecule you put in there, it's gonna be non-specific. And so you're gonna always deal with the side effect of the drug. But if you change to a different modality, like I think Robert is gonna talk about protein degraders later, that is really an exciting development because you can actually chop up the proteins through some mechanism. Or if you think about the modality of gene therapy, RNA silencing, or even linking the drug into with an antibody that directly deliver the drug to the target, then you don't have to have that much of a dosage required to kill that cancer cell. And all of a sudden, what previously think as a unsolvable drug or a target may be solvable. So you have to match the modality with the disease target mechanism. And the third thing is about the strategy, right? So can you figure out what the cancer is doing before you actually deploy the right drug? And then that's why you have so many options. If you have double mutations, and then what do you do with the second mutation? Is there such a mutation hotspot? Can you come up with drugs ahead of the time and ahead of the schedule to prescribe to the patient. Right, so all of these questions have to be carefully thought out. I'll just want to show you very quickly a platform, how to use the gene terrain. Actually, 3 days, 3 days ago, I was talking to a Stanford professor colleagues who's doing medical physics, radiation oncology. He told me, wow, Jake, I just found out an amazing platform to figure out the how to map the single cell into a platform that is similar to what I had in radiation oncology. I was really intrigued. And then they pull out a presentation that is almost like an earlier version of gene terrain. And I was very happy. I said, well, we can collaborate, but I come up with this gene terrain already more than a decade ago. So the idea is that you can actually visualize and map all of these functional genomic information in an organic way, such that the patterns would evolve. And once you see the patterns, you can actually map it to pathways. You can also see it longitudinally. And these patterns, all of a sudden, at least according to this radiation oncology data scientist at Stanford, will open you to a suite of software tools, not just a fancy AI software tool that Alex used that would have to use millions of dollars of computing power. But even with the naked eyes, you can tell that these are all different, right? So we believe that that some of the smart algorithm is still the key, and we don't really have to have that much of a money resource. And I think Asan already shown this, is a drug treatment to tumor cells. And we noticed that one of these dosage on the cell line has a very interesting pattern that show up. And then you can isolate that and find out what pathways there are and how the gene changes at the individual pathway and the gene level. And I would argue that if you can actually collect all of the data and discover disease biology this way, it's like going into Google Map and saying, where do we go for dinner tonight? You can find any kinds of dinner and any kinds of the best restaurant review. You cannot be too wrong, right? So I think I also want to mention that once you have this, the task of this is resting in the disease biology's hands, right? Even for a simple case of targeting KRAS, right? There's so many drugs that has already been developed, right? So you see all of these boxes are different drugs and the modalities, and then there are strategies at targeting every step along the pathways. So as a biologist, you ask the question, well, what is a better target? Maybe it really depends on the situation. It's not necessarily the best target for any condition, the best drug for any condition. That's why you need to have both the deeper understanding of biology and deeper understanding of the patient. That's why I was always thinking that the future of drug discovery would have to be more personalized. That's bad news for the pharmaceutical company because they want blockbuster success, but maybe it's better news for smaller institutions like us, because then we can democratize AI. We can actually make the drug discovery as easy as maybe pushing buttons and then sending to Alex's place robots and then send it back for maybe $10,000. That's still not too bad. And then coming back and then you say, for this patient, let's just treat it with this drug, right? So it does requires a lot of infrastructure, lots of expertise, but we can finally make the AI agent to do a lot of this groundwork, right? So you've already seen the PharmAlchemy. And here I want to say that another point that the gene terrain can be used to really look at the heterogeneity of patients. So here we're using these many, like more than a dozen cancer patients from single cell gene expression data. And then you can see that gene targeting signatures in similar regions are more or less the same, but then in different regions are very different. So imagine that you use a classical pathological definition of patients, and then you're going to get the very wrong patient enrolled in the clinical trial. Your efficacy is going to suffer. Right? If we have these tools embedded in the clinic to even use as a clinical trial enrollment selection tool so that we can guarantee to select the right patient to enroll, I would argue that the problem will be a lot easier. Of course, at the cost of maybe not so many people who can benefit from this technology each time. And in fact, we also have a tool called the SEAS to investigate the phenotype heterogeneity. And so you have this multidimensional data, and then you can embed it in the UMAP. And then you have different situations. You can click on any neighborhood and say, what are the clinical attributes in common? Maybe long survival or short survival. This is like pointing to a map and saying, is this region rich in crime or not? And then we can ask this question based on 1,000 other attributes, and then all at once. This is the power of SEAS, and it's already been developed. You can use today, right? And so Rahul also mentioned about the drug feature engineering. I won't go through this in detail. So lastly, I want to mention that here you hear a lot about the SPARC. The SPARC, we want to use not only artificial intelligence, but also we want to pair it up with the right approach to systems biology. And I think there are some systems biology expert who's going to talk about wonderful things that they do in their lab in the next session. And then I think with the quantitative pharmacology that Ed mentioned, and then perhaps combining these techniques, we can work together and advance the aim of the SPARC. What is the aim? We want to make drug discovery in the future more peaceful. Peaceful stands for personalized, accelerated, and economically accessible for all, right? So especially the economic factor, and I think it's really exciting to think about it. And so we have some goals that we want to create open data sets and the PharmAlchemy, and I've got the students in the 603, why don't you stand up? And so they all contributed to curating this data set. Good job. Congratulations. Oh, thank you. So I think I'm using the class, and so they don't have to do like a dummy homework project. So I made them to do the PharmAlchemy. And then we have digital twin models, and I won't have time. And I also won't have time to talk about programmable medicine in detail. But hopefully we want to have pipelines, and with some drugs populating, may not be all the drugs that the big pharma wanted, but then we have some drugs that we can handle and then fill in the pipeline. If you do have data, if you think your project is AI drug development ready, talk to us. And may not be AI drug development ready, and then we have a checklist, and then maybe we'll send you back. Maybe you write proposal, you develop it further. But hopefully that we will nurture an ecosystem that consists of us. And in the future, we also want to involve people from CROs. They're dying to get the business, but then we're dying to select which one is truly working well. Why not to create a directory where they all can be registered with us, right? And that's it. Thank you.
In the lab where data flows, revolutions underway, bridging gaps, set to
Music
show
statement.
ACE power in full display.
Well, come back at the
flagship sets, cornerstone,
show in
future
10
so
minutes.
bright, where machines
250, okay?
In the lab, where data flows, revolutions underway, bridging gaps, set to show ACE power in full display. Flagship sets, cornerstone, a future so bright, where machines learn on their own, turning darkness into light. Ethically sourced, we stand with trust and clarity. Across lands, hand in hand, we craft new reality. Software standards unify, language shared and bold. Data's truth we amplify, story yet untold. Fair principles our guide, accessible and free. In this journey side by side, we shape what's meant to be. Resources spread, knowledge flows through every vein, an artery bridging minds. Future grows in this AI quest. Workforce trained, ready, keen across AI divides. Biomedical scenes I've seen, world forever wide. With each step we close gap between machines and human grace. Between machines and human grace. Bridge your white on map, new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. With each step we close gap between machines and human grace. On that new dawn we embrace. Together on this path, in unity we trust, for future we craft. In AI we must. In AI we trust, in AI we must. In unity we find future just.
Dr. Chen gonna use Slido right now.
I'm still learning to trust IP. Dante,
Most of the work I did.
Yeah, I'll use my phone. All right, let's get seated. So Jandals, can you ask the people from outside of coming in?
Yeah, that would confuse.
All right, we're starting in 50 seconds.
I do use AI-driven random forest.
Okay. All right, 30 more seconds to go.
Somebody will call me.
Oh.
Not very much time here, but.
It must be. Yeah. This is the blazer.
Well, it's you know, it's be seen in green.
Yeah, you have to.
Smart.
The thing I've discovered.
You never wear pharmacology and sudden research.
It's the same color.
Yeah, it is.
Yeah, people ask, where's the yolk? Targeting. Where? Hoffman used to be.
All right, let's get started. Welcome to the panel discussion session. And this session, we're gonna actually be interactive. The first 2/3 of the time, I'll be asking rapid fire questions. And I would ask the panelists to just try to limit your answers to as short and abbreviated as possible, hopefully within 1 minute each. right? So you will comment only if you have additional opinion to add or you have completely different opinions. You can initially go in nice order because we want people to understand who you are, what your experience has been, and then you can go in any orders, and as long as you can grab the mic. And then the remaining 1/3 of the time, we'll ask people to send in the questions. We do have the Slido, so let's try out the technology. If you have a phone, go to that slido.com and then wait for I advance the poll, the live poll to that open question section. You can think about the questions and then we'll go through that. Okay, and then we'll have a hard stop at 3:45. Let's get started. So my first question is going to be really simple, right? Tell us about your experience for anything related to AI or drug discovery, starting from this side.
Good afternoon, everyone. I'm Xue Zhang from Southern Research. So, thank you for inviting me to this meeting. I learned a lot of new things today. So in Southern Research, we have integrated drug discovery platform. So we have some in silico drug discovery components using artificial intelligence to design drugs. But besides that, we also have a lot of wet lab capabilities to pair with in silico design. So we have, if it's a small molecule, we have a medicinal chemistry team. They can make the drugs. If it's a biologics, we also have a biomedical molecule support. Then we also have a high-throughput screening center that they will screen. They can screen millions of drugs, and those drug activity data, they actually can be used to build new AI models. And in addition, we also have bioanalytic teams that they measure the pharmacokinetics and the toxicity related property, the data which can also be used to build new AI models. Besides that, so we have some in vitro, in vivo evaluation capability. And so far, we have drug discovery projects covering different therapeutic areas like oncology, neurology, infectious disease, and some other areas. We also have external collaborators all around the nation, and including many people in UAB. And we also, like some of the in silico tools that Jake Chen's lab previously developed, for example, the predict therapeutic targets, we also found very useful. And we are also looking forward to the new tools your SPARC Center is going to develop in the future. Thanks.
Okay, thank you. It's hard to do it in 1 minute, but
yeah. Good evening, everyone. Good afternoon, Joel Dobbs. I am executive in residence in the School of Business here at UAB, which an executive in residence is basically a former executive who flunked retirement and came back as a teacher. So that's what I do here. I spent a little over 35 years in the pharma and biotech industry, literally all over the world. I was part of the team in the U.S. in the early 1980s. It started a little company in North Carolina that grew to become what you know today as GSK. We started the U.S. version of that. I worked for Schering-Plough for a number of years, Merck, finished my career with ASI, the Japanese company, literally lived all over the world, North Carolina, New Jersey, London, Tokyo, and retired by coming back south. Most of my time was spent in executive leadership roles, particularly the last 20 years or so, primarily in research and development, the last 8 or 10 in general corporate management. And I had responsibility for a number of different areas, including earlier in my career, areas like computational chemistry, high throughput screening, a number of those things that at the time were sort of leading edge technology. And were quite challenging to deal with at that time.
Hello, everyone. My name is Ik-Jun Tan. I'm from Utah. My experience with drug discovery is involving the things that you heard this morning. While my talk is starting with in vitro, in vivo drug screenings. I'm more recently interested in looking at this immunotherapy, which is requires some sort of like a syngeneic mouse models in order to find these tumor microenvironment. And I'm particularly interested in trying to find drug combinations because I think that is something we haven't touched a lot in this seminar so far. But how do we predict drug combinations that can increase the efficacy but reduce the toxicity? So one of the ways that we are using this is to use a lot of this real world data, particularly from patient multiple omics modality, as well as a clinical EHR to find these kind of signals. And then we utilize a lot of these commercial data provider where they give us some of these claims data with treatments and response. So using that, we are trying to build a computation model that can give us signals that can predict which are the biomarkers that can predict response as well as immuno-related adverse event. Thank you.
Hello, I'm Mark Banaszak, Hall Associate Dean for Research for the School of Engineering here at UAB. Worked in nanoscale delivery, targeted delivery for cancer agents for about 20 years, as well as self-injectable morphine for soldiers in Iraq during that war. Also, most recently working on aerosol delivery for asthma, COPD, and cystic fibrosis. The way we've used AI or machine learning, different techniques for years is in terms of basically in trying to engineer drugs. And it's a big part of engineering. It wasn't mentioned here much today, but engineers use different techniques to optimize for a long time. These techniques have been available. So that's where we've used them. And moving forward, this really interesting situation, right? Because there's so many great improvements in data that we can get. And they're including a much better ability to characterize the drug and drug formulations than we used to have. And so that's where I think there's a lot of
promise. My name is Rusty Arnold. I am a professor at Auburn University's Department of Drug Discovery and Development. Prior to my academic career, I worked at Wyeth Ayers Research in liquid parenteral formulations development, then went back into academia. By training, I'm a kineticist and I do PK/PD modeling, but my research interest is in nano drug delivery systems. My experience with AI is in industry. Back in the 90s, we were interested in the highest dose possible, and it seemed counterintuitive after my kinetics training. So what we've been doing is using AI in conjunction. I am not a omics person with Dr. Amit Bitra's pharmacogenomics core and a postdoc I recruited where we can look at molecular signatures of different exposures with the goal of optimizing nanomedicines in a more precision-based approach.
All right, thank you. So I'll provide some context for my next question. I recently had a conversation with an executive from Big Pharma about the rise and falls of any technology, including AI and drug discovery. And many people in the field vividly remember just in the height of the COVID pandemic, and there's a big boom, hype of biotech, and everything rise up. And then there's a huge investment trend in 2021. And last year, it was the most abysmal time for biotech valuations, and then many people dumped the biotech share, and the AI drug discovery go along with it, to the point that even Alex this morning mentioned that there are 500 companies out there, and many of them will die. So it seemed to imply that there's an overhype of AI and drug discovery. And so my question is, do you think that AI and drug discovery is hype, or there's still hope to go? And be short and to the point. Go ahead.
Same sequence? Or...
Yeah.
Same sequence?
Yeah, sure. Yeah, same sequence.
Oh,
okay. You're on.
All right. So yeah, I think that there's hope for AI drug discovery. So based on my experience, yes, indeed, I think there's, I mean, AI right now is not a very mature technology. So I think to make it more successful, instead of just perform standard operation protocol, you better to do the result, to do the research that result oriented. For example, using generative AI to design new small molecule. If you run the program, it will give you many new compound structure. But right now, a challenge is that many of the structures, they are not very feasible to synthesize. So if you just run the program, then it wouldn't lead you anywhere, but rather result-oriented, you further refine the model or work with the medicinal chemistry team to really make sure you generate some molecule that is feasible to synthesize. Then you can actually move forward the project. And once you have either preclinical or clinical candidates, then you prove the success of your
So
AI.
your answer is no. Okay, we got it.
Yeah, a lot of hype, some hope. I don't know if any of you are familiar with something called the Gartner hype cycle. Gartner's a technology consulting firm. And they talk about when new technologies are introduced, depending on the amount of press they get a lot of hype. And they define it as after a product's introduced, technology is introduced, when the hype reaches its peak, they call it the peak of inflated expectations. And as reality sets in, the next phase is the trough of disillusionment, when you think, oh, well, this wasn't so good after all. And finally, you discover what the technology was really good for. Today, artificial intelligence is at the peak of inflated expectations. If you are a public company chief executive officer and you can't spell AI and say it like you mean it, your stock's probably going to lose 10% of its value in the equity market. It's become a buzzword. The reality is we're going to figure out in a few years what artificial intelligence is really, really good for. And it's probably not going to be some of the things that we're actually thinking about now. And maybe a little bit later in the discussion, we talk a little about some of the things that probably are realities in the drug development side.
Yeah, I think it's slightly in between. I think if it's using AI just to discover all kinds of drugs, maybe that is not very useful, because for every one of these technologies, you need human to kind of like know what they are trying to ask the questions, right? So I see AI as a tool that can help researchers to ask a certain question. So what we are trying to listening to this morning keynotes is, it seems like the technology, if it's used in the right way, you can accelerate the discovery process. But I think humans' expertise still play a very important role.
I think it's critically important. I'll give you just a very specific example. Millions of people every day take Advair Diskus for asthma and COPD. That drug was formulated 20 years ago. When they formulated it, they didn't know the relative mixture of the drug in the particle. They also didn't know, they knew that it was great to have both components, the steroid and the bad agonist there, but they didn't know why. Now we know why. And so we can now integrate all the proteomic and transcriptomics data for the why. And we can now, for the first time, actually characterize the particles that are flown into the lung and optimize them. And so you can take the why data and you can take the design data, and the AI methods are incredibly useful for fusing that information to get you to the best formulation for delivery to the patient. So I think it could just be standard.
Short answer is yes and no. I think it's a lot like the nanotech world. They oversold it and it sounded like nanotech was going to save everyone's life and it's going to change the universe. Now we know what it's actually useful for, although COVID did give us a little bump in the direction of positive. However, for many of these things with AI, as someone who does PK/PD modeling, we say this often, all models are wrong. Some can be informative.
Okay, fantastic. And I would invite the panelists turn around and look at the screen. That is the your audience response. And some of the words were blocked. I think the yes is about 39%. That's the same proportion as the no's. And the actual detailed explanation, the yes is overhyped because the biology is just too complex for machine to really understand and engineer. No is that the AI that we see today is just starting and it's changing so rapidly. And if you think about the computers in the old days, it doesn't even fit, have a whole building and then do calculator and then how much transformative it is. So I think that that's what we're starting with. So there's no conclusion, but it seems to be somewhat interesting depending. Now, my next question is, actually, if you had to make the future some form of AI, maybe not the AI that we know it today in this current form, what component do you think it must have in order to really become useful? So open up to anyone.
Well, go ahead. I will go first. One of the things, and I tend to look at drug development because of my previous life. I look at the whole cycle. And discovering and developing a drug is a bit like drilling for oil, right? You start drilling a big, deep hole and you don't know if you're going to hit oil or not. In fact, most frequently you don't. And most of you probably know that drug development is a really high-risk endeavor. And so one of the things you try to do is you try to de-risk it as early as possible. We have a saying in the industry: fail early, fail fast, fail cheap, learn quickly. In other words, you want to make the mistakes early before you get into human trials, when you start literally sometimes spending hundreds of millions of dollars. So a good chunk of my career, I would spend a morning once a week with a group of people basically doing a review of different components of our drug development portfolio. And we were making risk management. Do we continue with this program at all? How much more of it do we fund? What are the decision points we need to take? Because the later you fail, the more serious the failure because of the sheer cost. Losing a drug in phase 3 can literally cause you to write off $800 or $900 million. Your investors generally don't like that. So when you look at potential for artificial intelligence, I believe one of the things is hypothesis generation. And so let me give you an example: predictive toxicology. The idea that if I can use a tool that helps me formulate hypotheses for things I need to test early, based on compounds, similar compounds, structurally similar class effects, those kind of things. If I can do that, then I can design early studies, either in vitro or in animal models, to try to rule those things out early on. Same kind of thing with clinical trial design. One of the biggest issues with clinical trials, the number one metric that we monitored was patient enrollment. Because the more patients you enroll who meet the inclusion criteria and the faster you enroll them, the more quickly you're going to get through the protocol and hopefully succeed. And time is definitely money. And so the ability to make predictions about how best to structure clinical trials, how best to, perhaps even where best to conduct them based on past history, those are the kind of things that I think are probably going to be useful very early on. But the key here is things that enable you to de-risk a program as much as possible early on. And there's certainly capabilities to do at least some of that today. And I think that'll probably develop a lot more fully in the future.
Yeah, I think AI has to be possess certain characteristic. One of them is explainable, right? Like not just hypothesis generation, but also how we can learn from the AI why they make this decision, so that when we do the validations or experiments, right, we can kind of like know what we are trying to expect. And then I think the other part is basically equitable. Basically the data that we use to train the AI model have to be kind of like not just for certain kind of like groups or certain type of diseases, but kind of like equitable to all races or ethnicity group. Expandable. So whatever that we train in one small cohort should be able to easily expand to bigger cohort. I think that is basically in the align with this kind of like clinical trial design. And I think finally it's ethical, right? When you make this kind of AI decision, help you to give you a list of drug recommendation to take on. Why do you pick the top drug, right? Or why do you take the second drug, right? If it's involving some sort of like a AI in clinical usage, right? So is that trying to be cost effective for the patients or more like a quality of life, right? So this is basically something I think a useful AI should be kind of like considered in the usage.
I would just say wherever you actually have real process engineering present, we already know it works. Huge plants all over the world are already run AI-based models helping to optimize the way the plant runs every day, every second. So we know it works. And it's really just a question in this space of figuring out where the, the appropriate spots to plug in are in terms of the process engine.
I'll quickly add a technical point. So I think in the future, there need to be new AI algorithms. One issue right now, almost all the algorithms is that it has this overfitting problem. Let's say if you want to build a model to predict whether a compound is active or not, you collect data from literature, but the data usually imbalanced. Maybe you have 20 compounds active and 2,000 compounds inactive. If you use that imbalanced data to train the model, the prediction is also overfitting. It may predict everything inactive. So that is a problem none of the current algorithm can fix. So in the future, if there's a new algorithm that even if it's an imbalanced data set, it will not have less overfitting, that will help increase the accuracy of the AI prediction.
I think it's going to happen. And the reason why is if you look, there's already FDA approved products that are using AI for image processing. I went to clinicaltrials.gov and there's over 1,900 clinical studies currently using AI or machine learning within their title. The US legislation has passed a law stating that FDA does not have to require animal testing, although it cannot change the way they review dossiers because the preclinical models are not sufficient. And the FDA now will allow one pivotal phase 3 clinical study to approve a drug if you can adequately model and capture all the variability. All that stuff is well suited for AI.
All right, great. Let's take up on that. So as far as I know, most of the current clinical trials that involve AI are all within the radiology space. Well, actually not all, but the majority of them. For example, using AI to read images, and that's fairly common. And it's not entirely clear for people with a genetics background and cell biology background, how to develop the drug from the beginning to the end to get your project AI ready. So my next question is related to that. So let's say Dr. Chris Willey is interested in getting his project, Glioblastoma project, AI ready so that he can employ some of the late-breaking tools. In your opinion, what are the most important 3 things that he needs to do to get it AI ready? I'll give some hints, right? So it could be coming up with farm.ai-like tools and get it very accurate, precise, or is it about making sure diversity, inclusiveness, and unbiasedness, and the ethics and trustworthy aspect? Is it, or maybe finding the real good clinical problem that is relatively homogeneous and easy to solve? Or is it about the high quality data, or is it about forming an interdisciplinary team? Go ahead.
Framing your question and having high quality data.
Okay.
Well, one thing I think very important is that the dry lab and the wet lab team work together to make sure we develop an assay that have well correlation with AI. I can give one example that in some of the assays, let's say, if the target, there's many mutations and your whether the drug is active or not, very sensitive to the mutations, then you may have a very bad correlation with the AI prediction. But if you have an assay that shows good correlation, then that can really have the greater synergy to facilitate the research.
We talk about good data, but the one area where you don't see is having negative data. So we need a journal of failed experiments so that we can train the models with data, with studies that did not work.
Yeah, I think high quality data is obviously important. not just high quality single modality, but multi-model data. Something like TCGA has shown that if they make the data kind of like AI machine learning ready, there will be a lot of new tools and discovery can be kind of like using that kind of cohort. Ideally, I think this will be the data that we collected from EHR that we in some way de-identify, have longitudinal follow-up for the patients, as well as omics data plus some imaging data that will be very helpful for train the model. And obviously talking to clinicians is a must. You need to have a multidisciplinary team. And tool as well is need to be kind of like easy to implement, right? Sometimes you have a very complicated algorithm that need a lot of resources that is not available in a lot of places. So how to think about those kind of accessibility and implementation.
All right. Well, you can turn around and see what the audience says. The audience don't have access to all of you, so they don't have even someone to talk about the AI drug discovery. So they think forming an interdisciplinary team is essential. So maybe you should reach out to those people in need in your organizations. I'm surprised that the AI tools is the last, right? But maybe I should, right? So
you shouldn't actually be surprised. Okay. Most technologies fail not because the technology doesn't work. They fail because of human issues. And you have to get the human issues right first in order for the technology to work. So I would argue that that's probably a pretty good order.
Mm-hmm. Okay. Now, earlier this afternoon, we talked about different ways that AI could potentially be helpful in just changing the paradigm. Maybe it's accelerating the, increasing the success rate, reducing the time, reducing the cost. What is the killer app in your mind that the AI could do? And if it can do that, that would really fundamentally change the drug discovery.
Drug discovery, the killer app would be the one that could identify the lead compound that was most likely to succeed in the clinical trial. You can do that. I'm buying your stock.
Ask ChatGPT which one.
Yeah, ChatGPT.
So I think one of the things is every time when we start the drug discovery, the traditional way is you have to look at compounds, optimize it, and then put into in vitro systems. And we know that there's a lot of like disconnect between in vitro and in vivo, right? And when we move from in vivo to first in human, the toxicity is the first thing that they assess, right? I think those kind of data probably already collected in a lot of these phase 1 trials that have been failed, but those data is not being shared, right? So everybody kind of have to start from scratch again when they have a new compound, what type of toxicity, that kind of like similar structures. If we have those kind of information tie in, maybe that will be something that could decrease the toxicity and then try to see how we can improve the efficacy. I think that is the missing link that I see currently.
So I think for peaceful applications of AI, okay, the killer app is the digital twin of the patient.
What?
The digital twin of the patient.
Oh, digital twin.
Right, that's what lets you do the personalized medicine, right? But that's where I think we're at the moment, we appear to be very overhyped.
One thing I think is if you can develop an AI model that the so-called confidence domain is not very limited, then that will be more useful. So to be more specific, so right now, the AI algorithm, if you have a lot of experimental data, the model you train will be more accurate compared to some model you only have a very limited experimental data to train it. But then the predicament is that if there's already a lot of experimental data, probably the question is already well answered. People no longer urgently need an AI model to do predictions. Those questions people want AI model to do are those that they don't have a lot of experimental data. So if your AI model can only make accurate prediction, that required to be trained with large amount of experimental data, then that wouldn't help to solve some of the urgent problem. So that will require some more, I think, novel approach to develop AI.
All right. So next question. Let's flip the previous questions around. What is the wrong way to implement AI drug discovery? Give one specific example or a worst case scenario.
That's easy. It's but it's the same for all the work of this type. It's garbage in, garbage out. So if you put in bad data, you're going to get horrible data back. It doesn't matter the power of the tool.
Okay, interesting.
I still don't think you can trust it 100%. It has to come out and it's got to be hypothesis driven. You've got to go back and actually check it and validate it. And I think the fact that as you real time add data, your answers are probably going to change temporally. So you've got to have mechanisms in place to be able to reevaluate what your decisions were or what its decisions were.
In regulated industries, particularly in areas that involve things that are regulated by good laboratory, good clinical, good manufacturing practices, all your technology has to be validated. What that means is a set of steps that you have to go through that is inspectable by the Food and Drug Administration and other regulatory authorities that prove that you, number one, defined up front what you wanted the system to do. Number two, you built it accordingly. And number three, you tested it to demonstrate that, in fact, that it does what it's supposed to do. And I think in any technology that you are using in a very complex endeavor like drug discovery and drug development, an important part is to make sure that you know up front that whatever the algorithm or whatever is in fact doing what you want it to do. Otherwise, just kind of throwing things out there and hoping they work is not a very good strategy. And anybody who has encountered or played with something like ChatGPT knows that you can get some very weird answers because it's still very much a work in progress. And I think what you're going to need is a lot of stability in the technology and a lot of essentially validation that in fact the algorithm does what you want it to do so that you have confidence in the results that you get.
Another caveat is that never take the face value of the AI prediction. So to be specific, one example is that we try to use the explainable AI algorithm as much as possible compared to the black box AI algorithm, because once the AI predicts something, the explainable algorithm actually will list what factors it make that prediction. Then based on our drug discovery expertise, we can kind of tell whether those factors make sense or not. So that can help us to judge whether the AI prediction is reliable. We should put some resources to do it, or be cautious, don't trust it. However, if it's a black box AI algorithm, you don't know why it gave you the result, then it's also hard for you to judge whether it's reliable or not.
All of it above.
Yeah, okay, great. Now, I think I heard a lot about the data, the importance of data. So we already know in other disciplines, sometimes data is very important, sometimes it's not as important. When you're calculating the starting of the universe, you can just use supercomputer and then use a theory and then let the computer simulate everything. On the other hand, if you want to even just do a fingerprinting system and then catch the criminals, you have to have sufficient coverage in the population, right? So in your opinion, there's a coverage and the quality. When is it enough? Is enough so that we can start leveraging on these data to do something? Or you think that we'll never be able to actually get all the data because hospitals are always going to protect their data. Public is always behind on the pharmaceutical companies. Pharmaceutical company, in order to be competitive, they will always lock up their negative trial information and not let people learn from it. right? So, and yeah, just I would like to hear your opinions on that.
I think simulation is good, but I still trust real data. So and I guess people are already aware that having access to EHR real world data is going to help their whatever research program that they are interested in, and they already have a lot of kind of like data vendors or data company that try to harmonize, curate, and then try to add in other modality from different kind of like molecular testing vendors or imaging data, right? So eventually this is going to happen. It's just that how do we encourage the process, right? I agree with you. There's certain privacy and security issue for patient data, but if it's a kind of like put into a certain honest broker, whatever cloud systems, right? That might be something that is going to be easily access those kind of data and then get them into some sort of like an AI machine learning ready, so that whatever new tools or development that you want to do, you can just use those kind of like a benchmarking or standard data sets. That is kind of like my hope.
You know, we already have chemical plants the size of the city of Birmingham that are fully digitally twinned and fully operated with AI systems in a reliable, very powerful way. Okay, so this exists. I wasn't kidding when I said the killer app was the digital twin of the patient. When we get to the point where we can model the patient as well as we can already model these digital plants, these plants that are the size of the city, with the same level of accuracy, will be there. You know, as you already pointed out today, the NIH is doing away with animal models where they can. Why? Well, they want to do away with them because of the ethical reason of using the animals, and they don't work that well, right? And we already know that the cell culture models, frankly, don't work that well. There's all sorts of things that are complete artifacts of that cell culture model that have nothing to do with people. And so what we really need, I think, to get to this point in this answer is we need that digital twin. So I was serious when I said what the killer app was. I also think it's incredibly difficult to get there.
All right. So this is the most challenging one, okay? So Alex painted a somewhat gloomy picture for academic in the morning, right? So he showed not only that all the AI software that can significantly improve the speed and the success rate of drug discovery, but also he's building up robot that can maybe take instructions from future AI, do experiment. Essentially, we have many hardworking bench biologists here, clinicians here, and some of you may be scared. And I'm not as smart as AI. I'm not actually, even me, I'm not as smart as GPT-4. And when GPT-5 and 6 come out, when Alex is deploying hundreds of thousands of these AI agents, am I going to be out of job, being enslaved by AI? Or there's a role on the positive side for experts, professionals, or even students here to actually come back and then maybe leading, helping, or serving the future of AI drug discovery?
So I think that is a very interesting question, and other people asked me similar questions before. So my answer is that I think that actually AI and robots, they actually help to prepare human for higher quality jobs. So because those AI and robots, we use them to replace some repetitive jobs so that we can actually focus more on more creative jobs. And that also requires the effort of the whole society. For example, the government also needed to upgrade their education system to prepare the next generation of workforce with more advanced skills, so that instead of you do the job that robots do, now you manage the robot. And because this is a more advanced job, you just from very pragmatic perspective, you also actually may have even higher salary. So it's actually make your life and job career better. But of course, there need to be a education system to prepare people for these new types of jobs.
You know, almost any innovative technology has the potential to disrupt everything going on around. And there's always the concern about the jobs that will be lost or be made redundant as a result of technology. But what always happens is that additional opportunities open. And so the counsel I would give people, particularly students, is be open to opportunity. Don't look at advancements in technology, regardless of what field you're in. Don't look at it as a threat. Look at it as an opportunity to you know, continue to grow and develop and you know, make the contribution in a perhaps a different way than you had originally envisioned. But I've been around on this planet quite a long time, and I've seen a lot of technologies come and go, and I can tell you that every one of them was perceived initially as a threat. But at the end of the day, you look back years later, what happened is they opened up a whole host of other opportunities. And I think artificial intelligence is probably no different.
Yeah, I mean, take GPT as an example. I mean, nowadays, I don't think any individual institute or group can develop a generative AI, right? I mean, model as big as those kind of company develop. I just came back from a Cancer Informatics Society meeting. And then the way that they are discussing over there is how do we refine or engineer the prompt, right? Because every prompt that you give out to this GPT, they gave different kind of answer. How do you refine it? Maybe it's a curriculum that potentially we should start thinking about how to teach our students, right? Because those prompts is going to eventually help you to use the tool more efficiently and effectively in whatever you are trying to do. That will be kind of like, I don't think it's going to replace human, but if human doesn't use the AI, will get replaced.
Okay, all right. So now we get into the audience questions, and there are a lot coming through. I would say that maybe we'll spend at most 2 persons on each question. Who want to go with the bottom one? Are there any studies on patient perspective on AI-led preclinical clinical studies? Do we predict to encounter AI mistrust as we see with medical mistrust? Let's jump in. It's sort of complex questions.
I don't know if there's any studies on patient perspectives. I have read there's studies where they're using diagnostic imaging and AI for, say, looking at mammograms. And I would assume that patients who are having their mammogram read by a computer could be concerned that they're going to miss it. But from those studies, again, it was still being read by a clinician and then compared to the AI. The AI did identify things the clinician didn't find, but it also went the other way as well. So I haven't seen any studies where but I can imagine it's like any technology when they put anti-lock brakes in cars, people had a heart attack as well. They didn't want to use them.
Okay. How do you define bad data, given historically no data was collected with future AI application in mind? So I sort of remind me in microarrays, right? So we did a lot of microarray hybridization. Now next-gen sequencing comes, and then everything was going to be redone. And now single cell comes, everything is redone. It's always the previous data is bad data.
I don't think it's like a bad data. It's just that technologies, right? At that time, microarray is the best tool out there that can comprehensively give you an overview of this gene expression. And obviously, it has limitations that we know, but it's also led to some of this signature that has been discovered in microarray, but can be reproduced in RNA sequencing level. So I think if the questions or the hypothesis is correct, and then the data it's just a way that we collect it at that particular time, right? Technology will basically be able to refine it. That is kind of like my opinion.
I would define bad data in two ways. One way is that there's a lot of scientific reports in literature papers that are incorrect. The experiments were not done properly, and the papers are wrong. And the problem is that most people running AI are very, very proudly tell you how they've scraped all the literature. They don't tell you how they've done a quality control on that literature. And so that's one place where AI really breaks down as compared to the human researcher. Human researchers are a little more discerning and concerned about the quality of the groups that they're using the data from. Whereas if you've just done a broad scrape, you're building in bad reports into your data set. That's one type of bad data. The other type of bad data is work that is, I would say, the experiment was a fine experiment, but it wasn't designed to give the answer of the type that the person using the AI is now trying to take from that data. And so they really shouldn't be extrapolating the way they're doing it. And that's the other really big problem.
All right. Let's move on. The next question is human in loop, a solution in AI-based precision medicine. In the loop, maybe? Inform the human.
Yeah, like I mentioned, human in the loop is important, right? Because whatever that we just discussed over here, it needs validations that require human, right? And then anything that you kind of like checking the quality also require humans. So I think that is the basis of precision medicine.
Okay, next question. How can AI prioritize target identification? How can you tell which molecular target is more oncogenic or better target than others? It seems that I can answer if that's okay. Oh, no, I think there are different ways, and one is more looking at the target protein intrinsic functions, and the other is the contextual functions that infer to the target. So intrinsic function is, let's say you study a protein sequence. You look at also the gene mutations, and you try to figure out what are the position by position functional significance of that proteins. If you replace that, would anything happens? What are the protein domains? What are the three-dimensional structure? That's more traditional, right? And then by looking at the contextual is what the network biology is trying to say. I use that example. You can look at functional interaction partners and see what protein, other protein that have different function, sort of do it inference based on Gilbert association. Or you can look at where does this protein get expressed in different perturbation, stress conditions. Eventually you can infer that function. And in the end, you can put a score based on every measure that you have, maybe some intrinsic, some extrinsic, to come up with a ranking. And that ranking is only relative to the problem. There's never an absolute ranking, like the best medical school in the US. You can say that the UAB right now on this topic, we're probably the best. But if you look at the funding, they're probably more straightforward way of measuring. But then there are many different ways to do it. And there's more of an art than engineering. Okay, how would the peer review process work for AI-led research?
You know, the question is, can AI peer review AI? If it's peer reviewed, it is being reviewed by artificial intelligence by definition. So let me tell you a quick story. This last year, I had a student who wrote a written assignment, and it seemed unusually clear and lucid for that particular student. So I copied and pasted it into ChatGPT and asked the question, did you write this? To which ChatGPT responded, why do you need to know? There's, I think one of the big challenges with compositions that are done by artificial intelligence is not only the accuracy of them, but it's being able to know exactly who did them in the first place. In other words, did a human actually do the research? Did a human actually write this and review it, or was the whole thing concocted based on a series of questions or propositions that were put to an intelligence engine? And I think how you peer review this, it's got to be reviewed by human beings who know something about the subject matter in which they were viewing, very much the way we do peer reviews in academic journals today.
Just to add to that is, I'm sure that you probably see some retraction paper that basically just copy and paste of ChatGPT reply, right? So some of them is kind of like very obvious that the researcher are basically using that ChatGPT to write the manuscript. The other thing is also, I think recently there's some kind of like looking at some of this publication, there's certain terminologies or words that is not commonly used by human, but by using ChatGPT to write, they often seen it as like those kind of words is have a higher frequency. So that might be some way to spot this kind of AI generated text.
Okay, we are limited by time, so we can only entertain one more question. So I'm going to cherry pick, and let's say here, please give us your insight into developing ML AI model based on mechanism of action of a particular class of therapeutic drugs.
Well, so I think one good way to develop is that you develop a hybrid approach combining both the protein binding affinity, specificity, and the phenotypical activity. So, for example, if you, there are in many cases once a drug may bind to the protein, but it doesn't show any effect on the cellular level. So, then in order to have a robust prediction of mode of action, you first either use AI or some molecular modeling approach to predict whether the molecule you are studying binds to the protein or not. But then you also have another AI or other approach to predict whether it shows the phenotypical activity, whether on the cell level or on the whole body level. So if the molecule predicted to bind to that protein target, and then also predicted to show the corresponding phenotypical activity by the other approach, then that double hit is the most reliable of.
Okay, well, the last one before we finish this is going to be a gesture, right? So no, not coming from there, it's coming from me. So the gesture is, I say moving forward in 10 years. More than 50% of the drugs are developed by AI. Thumb up, thumb down, or even. Thumb up meaning that you say yes. In 10 years, more than 50 of the FDA approved drugs will be based on AI. Thumb down, no.
All or
Yeah,
completely based.
all. Right, so I want to see hands. Okay.
I mean, it'll be.
All right, let's make
You're
a
gonna
picture.
play a role.
Yeah, let's all right, can you stay on the, yeah.
Guys?
It'll play a role, but
Yeah. Yeah.
Okay,
I'm with you on that.
we only have one thumb down, right?
I'm the thumbs down, but I'm in agreement with him. You say developed, you're by definition, that means soup to nuts. No way. I think the other thing is that we've not talked about is the fact that all of these things have to be approved by regulators. In Food and Drug Administration, you got some smart people, but they don't exactly move with lightning speed, and they're extremely risk averse. And so anything that is happening in this field is going to require, they're going to have to be on board. And I don't know what that looks like.
Okay.
I'll just make a comment for there. If you're interested, there is 2 current white papers out on the FDA website looking for comments related to AI in manufacture and AI in drug and biotech development.
And I would just say the FDA already has been working on AI for approval. So they're already.
All right, well, let's give them a round of applause. Thank you very much.
We're just moving straight on, right? There's no break.
Down.
Sounds good.
All right. I'm gonna help moderate this final technical session. Got some strong group. Unfortunately, it's always at the end of the day, so we'll see how much attrition we face. But I think this outstanding group. So our first up is Dr. Ben Chen, who's at Michigan State. He has really a kind of a perfect background, I think, to lead into this the topics that we have here. You have somebody who was chemistry, then into software engineering, then to chem bioinformatics, then in pharma and back to academia. Quite a row of positions. So with this, I think we're gonna move on just for sake of time and let him begin. So, all right. Sorry. There we go.
First, thank you for the invitation. It's very good to be here. It's really truly honor to share our work after the outstanding panel discussion. I'm going to share a few real AI-based drug discovery in academia. And we certainly experienced many of the challenges we talked earlier. I'm going to share some of them. And I'm very glad to be here because one of my collaborators is really based in UAB. I'm going to share some of our collaborative work as well. I'm running a competition drug discovery at Michigan State. The translational research from patient to get the tissue and tissue characterization to identify drug targets, biomarker, drug hits, to even selection of the right model for the validation. We think every step can be driven by the big data and AI model. So my lab developed a bunch of tools to support this stage, especially we using transcriptomic data, which are going to be most widely modalities. But people mainly use this data for the understanding of the disease mechanisms. But we are thinking how we're going to use data for drug discovery. My lab has compiled a big transcriptomic database comprising over 20,000 samples, including pretty much all the cancer and cancer subtypes. We're using same pipeline process all of the data that makes them comparable. So you simply look at the cancers, compare to control to get the disease mechanisms, right? From the drug discovery perspective, we really want to find the drug to push the system from the normal state to the from the disease state to the normal state. But when you look at the transcriptomic profiles, you always get hundreds or even thousands of disease genes, right? And there's no magic bullet. And if we're using traditional drug discovery approach, target-based approach, we may have some challenge. So my lab, we are using the system-based approach to sort of identify the drug to reverse disease gene expression at a global level. And this morning, Dr. Tan already briefly introduced us the method and followed by a couple other speakers, their talks. So I'm not going to talk this in details. And actually, I was inspired by the drug gene disease sort of work by Dr. Jackie Chen when he was a faculty member at IU and when I was a graduate student there. So I learned a lot from their work. And this just simply should show you how this drug can reverse disease signature. This first one just shows a perfect case, but in many cases we couldn't see this type of say relationship. And my lab, we in the past period we extensively implement this approach and also apply this approach in the real drug discovery. We identify drug for Ewing sarcoma, hepatocellular carcinoma, basal cell carcinoma, Alzheimer's disease, COVID, melanoma, and prostate cancers. We have pretty much all the drugs that we validate in preclinical models. To facilitate the community to use this approach, we developed the Octet, the open workspace for virtually screening therapeutics targeting prostate cancer patient group using just gene expression features. The idea is very simple. You just give a cancer, specific cancer, or a cancer subtype based on molecular feature. For example, MYC amplified lung cancer, stage 3 breast cancer, and our system can pull the relevant samples. And we have the AI model to select relevant tissue sample. By comparing disease sample and tissue sample, we're able to create disease signature, and then just feed the signature to our drug discovery pipeline to identify the drug hits. We have the web portal available and recently upgrade, make it much faster. You're welcome to give a try. We also have our bioconductor package where you can perform the large scale drug predictions. Today, I'm going to quickly use one cancer example, the DIPG, diffuse intrinsic proton gonema. This is a collaborative work with Rentalo, Dr. Rentalo Hashtum, who is a faculty member at UAB. The DIPG is a rare pediatric cancer, only 300 cases, but it's very fatal disease. Their 5-year survival rate is pretty much 0. In this disease, there one challenge we are facing is that we are able to get the adjusted normal tissue because we know the pediatric patients don't want to donate their adjusted normal tissue for the research. So we spent some time to develop some AI method using other resources to be able to create a disease signature. We also using the single cell data to create a signature for individual patient. By for each signature, we get a bunch of disease the drug hits, then we combine them, and then we did the in vitro, in vivo validations. Before we started our experiment, also did a in silico validation, where we pulled the compounds which already have the drug effects data from other studies, and we found that they predict potency to reverse disease gene expression really highly correlated with the drug efficacy. That means if our compounds we found to be able to reverse the disease gene expression, DIPG gene expression, very likely this compound can be effective. Then we, using this approach, we screen the entire links library, and we identify 4 compounds which can reverse the DIPG signature, and they're quite novel. Look at the structure, very different. 2 of them are actually referred to the same drug as the morphetels, MFF, which is a immunosuppressant. All of the drugs are effective in the DIPG cell line, and especially MF is particularly appealing because this is immunosuppressant. We think we may be able to repurpose this drug for DIPG. Then we test this compound in intracranial tumor models where the tumor grow inside the brain. Unfortunately, we didn't observe this compound there any anti-tumor growth because later on we found this have some penetration issues. This compound was not able to penetrate into the brain. But this compounds indeed can inhibit the tumor growth in the Xenograft model. This simply tells that we need to improve our compounds. We need to design the better compounds which can penetrate the brain. And this allows to actually lead us to spend a few years work on our AI model to use our approach to do novel compound discovery. Another reason because we have been working on this approach for many years, so we have we have identified the drug candidates for over 7 indications, but we're not able to move this to the clinical trial. One of the reasons is that in general, drug repurposing does not have huge interest to the pharmaceutical company. So it was very challenging for us to get investment to move that forward. And all the feedback I get is that you guys need novel compounds. So in the past few years, we spent a huge amount of effort to use our approach to do novel compound just discovery. Using this approach, we have to get the compound gene expression, compound-induced gene expression in place, but there are millions or billions of compounds, right? It's impossible to generate such data. However, there already have massive drug-induced gene expression data. We can use the machine learning model to build model. Essentially, if we can use the machine learning model to predict the drug-induced gene expression solely based on the chemical structure, then we're able to do the novel compound discovery. But of course, this is very complicated. You can think about the gene expression really depends on many other factors, chemical structure, cellular context, biological conditions. So in the past few years, we developed a Serous model to improve through the collaboration with the computer scientists. This is a simple model neural network. You have the chemical fingerprints, gene feature, we simply predict whether this chemical can change the expression of that specific genes. Then we implement the graph neural network for the chemical structure embedding. And this, the chemical, this type of deep learning is you're data hungry. And we figure a way like build the contrast learning for pre-trains to address the data sparsity issue. Just now we mentioned about garbage in, garbage out. For those of you who work on the links, you probably know over 40% of the data usually have poor quality. You probably don't want to use it. If you use it, probably that's kind of the usual garbage. So we spend lots of efforts to build the model, collect collaborative learning to improve the data quality. The key point is that now we can have a model to really be able to predict the drug gene expression solely based on the chemical structure. With this, we can perform a virtual drug screening. We can create a virtual gene expression profiles for any drug library. So this is the Zinc library. They has commercially available compounds. We did also computational validation, cynical validation. This just simply shows that if we choose top 2% of the compounds, our hit rate is 50%. So this is pretty amazing. Then we go on to do the real drug discovery. We screen the entire Zinc library, 7 million commercially available compounds, narrowed down to 2,000. And then we test about 20 compounds. With about 2 compounds, we were pretty positive. And those compounds, we think they can predict to be able to penetrate into the brain. Now we just recently finished the toxicity study. We are waiting for the in vivo study. Well, with this model, we can even do much more. We can search the entire chemical space, which is like 10 power 60 million. It's 10 power 60. And we developed computational tools to be able to search the chemical space and to actually do the lead optimization. So we have the lead compounds, so we can design novel compounds and to synthesize. We haven't implemented this in DIPG, but we do have some success in liver cancer, which is the cancer I've been working on for over 10 years. Similar idea, we create a disease signature by comparing the transcriptome profile, and then we use our approach to the drug identify drug candidates. We identify nicosamide or the HSCA treatment. And the last few years, we really spend pretty much all the efforts trying to use the novel approach to identify the compounds, novel compounds. This is pretty much like the traditional drug discovery process. Do the virtual drug screening, identify the lead, to do lead optimization, followed by the PK and efficacy study. We're very lucky to be able to identify the needed compounds. And this entire process takes us over 3 years. So we build only spend a few months build the model, but another 3 years to work on this. So I'm very, very impressed by Alex Keynote, able to move the compounds within a couple of months. But in academia, I think it's very challenging. You just think about how to get the compounds during the pandemic. Even order the compound take us a couple of months, and you have to reserve a few months for the synthesis, and you have to do a new optimization. That's multiple iterations in terms. As a result, it's to take a couple of years to get this there. But this just show you proof of concept that we can use this AI approach to identify the drug candidates. At MSU, we are building a transcriptome-based drug discovery program. We are mainly using the transcriptome data. For the given patient from the hospital, we perform single-cell RNA-seq, and we're screening this for the link library for repurposing candidates. We also perform the virtual screening to identify low-level compounds. Once we identify the compounds, we do individual screening. And typically, we order just 30 compounds. Our hit rate right now is about 10% for the novel compound discovery. Repurposing, our hit rate can be up to 30%. Afterwards, we synthesize the compounds, and we also consider the IP, etc., many other medicinal chemistry properties. Afterwards, we do PK in efficacy study. If this is repurposing candidates, we want to launch the clinical trials. The one thing I didn't mention that we also focus on the patient. We using the patient-derived ex vivo culture model to help us to validate. Right? And once the compounds validated, we hope to be able to launch a clinical trial. If this compound is novel, we are going to spin off to hopefully to fund the company to move this forward. And we have built the web portal for the community to use. This is the purposing portal, novel compound screening portal. We have another portal to really mine the transcriptomic profiles. Given any transcriptomic profiles, you can get the feature of other modality like their protein abundance, the metabolites expression, etc. We have another bunch of the models for like the biospecific antibody identification, cell surface protein from the single cell transcriptomics. If you are interested, feel free to check it out. With that, I would like to thank all of my collaborators, especially Dr. Rentoro Hashizun, who is here, for the validation of our compounds in DIPG. And we also work with the group of one group at Stanford to do the novel compound drug discovery in liver cancer. Right, thank you all.
All right, thank you. We have time for questions. All right.
So your engine is able to basically predict any novel chemical scaffold that has a gene profile to be expected, and how many, like, what's the size of the gene panel you currently correlated with?
Yeah, so we there are about 2,000 genes are predictable, not the entire transcriptome. Yeah.
Yeah, so this is a great slide. I really like it. I guess one thing I'm wondering is you're taking the patient, you're getting their information, and then using that to find the drug for that patient, right? Which is great, but you don't. So how do you think about it? This in terms of normal drug approval processes, because you can't do a safety profile for that one patient. You can't do an efficacy profile for that one patient before you give it.
That's right. So we have two directions. One is to repurposing existing drugs. So this is the IPF, is idiopathic pulmonary fibrosis. There's no therapeutic. So we work with the clinicians and hopefully they able to launch a clinical trial based on the our preclinical validation data. That's the only repurpose. So this means the drug, there's no concern about the safety, other issues. But for the novel compounds, that's a different story. We're gonna fund a company and hopefully to get investment to move that forward. Right. Yeah, it depends on we're doing both. So we'll say which one we eventually can to be able to benefit the patient. But I think the purpose is probably much faster. However, I don't think in practice it's a challenge because it's hard to get investment.
Yeah, I think some of it does remind me of the functional precision medicine like they're doing at Oregon for, more simple tumors like AML and so forth, but for repurposing, testing and implementing, but for novel stuff, you still have to deal with the regulation. So. All right, any more questions? All right, thank you very much. All right, so our next speaker, Zhongming Zhao, hopefully you didn't mispronounce that too bad, is chair and professor and precision health director at UT Houston. I think we may have overlapped a little bit at Vanderbilt when I was there, but bottom line is really has a very impressive CV and has been involved in so many different projects and will give us some insight into kind of precision medicine using more advanced AI models.
Okay, thank you, Dr. Wiley, for the introduction. Thanks, Jake. We have known each other for many years. It is really great to visit your university and the center. And I really enjoyed the program, a lot of exciting presentation. And my talk here is a little bit more focused, try to actually relate to the previous Dr. Bing Chen's talk, but we are not focusing on like 3 million compounds or like 30,000 drugs. So here is mostly focused on the cancer drug and the prediction. So before that, I'd like to give a little bit overview. As Dr. Wiley mentioned, I have been very luckily able to run a large computational lab for many years. So the cancer drug or related potentially drug target has been one of the main foci in my lab. And this, because of time, we are behind the schedule. I try to make a brief about my talk, but this one is only one slide to summarize what my lab has done in regarding to, to leverage the largest scale multi-omics data in cancer genomes, and then how to find the potential features or ideally the potential drug targets. So this slide basically summarizes several major features in cancer, drug genome, the cancer genomes for the drug, those kind of potential drug targets. So we use a positive selection, mutation hotspots, but most importantly, we use like a functional domain ligand binding site, phosphorylation site, and the allosteric site, and the gene fusions. Obviously, it's not only my lab, many labs have been done this very actively, but my lab, we are very lucky. We developed some novel computational approaches, and some papers were published in the journals summarized in the right side. So here, I like to take probably 10 minutes because we are behind the schedule. So talk about the work focusing on cancer drug prediction in cancer cell lines. Again, everyone say, why you work on cancer cell lines? When I worked in Vanderbilt, many people ask me this one. I said, well, give me the real world data. So if you do the tumor biospecimen collection and through those kind of clinical trials, like today we have a couple presentation about the clinical trials. It's not only very labor extensive, but also very costly. So we have to use some like animal model or cell line to find the potential drug candidates, and then goes to the real world, the patient care. So this work was done by a few members in the lab, mainly Pei-Ling Jia. Now she became the independent faculty in China, leading a very large precision medicine program. And Ruifeng Hu moved to Harvard, and Yin Wang became a junior faculty. So actually, I have another slide. Unfortunately, I deleted because of limited time. But this one still can tell how this the generative model, we call it VAN, variational autoencoder, because it can really take care of different kinds of subset of data. And then we follow the elastic net to find the optimal drug prediction. I will give more detail next slide. So in general, we use the two available large-scale cancer cell line-based drug response data set. One called the CCLE, Cancer Cell Line Encyclopedia. It has 1,100 cell lines, covers 33 cancer type or subtypes. The good news, this kind of data, even though cell line has all the genomic features like the gene expression, mutation, copy number variation. Bad news, only 24 drugs. Then we choose another one called GDSC, genomic drug sensitivity in cancer. It has same 1,100 cancer cell line, but it covers more, 250 drugs. And these two data sets were generated by different groups independently. And then good news, when you develop computational approach, people always don't trust your work. Even you show the beautiful AUC value, the very good prediction. So you need a validation. And the validation can be external, which is very common, but also can be internal. In this case, internal, we use a 14 drug between these two data sets. So this same drug, so we can see how likely the robustness of the prediction between these two data sets. Then we also use TCGA, which has about 10,000 cancer genomes with all those kind of molecular background. So for the validation, and we also use another 6 independent data set for validation. Because of time, I don't give a lot of details, and these slides show how the workflow of this pipeline. This is a little bit detail about the model we developed. So again, remember the transcriptome we typically talk about the gene, has over 20,000 genes. And then you have 33 cancer type and 1,100 cancer cell line. So it's a matrix. Then you have 251 drug from GDSC, 24 drug from the CCLE. So it becomes a super matrix. And then we use an autoencoder, the encoder to convert the data to the vector. And the vector, we have two kind of vector. One, naturally, you have a mean value of the vector, another is standard deviation. Then we put it in the latent vector and use a convolutional neural network, decode the features, and recover the data. So we have the output. When we look at the output, we found that each time we run the same procedure, the output could be slightly different, even though overall is consistent. Then we decided to run a pool, 100 those kind of models, same process. And then we use elastic net to find those kind of consensus of the drug prediction. So that's the bottom, the drug response. Unfortunately, I couldn't move. Oh, okay. So it's the bottom. Then based on that, we find the optimal drug prediction. And then we predict every drug in each cell line. Remember, even though these kind of two data sets have been very well developed, you still have a low quality of drug response data and also some missingness. So through this, we can also fill in any gap. And finally, we use TCGA and 6 other large set drug response data sets in cancer, mostly real tumor-based drug treatment. So here is one result. First, this 1,100 cell line, they came from different cancer type, which means cancer is, those kind of cell type represent the tissue type as well. So when we do the t-SNE clustering, you can see those 1,100 cell lines, they are not scattered randomly. They form the cluster. And those clusters, you can see, can be formed by those kind of tissue type, or obviously cancer type as well. So this, what does that mean? That means, you know, those kind of cell line has a genetic component, has a mutation, gene expression. They form the basis for the drug response. That's why we can do the drug response prediction. Then we apply a very interesting kind of mathematical modeling called EREX, expression regulatory component. This EREX was, you know, originally developed by Scan, my former colleague Nancy Cox. For the gene expression as used through this EREX can serve as an endophenotype. So linking to the genetic variation through this EREX can predict the phenotype, which is including the disease. And this time, first time, we also show this EREX can serve as an endophenotype, can predict another phenotype. In this case, is drug response. Under the D figure here, you can see the CCLE and the GDSC. This expression regulatory component accounts for about 2/3 of the genetic component towards the prediction of the drug response, which is really elegant because you through this, and because these two data sets independent, you also see the consistency, showing this EREX potentially can be useful for the drug response prediction, and also forms the foundation for those kind of the deep learning model. Then we predict the drug, and there are many results, obviously. And here I just show one drug called the 17-AAG in CCLE, and the same drug has different name, common name, and called tenascimicin. This drug is an antibiotic for the HSP90, mostly used for the those kind of cancer, both liquid and solid tumors. So we show this drug in this kind of 1100 cancer cell line. So if we observe the drug response in GDSC and the CCLE, the consistency is pretty high. And then what about the GDSC model? We found a prediction and the observed one still have good consistency. Then likewise, CCLE the same. So what about those 1100 cell line? Both predict the result. One is based on a model learned from GDSC, another from CCLE. We still show a very good consistency, suggesting the prediction can be useful and robust. Then this is another slide showing the more detailed result. We look at the specific 14 drugs common in these two data sets. And then in these 14 drugs, we look at those kind of prediction in the 33 kind of cell line, which kind of consistency measured by Pearson's correlation coefficient. And remember, this log P is a y-axis value is log P about the consistency, and it's showing most of the cancer cell line, the consistency is very high, and especially for the last 4 parts, the very high consistency. Then we did a lot of validation. This work was originally submitted to Nature Biotechnology, reviewed 3 times through 2 revisions, and they asked a lot of validation. Eventually, 2 reviews said acceptance. I don't have any further concerns. The other one said you address all my concerns, but I still think it's not a Nature Biotechnology paper. Then it was rejected. But we show a lot of good validation result. In this case, we focus on erlotinib, which is famous non-small cell lung cancer. So through our prediction, we also showing those kind of the risk, the high and the low risk value. We show this is consistent, and then we also look at the different model and in the different data set, and showing this kind of consistent result in different subtype of the cancer, like BRAF V600E. And the second one is we also look at the other type. One is called a PCR, or the pathological complete response in breast cancer. And this is somehow you couldn't see all of them, maybe because of different computer. This is called a Taxol, which is a very commonly studied drug in breast cancer. So we use this to predict this PCR as a measure of the success of the cancer treatment. And the pathological complete response for the beta response means they have better survival rate, survival outcome. So through our prediction using CCLE model and the GDSA model, we see this Taxol has a better prediction for the pathological complete response. And then we found another 2 data set, and using the both model from CCAE and the GDSC, constantly showing this drug has a better, PCR outcome based on our prediction model. So suggesting this, the model can identify some other clinical outcomes through the prediction. So because of time, we did a lot of validation work, including tumor microenvironment. So in summary, through this deep variation of autoencoder model, we were able to train 1100 cancer cell line and use the model to predict the drug response reliably. And we also first time to show this expression regulatory component, EREX, is a useful measure to predict the drug response as a phenotype in the cancer cell line for many drugs. Okay, I took another 2 to 3 minutes very quickly go over my lab, since I think in the informatics conference, we usually like to introduce the program and the resources and the tools. So my lab, I built this bioinformatics system medicine lab since 2009 when I was recruited to Vanderbilt. And later I moved this lab to my current university, UT Health, Houston. So we did a lot of work related. One main focus is the genetic variation. How can we develop a computational analytical approach can identify the role of genetic variation in complex disease? In cancer, we try to find the actionable mutation, which means those mutations not only driver mutation, they have therapeutic benefits. And in non-cancer or other complex disease, we usually develop a computational approach to better predict the risk to the disease. And then we do those kind of genome-wide association, pheno-wide association, and linking of the electronic medical data. And we also study a lot of single-cell sequencing, develop tools, and then study disease and also early development. And also, like many talk here, we do drug targeting interaction and linking to the EMR to see the drug side effect and outcome. And now many people like me, my lab too, try to develop deep learning for the genetic feature mining and the drug response. So here's one last slide to showing the single-cell omics developed the method and the tools developed in my lab recently. So we developed MTNN, and inside both are those kind of the deep learning, those kind of approach. One is addressing the missingness of the data. Second is address the match effect, because same disease or same condition, you may have multiple data sets. Some like it can be over a few million cells, other only maybe a few thousand cells. How can you deal with data? And we develop a driver to study the transcriptional factor and the related program in the single cell data set. And the mitochondria, you can see is a mitochondrial variance. And then we develop a DECS, decoding cell type specificity of the single cell data set in the specific condition. And we developed the web server. And then about 2 years ago, we developed a single cell for the genome-wide association study. And finally, I'd like to thank many people in the lab. The highlighted the board here is mostly related. Many people become faculty, and also my Vanderbilt colleague and some work in my current university. Thank you for your attention. I stop here. Thank you. Okay, finally, I'd like to mention this conference with Dr. Jake Chen's permission. So October in Houston, please welcome to join us.
All right, thank you. Questions?
Okay, good.
Algorithm, is there any component of explainable AI in the algorithm that you might, explain?
Oh, AI for
Or
G-Rex.
any
I
new
should talk with
biological theories involved that
yeah.
it's actually
G-Rex is it's an overall studied model. They put a gene expression and a relationship, and because of the gene expression, reflect the genetic variation. So that's kind of an idea. So for AI, that's a good question whether it can be applied to AI. But indeed, we talk about it's a follow-up, not a G-Rex. It's a follow-up. We talk about it in the morning, like foundational model for cell type specificity. So we have a lot of those kind of serial component-based gene expression model developed like, most recently called SCGPT, single cell GPT from Bo1 to Render. And last week, I also heard about another talk, similar foundational model. So this kind of model, they have genetic variation, they have gene expression, they have very large scale, like the most recent single cell based foundational model for prediction is about between 45 million cells. That's crazy. And now how can we put this for the drug response prediction? I think that's something in my lab we are talking about it. If we are interested in, and Pingzhen, Jackson, many people here, potentially we can work together. Yeah.
Additional question.
Oh, another question. Okay.
Thank you for a great talk. So I think it's my question is, because the tumor cell or tumor by itself, any cancer like a heterogeneity, and then after treatment, they are changing subclonal evaluation to, acquire to resistant to the therapy. So I think it's, AI can predict, subclonal evaluation during treatment, how, changing, subclonal population. So AI can predict the kind of, evolution of changing in response to the treatment.
Yeah, you are talking about like different ethnic background, those population, how the AI can have a better prediction, for example, drug response, but also potentially can be many other outcome, like phenotypic outcome, right? Yeah, indeed this is, and this model exactly can do it because the baseline of this model is based on genetic mutation, genetic variation, caused the gene expression, and then as one of the outcome, because here treated by drug, it's a drug response. So you can, and it's well known, and many people now like called M-AHEAD, it's a bigger NH-funded AI network. It's a specific focus on ethnic background, and the genetic variation is well known. It's an ethnic different. So in like breast cancer, many mutations are Asian specific. So if you put this AI model in, you can better predict the drug response only in Asian population, but not in other population. I think this is very important and it's doable. Yeah.
All right. Thank you very much. All right. Moving a little bit closer to home with Dr. Mitra, who I've actually gotten to know a little bit through interaction in the Drug Resistance Sensitivity Network, now ARTNet. So he's going to talk to us about omics and data science. He basically has a lot of interest really about everything we've talked about today, and will give us his perspective.
Thank you so much for the opportunity. I'd like this opportunity actually to introduce my lab and what we have been doing, more close towards drug development than actually drug discovery, using both dry lab and wet lab methods. Basically, we combine bulk and single cell omics with wet lab to do what we do, and using AI in the process for the last 10 years. My primary focus in the lab is heme malignancies, multiple myeloma, mantle cell lymphoma, although we have recently expanded to, with Dr. Arnold and other groups, to solid tumors. And because multiple myeloma is a blood disorder, immunity is a blood disorder, so we have been also working with the National COVID-19 Collaborative, where I'm leading the myeloma group there. So today I'll be more focusing on heme malignancies and what we have done so far with that. The research focus and the focus of the talk today, and I'll rush through most of the slides and give you a brief overview of what we have done and what we are currently doing. And it will be more about the target disease, or the example in here would be myeloma, and the target drug or primary drug, proteasome inhibitors. So, and I'll have three different parts or three focus areas, which I'll explain one by one later. So, brief overview of multiple myeloma. It's a blood cancer. It's a B-cell malignancy. It's the second most common hematologic malignancy. It's a cancer of the antibody-producing plasma cells. It's a bone disease, as I said. So there are The symptoms are classified as myeloma-defining events, and they are called CRAB symptoms: hypercalcemia, renal insufficiency, anemia, and bone lesions. More importantly, it's mostly an incurable disease, although it's a cancer with one of the highest number of approved drugs, including CAR T therapies, including a lot of targeted therapies in the last few years. It's still, the 5-year survival ranges at below 60%. So our idea was to first understand the genetic basis of drug resistance, and then get to, from this pharmacogenomics, to predicting secondary therapies. So the first focus area when I joined the myeloma pharmacogenomics lab in University of Minnesota was, can we use genetic information to predict, not just stratify, but to predict between tumor heterogeneity in therapeutic response? In 2014, 2015, using AI was not considered the first to-go option. It was more about identifying what went up and what went down in a gene expression profile. But I wanted to understand what actually is the inter-tumor or between tumor heterogeneity and what drives that. So at that point of time, what we started doing was accumulating human myeloma cell lines. And at this moment in the lab, we have more than 50 human myeloma cell lines representing the diversity of response, chromosomal aberrations, cytogenetics. And what I did, or my group did, was to generate cytotoxicity profile to evaluate the response of anti-myeloma drugs. Here we did it on 4 different proteasome inhibitors: bortezomib, carfilzomib, ixazomib, and aprozomib. And what we found out, and this is a dose response curve for all those 4 drugs, and each curve represents a single cell line. And you can see there's a wide inter-individual variation in the dose, in the response to these drugs in terms of % survival values, which it's also reflected in the IC50 range, where you can see the best responders, which is represented by the lowest IC50, and the worst responders are 50-fold different from each other in terms of the response for all these drugs. What we did then was we took the extraordinary responders, the top and the bottom responders, and did RNA sequencing and derived a 42 gene signature based on, at that point of time, as I said, those were new. It was in 2015, 2016, random survival forest and other machine learning algorithms, and then validated that with SOMA's DXY rank correlation. And what this 42 gene signature did was not just to stratify response, but also predict a good response cluster and a poor response cluster. And then what we did was we took clinical trial data from 3 different clinical trials. Millennium is a company which is now owned by Takeda. It is a company that developed bortezomib. And then we had a European trial, the HOVON trial, and then MRF trial, which is a COMPASS trial. And wherever bortezomib was in the arm, we could actually predict the survival between the good response and the bad response clusters in all the 3 trials. Next question was, is this signature survival specific for bortezomib, or is just a random signature for tumor aggressiveness? So we also looked at the other arm of each of these trials, the dexamethasone arm for Millennium trial, the VAD arm, which did not have bortezomib, for the European trial, and the same in the MRF trial. And we could see that there was no stratification in either of these trials, so which means that the 42 gene signature was bortezomib treatment specific. So this was my first venture into AI, a little bit of AI, but where we could predict progression-free survival using a gene signature, predictive gene signature. And what we then did was we reached out to our collaborators in Mayo Clinic, and they were running a prospective trial on proteasome inhibitors. What we could do is we could then predict the good response and the bad response clusters, and showed that anyone who was in the poor response cluster died, many of them died, and whereas none of the predicted good response cluster participants died in the course of the observations. The next question was acquired resistance, and I guess Dr. Willie recently just mentioned about ART. That's a recent focus of NIH, is not just resistance, but acquired resistance to therapy. So from the cell lines that we had, we generated some clonally derived drug-resistant cell lines from the parental cell lines, the Rose escalation. So basically, this cell line is the parental cell line. Yeah, this cell line is the parental cell line, which was, and this one was derived from this cell line following a year of continuous drug treatment. And you can see that this cell line, the resistant cell line, is 20-fold more resistant than the parental cell line. So the idea in here was that these residual cells, which were not killed at the beginning of the treatment, might have resulted in a resistant outgrowth, and this cell line was generated. So our hypothesis was that the initial tumor, myeloma tumor, had a large amount of sensitive cells and a few resistant subclones. When we keep treating them, the sensitive subclones will be killed, and the resistant subclones would outgrow and form the dominant subclone in the tumor. But how do we predict this? How can we get to a level where we can predict this residual resistant cell in a pre-existing environment of a sensitive cell line or a tumor? So the second focus area that we had was, can we use genomic information to predict within tumor or within individual or intratumor or subclonal heterogeneity in drug response? So basically, can we predict these subclones within a premature or primarily sensitive tumor? So we were using single cell analysis. Again, this is not 2020. This is not 2024. We started doing all that in 2014 and 2015. Using AI at that point of time was not considered very normal in single cell analysis. There was no single cell analysis software or tool to do any kind of single cell analysis. Fluidigm, the company which introduced one of the first single cell systems, they paid us to do the analysis and run their first CUI autopref system. We did that. Following that, we also did 10x single cell genomics analysis, which is pretty standard nowadays. And then what we created was a software algorithm. So it's an algorithm for the classification, prediction, and quantification of drug sensitivity of single cells. So the algorithm, what it does is it filters missing data, performs gene selection. It predicts drug response of individual cells, intratumor heterogeneity based on the top genes using machine learning algorithms. It does feature selection. It also does resistance prediction within single cells. So what it does is it derives a SCAT score for individual score. A SCAT score is a prediction of PI response or PI resistance. It used a random forest, support vector machine. At that point of time, the most common machine learning methods and triangulates that to generate that score. It's universally applicable, so we have later applied that not only to multiple myeloma, but for other cancers as well. So when we use the SCAT score system or the SCATome software to the extreme responders, both extreme sensitive and extreme resistant cell lines, representing the individual variations in PI treatment in these 11 cell lines, this is what we got. On the x-axis, you can see the cell lines in the increasing order of the IC50 values. On the y-axis, it is SCAT score. And then what's generally observed from this is that the median SCAT score was highly correlated to the IC50 values. But more importantly, if you can see in here, in the sensitive lines, there would be some, and each dot in here represents a single cell. In the sensitive lines, you would see some resistant cells, whereas in the resistant cells lines, you would also see some sensitive cells, which would not be predicted otherwise using a bulk RNA sequencing method or just a clustering method without a prediction of the sensitivity of the cells. We also showed that individual samples, individual tumors with number of cells higher than a SCAT score of 35, they would respond very poorly to the drug. So then we applied that to myeloma patient samples. What we found out that, and again, these are prospective clinical trial, drug knife prospective multiple myeloma clinical trial patients where we did a single cell analysis, RNA sequencing. What we could show in here is there is a wide range of responses among this. We also could show, based on our prediction, that the number of responders and the non-responders for PI therapy based on a SCAT score greater than 35 And what we can also show is any patient who had a SCAT score, higher number of single cells with a SCAT score greater than 35 died eventually. And this is what we got from a variable gene selection. We could show that whatever you could predict with like thousands of genes, we could also do that with the first top 8 genes, and we got a more than 95% prediction with that. When we looked more into that, among these top 8 genes, some of these genes were previously implicated in myeloma, and the rest of them were proteasome subunits. Proteasome, again, is the target of proteasome inhibitors. We ran gene selection using lasso, the linear method, and we could also see there's an overlap between these. These two are 2 different methods. One considers genes as independent variables, another considers genes as dependent variables. However, we could see considerable overlap between these 8 genes. The final question that we had, and which we are now pursuing in the lab, is say a patient has just one option for having a one drug, and single cell analysis predicts that that drug would not work on that person. Then what do we tell the patient? Go home, there's no drug for you, sorry. Or can we do something about that? And not just a single drug, because no clinical trial nowadays does a single drug analysis or single drug treatment. That's most of them is combination therapy. And again, a combination therapy, generally, you can go to a doctor and tell the doctor to give 4 new drugs or just eliminate the first drug and give a new one. So I'll explain what we're trying to say here is, can we develop an algorithm to predict alternative therapies in drug-resistant myeloma? Primarily, can we develop an algorithm to find secondary drugs which can combine with the primary drug to increase efficacy? So consider this all as drug-resistant tumors. So there would be drugs which can, say, kill 50% of the resistant tumors. Another drug that can also kill 50%. Can you add these two and get 100% kill? Can you actually consider this as, say, subclones, and then each resistant cell, or consider each resistant cell as component of this subclone? Can we have a combination of drugs that kills all subclones by killing by having one drug killing each subclone, right? But if you have, say, drug 1 and drug 8, although they're 50% each, their total coverage would not be more than 60%. There might be drugs like drug 7, which might be like a 20% coverage, but if you add drug 1 and drug 7, you might get a 70% coverage compared to a drug 1 and a drug 8. That's the reason behind this is the drug sensitivities overlap, right? So can you create an algorithm that takes into account this intersection among the drugs? So ideally, what we want is, we want to find an alternative set of drugs that kills all the resistant cell lines when used as combination, like this, right? So can we formulate something using AI or using some other tool to get to this, rather than looking into each and then placing one at a time? It's almost like counting money, right? How do you count $6.35? Can you use 635 nickels? Or can you actually do something else with that? Because if you use an analogy of this one, you cannot give someone 635 drugs. So the best solution for this, with the least number of bills or coins without overpaying, is having a $5 and $1, 25 cents and a dime. Okay, so how do you do that? You do that using something called a greedy algorithm. So basically, what we created was an algorithm, which is a modified greedy optimum set coverage algorithm for the prediction of a secondary drug in PI-resistant myeloma. Again, this is universally applicable. You don't consider PI-resistant myeloma, consider some other cancer and the primary drug. So it can work against any primary drug, against any cancer, and just not give you the secondary candidate, but also give you the prediction of the secondary drug combinations. Okay, again, we used the GDSC database, which was just explained, and right now it has expanded to include about more than 1,000 cell lines and more than 600 drugs. This is the algorithm. I'll not explain too much into that, but again, remember, you can kill 100% of the resistant cells, but then you might need 20 drugs to do that. So there has to be a point where you get a desirable amount of coverage, but don't get more than 3 drug combinations. because then you cannot go to a doctor and say, oh, this drug doesn't work on this patient, give them 20 more. You have to be reasonable in convincing a doctor. So what we did was, it was the optimization in here was followed by regularization. We asked software or asked the algorithm that once you reach 80% coverage, then if a drug adds just 1 or 2% more, don't take the drug, stop there. So it's an optimization, regularization algorithm. That's why you have regularization at the end of it. These were the top 10 combinations in with PI. Again, it's with the primary drug proteasome inhibitor. These are the top 10 combinations that we found out. And this is how we approach the validation part of it. Again, as I said, ours is not just a dry lab, it's a dry and a wet lab. So we first predict, and then what we do is we perform single-cell RNA sequencing. Then we perform to understand and screen the drugs, to perform in vitro validation, to test the drugs. Then we do a bulk and single RNA sequencing pre versus post-treatment to understand the pathways and genes that are regulated by these drugs, and to understand the biomarkers. Finally, we collaborate with Mayo Clinic to perform ex vivo validation in primary bone marrow samples from patients and see if the drugs do work in patient samples. And then we do functional validation analysis using various techniques. Just I'll brush through this. This is all the top seq drugs as single agent. They do work in myeloma patients. Then what we did was a single-cell RNA sequencing as a seq drug screening tool. So basically, this is a sensitive line and a resistant line. What we showed is the sensitive line had higher expression of HSP90A and B, as well as NA and PT, which are the targets of these two drugs, 17-AAG, which was just explained, and FK866. When combined, we could show there are multiple single cells within these two cell lines where there was an enrichment of these drug targets, which gives us a fair amount of idea. If you use these drugs as combination, they'll hit the targets. And again, this is a combination therapy study, which firstly shows the combination of 17-AG with proteasome inhibitors and shows a very low combination index CI, which means synergy and very high level of synergy. Because of scarcity of time, I'm showing, but 17-AG also showed synergy with IMiDs, which is a standard of therapy for multiple myeloma. Basically, the most standard therapy for multiple myeloma is VRD. V is for Velcade, which is a proteasome inhibitor. R is for Revlimid, which is an IMiD, and D is for dexamethasone. So, and FK866 also showed synergy with 17-AG. Then, as I said, we did bulk RNA sequencing, pre versus post, to understand top genes and networks associated with response to the seq drug. And that's a standard practice for most seq drugs that we work on. We also did pre versus post single-cell RNA sequencing to see which clusters change from pre to post, and then trying to identify biomarkers at the subclonal levels. Then we went back to the patient primary samples and identified what happens ex vivo. We see the desirable effects ex vivo in myeloma patient-derived CD138 positive samples, as well as direct bone marrow samples, B cells and T cells. We could see that elevated caspase-3 levels, which is apoptosis, and then also downregulation of genes associated with myeloma cell survival. We could also see newer drug targets, as well as, genes, which gave us insights into the functional aspects of the seq drug treatment, downregulation of Icaros, ILOs, RF4. And this is customary to any wet lab experiment. You do a lot of functional validation, Western blots, is a knockdown, CRISPR knockdowns, flow cytometry, as well as we showed that 17-AG impacts superoxide levels, intracellular ROS generation, as well as mitochondrial membrane potential. Finally, this is an interesting thing to show that this is our NIH network, DRSN, Drug Resistance Sensitivity Network. These were the 3 projects involved, one with my group in here at Auburn and Mayo Clinic, Rochester, another with Mayo Clinic, Arizona. And Mayo Clinic, Arizona, were doing an experimental direct-to-drug screening. We were doing a computational greedy algorithm followed by wet lab. So basically, our wet lab was minimized to the computational results, whereas they did a direct-to-drug screening. So what we could generate in 3 months, they also generate a great amount of good deal of data in 2.5 years. And we get to the same drugs at the end of it, which were then validated in mouse models of responses, which are systemic multiple myeloma models. At this moment of time, one of those drugs, PIK5 inhibitor, is in clinical trial for multiple myeloma. This is another drug that we are currently working on. It's another Seq drug. I call them all of them Seq drugs, which was presented at ASH. We are working on that, and there are multiple Seq drugs which each of the students is now working on. Finally, now because Seq drug worked somewhat in multiple myeloma, we are reapplying that in other cancer models like prostate cancer in here, as well as breast cancer. So yeah, collaborations are welcome. We are validating the prostate cancer work in MCRPC patients using single-cell multi-omic analysis. That's all, and thank you so much. These are our collaborators, including Dr. Arnold sitting here. Thank you so much.
I may have time for a question. Okay.
What are your thoughts about ability to decrease concentration of cancer drugs so we still keep them functional but decrease chance of resistance? And also in terms of sequence and regimen, certain drugs in ovarian cancer like PARP inhibitors are used for 2 years. Do you think we should go constantly for 2 years, or on and off will be a better approach to decrease resistance?
Thank you. I just went back to the slide because the DRI in here means dose reduction index. So to answer your first part of the question was that DRI would mean you could decrease the dose by that fold, the primary drug, and then add, if you add the secondary drug. So I think that answers your first question is, if you add these drugs, then all the DRI values are the fold amounts you could change the primary drug to. So you could still keep the primary drug. Maybe it is more efficacious at that point of time too. But if it causes toxicity, bortezomib has a lot of toxicity, xazomib has toxicity. So it decreases that. The second one is
Metronomic therapy.
Like a metronomic therapy, I guess. I think Dr. Arnold was mentioning his poster outside talks about that. Is there is an evidence of metronomic therapy. When we went to the OHSU for a DRSN meeting, they hired a director from MD Anderson who was actually mentioning about metronomic therapy. And there is a lot of interest in that. But again, like you said, if you decrease and increase and the doses, there is also a fear of developing resistance. So is there a critical limit for that? Is there a limit where it develops resistance or it develops more sensitivity? If that maybe an AI technology can find the critical limit of developing resistance and developing sensitivity, that would be great. I think there's a question.
Maybe a shorter question. That's really wonderful presentation. Congratulations for such kind of very impressive program. So my question is, for the single-cell proteomic data you generate, two related questions. First, did you use similar sample for the single-cell RNA-seq and you see the consistency? The second question is for the single-cell proteomic data, which usually is very difficult to generate. How many proteins roughly?
Yeah, so it's a single cell level proteomics. Basically, it's a CyTOF. So about 40 to 100 targeted proteins are generally done. If you're doing CyTOF, it's around 38 to 40. If you're doing Aqua Biosciences like PhenoCycler, it's 100. And the first question that you had is, have we actually corroborated the data? We have recently done that. We have recently identified cell types using single cell analysis. And with the same sample, we have done proteomic analysis too, like using CyTOF. It was recently presented at the blood cancer meeting last year. And we could show there's a synergy between the number of the classification we have done. Thank you.
All right. And now, saving best for last here. Let's get to it. Oops. Is this you? Yeah. So Robert Reynolds has, I guess, a nice long career here in Birmingham, both industry, pseudo industry, and and academics. So I always kind of feel like having Southern Research is like, we have our nice little pharma company right in town, twice as old as UAB, and help develop drugs back in the 30s using mice, probably for the first time, I think, for most investigators in the US. But anyways, we'll talk about a really hot topic now, this targeted degradation. And I'm excited to hear about this.
Thank you, Chris. I moved to Southern Research, or from Southern Research to UAB in the 2012 timeframe, and then moved from chemistry at UAB to Hem-Onc in about 2017. And in that time frame, I decided I would reinvent myself a little and move towards things that looked a little more exciting than standard mycobacterial drug discovery. So I started looking into PROTACs. And one thing that I discovered fairly early on, even though the field had advanced quite a bit, is that there appeared to be very little activity at UAB in terms of education, research, or in the clinic. So I thought it would be a good way to develop my skills at UAB, particularly in Hem-Onc. Pardon? What advances the slides here? The arrows aren't doing it, and the oh, sorry.
Should click now.
Okay, thank you. So in lieu of an outline, since this is so short, I thought I would adapt this figure from Craig Cruz's paper in 2022. It's an excellent paper. It's very PROTAC biased, which is a term coined by Craig Cruz at Yale. He developed what I would consider the heterobifunctional PROTACs technology, and then licensed that technology to Arvinas. So you see PROTACs throughout the timeline, and then ARV this and ARV that. Those are Arvinas drugs. What this talk will focus on is a little bit what I consider pre-PROTAC technologies. Then I'll talk about PROTACs, pros and cons, mechanisms, and whatnot. And then I will advance on to newer directions that came since PROTAC development. So this is a very simplistic drug inhibition schematic, in that you see a drug or inhibitor competing for a protein target with a substrate. When the inhibitor is bound, that's when you see inhibition, and that is considered occupancy-driven inhibition. That is opposed to a PROTAC, and the functional unit for a PROTAC is a heterobifunctional targeted protein degrader that consists of a drug much like in that last slide, linked to an E3 ligase ligand that recruits the E3 complex for protein degradation. And so you see there is some reversibility of the drug binding like in the last slide, but when a ternary complex forms of the E3 ligase system, when it is recruited by that ligand to the drug-bound protein, it's a fairly intimate complex that rapidly leads to ubiquitination, polyubiquitination of the protein target, recruitment to the 26S proteasome, and degradation. So you will hear the terms or the comparison of event-based degraders versus occupancy-based degraders. What did I do here now? Okay. Yeah, these slides are showing up a little different. I've noticed throughout these talks on this system for some reason. But anyway, what this slide shows is a schematic of a typical colon ring ligase system where the protein of interest is recruited by a heterobifunctional degrader having a protein binding or target drug and then a ligase recruiting piece of that degrader. And you form this complex that what is crucial here, and Cruz warns about going after proteins that have lysine deserts, is you have to have surface lysine residues in the ubiquitination zone or a degrader will not work. So this is one of the grants that we were awarded in around the 2019 timeframe after I got into this. And the point of this grant is to design degraders against HIV reverse transcriptase. As part of the grant, the group that I work with, the computational group at the University of Miami, modeled this complex, and you will see that it is the reverse transcriptase linked through a heterobifunctional degrader to the E3 ligase system. And this is rilpivirine, this is the linker, and then this is a pomalidomide or a cereblon recruiter. The first thing that's important that you have to understand or is important to the function of a targeted degrader is that you can abrogate resistance. It's one of the benefits. And the reason that you see that is in this case where you just have the drug itself bound to reverse transcriptase, this surface area of interaction is what defines the actual binding and the inhibition, but it also defines where resistance will develop. And it is a very tight area around that binding site specifically. Now, what is important and why I mentioned that it is a very intimate complex with the E3 ligase system is you get a huge surface area of interaction now that helps drive the degrader down towards, or the TPD itself down towards degradation rather than having reversibility like a drug. And those additional interactions actually define the reversibility, but they can also define selectivity in the case of a promiscuous warhead. So in the kinase field, you saw in an earlier talk, some very broadly active kinase inhibitors. You find if you make a degrader out of one of those promiscuous kinase inhibitors, then in fact, it doesn't inhibit all those targets anymore. Because of this interface, not all interfaces are equal. And that's where you can get selectivity out of a degrader. So in terms of pros and cons, I've talked about some of these. Since the degrader itself has the drug bound onto it, you will see simple drug inhibition because it's binding the target. But then at the same time, I've discussed the fact that these are event-based and you get degradation as a result of recruiting the E3 ligase system. Degradation of the target is absolute, and so that's an entirely different system, event-based than occupancy-based. And then, of course, there's potential significant selectivity both at the target and the tissue for some reasons I've discussed. And then, of course, because of the fact that you form that heterobifunctional E3 complex, target E3 complex, you can abrogate some of the resistance issues because typically resistance is not absolute. It's piecewise or mutation-wise until all those mutations compile to get very high resistance. And what happens with a degrader is those can still bind because the mutations don't define absolute resistance, and those will lead to downstream degradation, and it will be biased towards that because of that E3 complex interface where the resistance doesn't show up. There's some consideration that TPDs may expand the undruggable protein portfolio. And then what is also important for these is many proteins, kinases, for example, can be part of multi-protein complexes, and they have a scaffolding role. And an inhibitor will not affect the scaffolding role, but a degrader, by degrading that protein, will do that. Now, it's important to note that in a multi-protein complex, you can actually end up with a ligase sitting on a face and putting lysines on associated bystander proteins in that complex. And that is considered collateral damage because you can lead to non-target or targets you're not interested in actually being not only inhibited but degraded. Now, if you're looking at something like antiviral targeted protein degrader and you're interested in antiviral drug design, that could be a benefit. These compounds are very complex. Many of them are chemical. I won't go through them specifically, except to say that these are high molecular weight, beyond rule of 5 drugs typically. They're in this range, and that leads to a number of potential problems, including bioavailability, dosing amounts, penetration, metabolism, and clearance. And then, of course, they're more challenging to make and more costly to make. And then on top of that, the biology and the mechanism, proof of mechanism of action is also much more complex. I have discussed some of these issues in terms of what makes a good target, avoiding lysine deserts, for example, and I won't go much into those. I've mentioned resistance. The argument is, and it has been shown, that resistance is less likely with a degrader. It is not absolutely stopped, however. And in fact, at least in vitro at this point, downgrading the target or the E3 ligase can actually lead to resistance, significantly reduced warhead binding. So even though mutations can impact, or you can still get degradation of mutated targets, you can compile enough in a specific cell to lead to resistance. And then, of course, because these are large lipophilic compounds, typically, they can be substrates for transporters, MDR transporters. So now I'd like to segue into some of the technologies that are considered degraders. Most of the earlier discussion was about PROTACs or heterobifunctional degraders. This panel on the left actually shows a timeline for what are considered molecular glues. These are compounds that are typically smaller molecular weight. They were discovered out of the standard drug portfolio that have an activity of when they bind a target, they lead to recruitment of the proteasomal system and destruction. These are not the two-part with a linker. They're simple small molecules that for some reason have this activity biologically. Many of these, as I said, came out of known drugs, and the activity was discovered in the 1990s. PROTACs is considered around 2000 when Craig Crews coined the term. And of course, later compounds, and some of these you'll recognize, are very small molecular weight. And they are actually typical small molecule drugs that have this activity. As I've said, these are usually, or these have been up to now, fortuitously discovered. And because of that, they typically don't know the mechanisms very well, which makes these particularly hard to optimize. And that's a problem with molecular glues. They're great compounds, but they can be tough to optimize. And then, of course, now, and actually Stuart Schreiber, who published that panel on the left in Cell, has recently published an assay where he designed a screen to discover glues and found some. It's a 2024 publication, I believe. But there's a lot of interest in these molecules because they have advantage over targeted degraders in terms of size and bioavailability. And that is the last point on this slide. some other targeted, I call them TACs because they're not PROTACs, they're targeting chimeras that can do a wide variety of biology different than what you would consider a PROTAC. You can for example, target membrane targets and drive those towards lysosomal degradation. So that's a different system than the proteasome. You can actually bind these same types of targets with ATTACs or ATTACs and drive them to the autophagosome and the autolysosome. So you're starting to see post-PROTACs new developments in the TAC area that use different biology, and they don't rely on one type of biology. Now here is an entirely different recruiter degrader that actually directly targets RNA. And this is called ribotax or pinads more recently. And what you have is a system that looks like this that then leads to downstream degradation of RNA. And that is particularly of interest it's been driven by Matt Patricelli at Scripps, and he is particularly interested in antiviral RNAs, for example. So that's an application of that technology. Okay, what have I done here? Now, and again, I'm pointing these things out as different degrader technologies, but at the same time, depending on your target and the biology you are interested in, you may not want to do a typical PROTAC. And there are a lot of variants that are interesting. If your protein relies on phosphorylation or dephosphorylation for its activity, you can now design degraders that actually recruit a phosphorylase. So it will dephosphorylate your target and change its downstream biology. You can also recruit a kinase in a degrader and actually, or I call it a degrader attack, that will recruit a kinase and lead that to downstream phosphorylation. That phosphorylation can drive degradation, but it can drive other biology as well. And these are being looked at primarily now for chemical biology tools, but they will be useful in the drug market later on. Another interesting variation in a heterobifunctional TAC is recruiting a deubiquitinase rather than a ubiquitinator. And what that does, and it's seen specific application in cystic fibrosis and P53, which is an interesting cancer target, in that both of those, in those cases, you can end up with ubiquitination and degradation that causes downstream pathologies. And by recruiting a deubiquitinase, you remove the ubiquitins, and you can actually end up having a functional stabilized protein. And finally, the last of these examples, and one that I'm particularly looking at, is what I call a heterobifunctional TAC or degrader. Instead of recruiting a specific E3 ligase, you can actually make ones that recruit HSP90. And this technology has been advanced primarily by Rannoch Therapeutics. If you go to their website, they've got some interesting information on the website. They now have a compound that's going into clinical trials. More recently, a general paper has been published by Lee et al., and these are referred to as heme TACs. The advantage that this approach has is that along with HSP90, it carries about 1/3 of the human E3 ligase systems, which means you don't need to make multiple TACs with multiple E3 ligase recruiters, and you can end up with more widely varying tissue activities depending on the E3 ligases that are available. That is just a small subset of all of the potential attacks out there. I could talk for days and days, but they're not going to let me do that. So I cut them all out. But I wanted to sort of end all of that tax stuff with the fact that although you hear about PROTACs, because it's the most advanced and it's an elegant chemical biology tool, and a lot of compounds are going into the clinic that I'll show later based on that technology. But all of these tacs are in fact what's known as combi molecules. And that phrase, if you look in the literature, showed up post-PROTAC around 2003. But in fact, one of the first projects that I worked on was making molecules, not making them, but actually designing molecules based on hormone receptors. And we made molecules, or we conceived of molecules like this that would impact homodimerization and heterodimerization of hormone receptors, for example. And that was back in the late 80s in a design project with UAB when I was at Southern. So this technology has been around a long time. You can envision a wide variety of ligands that are effectors in a wide variety of proteins, as well as oligonucleotides, through a linker recruiting a wide variety of other biological activities that can be effectors against these targets that you're looking at. And again, depending on the biology you're interested in, and depending on the outcome that you'd like to interrogate or actually derive a drug for, there is I would say that there is a lot of room for evolution of this technology towards other biology that people should consider. There we are. So anyway, I have sorry, folks, having a hard time with this. I've discussed a lot of the pros and cons. These are huge molecules. There are potential problems with them. And the medchem purists, when this first was proposed, were very negative on it. And there were a lot of blogs. Derek Lowe's blog, for example, came out and said, why are we even doing this? Well, this is the bottom line. This is as of 21, there are even more than this. You can see the ARV compounds through our Arvinus are actually the most advanced, and they've advanced even further since this paper reported these. And what is actually interesting is the negativity was way overblown, because Arvinus has said, if you listen to Craig Crew's talks, that they will only target and make compounds that are orally available. So these are orally available, very large molecules, and they're actually working in the clinic. And that's the bottom line. And I think at UAB, the paucity of what is going on in the clinic in this area with the expansion and I'd say the explosion of this technology, that it ought to be looked at for recruitment into clinical trials more. And finally, since I added an extra slide to an older talk I had, since we're in an AI time, and the point of this is AI, with the growth in PROTACs, and you will notice that these are all applications to PROTACs, but there are potentially many more tax technologies out there that I think the field is wide open for AI development, computational developments and AI developments. And I don't think a lot of that is going on here at UAB either. And I think just from a basic biology, interrogating E3 ligases and interfaces with proteins, there's just a lot of room that this work could be done. And I'd love to see more of it here and find people I could work with computationally. And so after all of that, and what I think is going on at UAB, and what I think I know is what's going on at UAB, we have started the DIG UAB group. And it's the degrader interest group, and it's now accepting members. So if anybody out there is interested in learning about the field and being a member and contributing, we'd love to hear your thoughts, and contact me and I will add you to the mailing list. Thank you.
All right, questions?
And I think I went on a bit with all those shenanigans. I apologize.
So thank you for this interesting topic. I found out that most of the current projects, they are either recruiting the E3 ligase cerebron or VHL, but not too many activities about recruiting other E3 ligase. So do you think what are some challenges that makes it harder to develop projects recruiting other E3 ligase?
Could you repeat that with them? I can't quite tell it because of the reverberation. So if somebody could tell me exactly what your question was.
So
my hearing aids and they're miserable in this.
So my question is that right now, most of the projects, they are either recruiting cerebron or VHL. So what are some challenges make it harder to develop projects recruiting other E3 ligase besides cerebron and VHL?
So if I understand what you're saying, you're asking about other E3 ligases? And
Yes, challenges for VHL, cerebron, and others.
Yeah, that's something that I had to be expunged from the talk. It is known that there, I think from just sequencing, that there are probably 600 to 700 of these systems in the genome. Clearly, the ones that we saw had tool compounds available. And that's an important part for any of this. You need a recruiter. Thalidomide was well known since the 50s, and its binding activity had been discovered. VHL was developed more recently. And so you will see typically most academic papers, and to get more shots on goal, they will design PROTACs against both of those, or recruiting with both of those. The tools for the other ligases are not as well developed, and I think at this point in the literature, there may be 8, 10, or 12 of those now, where you can actually get a chemical and use it as a recruiter. That is actually one of the things a lot of the biologists are doing, are setting up high throughput screens against these other ligases to discover potential recruiters. So there's a lot of work to go on. The question is, and this is something I don't understand, why one would be better than the other. It seems VHL and von Hippel-Lindau and cereblon are workhorse recruiters. Now, what does that mean from where they are in the tissue and in the various tissues, various cancers, various, like how important they are in virally infected cells? And this is all something that there's a lot of room to do biology in, but you know, on our grants, we had R21s. We couldn't make recruiter systems more than the simple ones. And so I have no experience with those, and you'd have to open the literature up and explore that.
Are there any guiding principles for allowing these to be more orally bioavailable and stable inside the bloodstream?
Again, I can't quite hear you.
Sorry.
There, so.
I just was wondering, you know, if there are some guiding principles to how they can successfully make these stable in the blood and orally bioavailable.
This type of technology, for example, and I didn't really discuss it, it was in my slide, but if you look at antibody drug conjugates, they are heterobifunctional systems. Fluorescent tracers are hetero. So the literature on this linker chemistry has been done for decades and decades, and they typically know what types of linkers will work. PEG is a standard one because drugs typically aren't ideally soluble, and when you link two of them together, it can be bad. PEG is a typical one that has has metabolism issues, but that's usually the linker that people start with. There are amide links, there are alkane links, there are hetero aryl systems. And so there are a wide variety of these have been looked at. You will see actually the more academic papers start with problematic simple linkers, but then if you look at clinicaltrials.gov and you look up the molecules, they're much more complex and exquisitely engineered the way you're saying. But suffice to say, and maybe I should have just said that linkers are out there that do work. And you can see that from the clinical data that Orvinus.
Very nice. All right. I think we'll probably need to move on. I take it.
So I think I'm not enough damage here. Thank you.
Thank you, Chris. I know it's late, so we are stopping people from having fun. So we'll be as fast as we can. So we should all be staring at Amy. She holds the secret to who will win the poster awards. I pledge that I did not have anything to do with it, and I will be heavily conflicted if I were. So do we need to use your computer or? Oh, okay. All right, then we don't want to disclose the winner before you announce it, right? Okay.
Sure.
All
Well,
right.
the judges have been, I think this mic, right? That's right. The judges have been heavily working through the evening gown and talent competition. So now we have our finalists, and then now we have our announcing our winners. Also, housekeeping before I do that. Did somebody lose an Apple laptop charger outside? Good, because I know these things are not cheap. So thank you. All right, so with that, I know we're keeping you from food and fun. So, all right, so we're going to have our poster session. We had 13 posters, which is a record. That's probably like twice as many as we had in the past, and we actually had to divide them up into 2 sections as opposed to last year. So thank you for all that. I also want to thank everybody who contributed to it. Now, not only the people who made the posters and the mentors, but also all the logistics, and people had to get all the the poster boards and the space and everything. And I think they're putting those boards away now as we speak. And not to mention everybody else who assisted at every phase of that. And also our judges. Our judges really helped out stepping up to make sure that we have a good evaluation. Also, the presentations are, thank you very much for all these posters, that they were all, we were surprised how good they were. And they covered all different aspects, different kinds of cancers, different processes, different phases in the treatment, diagnosis, treatment, drug discovery. And, so it's very, very interdisciplinary, multidisciplinary. So without any further ado, the awards. So 3rd place goes to Christina Lee. Actually, I don't have the title of it, but and I know the judges were really asking a lot of questions there. So congratulations. And
where's the question?
Yeah, if you just come down real quick. Hopefully anybody's still here. You can come down. Oops. Yeah. Oops, that was slideshow. Where's that?
Can you take like photos?
Oh, here. Okay.
Well, we don't have the real certificate.
This is the certificate. It's like I didn't
Okay.
print it. Yeah, but
So maybe what we'll do is we'll take
pictures. Yep.
Yeah.
I can.
Okay. So
Congrats. Here's a little
yeah.
special gift.
Okay. All right. So that means that really.
Actually, let me get in there too.
All right. Congratulations.
Second place goes to Deepal Shah. I know this was also a big collaborative effect. I mean, collaborative efforts.
Oh, congratulations. I know you came all the way from Finland. Right? Oh, Well, that this matching color, that's
Complimentary.
Yeah.
Finally, drum roll. First prize goes to Taraswi Mitra Ghosh. I think you're also joined up with Robert Arnold.
Wow.
I don't know if anybody's still here or can accept that on
all right.
behalf.
Wow, congratulations. Yeah. Where's your student? Let's see. Oh, okay. She's at Harvard. No wonder. Wow. It's great.
I know all of these are joint efforts, so hopefully.
Thank you very much.
Then, so congrats to all award winners, and we hope to see you next year, even bigger and better. Right, and I'll turn it back over to Jake.
All right. Well, I think as with all the great meetings that were coming to an end, I want to thank all of these organizers again. They are program committee members and people, staff members, and even student volunteers. Don't you agree that they've done a great job? All right, let's give them a round of applause then. Especially Jendel, right? So that was such a professional, fantastic job. And also, I think a meeting like this, and it wouldn't be possible without the sponsors, and each one of them contributed fund. I think some of them are here. I think I see Teresa standing in the back and representing the pharmacology department, cancer center, CCTS, and they all contributed. We're really grateful, and they're giving us the food that's important. And
in the lab where data flows, revolutions underway.
Yeah, a little trivia about this poster. So I wonder who know what this origin of this poster come from. Raise your hand if you know. There's a movie actually. Anyone knows?
That was young Jake Chan.
Yeah. Yeah, in addition to that, right? That's actually the futuristic Jay Chen. I would have to fuse with a robot that Alex made in order to compete. No, so actually this is a movie called When Harry Meets Sally. Yeah, so it's a classic. It talks about a guy who graduated from University of Chicago, met a very beautiful girl, but their life take many different twists and turns. They met and have separate lives and eventually come together. Right? So that's just assess how difficult life can be. So that probably is an allusion to my main message. It doesn't matter whether AI is a hype or hope, but I think what is important is that many people think that AI is essential, whether it's a revolutionary to take the lead for drug discovery or just embedded. And it's gonna be an up and down battle, right? So we're here, we really want to appreciate that. And then one final thing, I was told that we had to actually do a 3-question survey. So I mean, it's very quick. I wonder if you can just quickly fill out these 3-question survey. What did you enjoy most about today's event? And then they're all very quick ones, so they're not gonna hurt your brain by answering that as if it's in the panel. This is really amazing to see the things changes in real life. Research session. This is really surprising. In the past, we always thought that the networking or the keynote was better, but that simply showcase how diverse the presenter are and how good you are. Especially we have people from out of town, Michigan State. Actually, Alex also came from, graduated from Michigan State. Yeah. He got a master from Hopkins, but a Ph.D. from Michigan State. And all right, well, currently the research session is a winner. Let's go to the next one. What are some things that you have learned in attending ATTIS today? At least the AI, what AI stands for. Wow. Yeah, Zhongming, that you have introduced the keywords and someone else have learned. Drop everything and get into AI drug discovery. Yes, I think that's what Alex said. But his original words was to drop everything and get into generative AI. So. drug target tag. Thank you. Yeah, the last, it's I think that's also doesn't mean it's necessarily the least priority. PharmAlchemy. Yeah, the PharmAlchemy team. You are like a molecular glue, and then you always glue together the whole day today. That shows the teamwork and the good product. And did you actually cheat by just saying PharmAlchemy yourself? No? Okay. All right. I think let's move on to the last one. What are some topics you would like to see more at our next ATTIS? So ATTIS has been going on for 8 years, and every year we are finding different topic. We want to chase not the long-term bubble, but something that is up and coming, and in short term, and it's going to make impact. And let's see. Tool demonstration, more diverse data integration, aging research. That's interesting. I want that. From AI to wet lab, novel disease models, path to personalized medicine, AI in large molecule research, how to implement. I think a lot of them is a very practical topic, it sounds like. Applying AI to clinical setting, clinical trials. In fact, we did have themes that talk about the clinical informatics, clinical decision making before. Co-training. Oh, for co-training, in fact, Professor Amy Wang has been in charge of the hackathon every year. Hackathon usually takes place in the summer, the end of the summer in August. And that's normally a month-long training of coding followed by a combination of a competition for 2 days. We don't know what will happen this year, but maybe we'll bridge to use a bridge to AI data set, or maybe we'll find something else. Yeah. So my suggestion is keep in touch. If you are interested, then get in touch with us through the mailing list, through the Slack channels, and then we'll have great program coming up in the future. With that, I want to thank all our speakers and every one of you. You lasted till the very end. This is Friday, and let the party begins. All right. Thank you.
Well, thank you again for the invite. Well, sorry for the tech note.